Hello students, welcome to Vedantu NEET English. I am Miss Gopika, your own biology master teacher. Please let me know if I am audible and visible to all of you. Come on. Yes, ma'am came, ma'am came. Quickly let me know if I am audible and visible to all of you. Yes, are we live? Just refresh once. Yes, we are live. Awesome. Perfectly audible. Please let me know if I am audible and visible to all of you. Perfect. Perfect, okay. Yes. Just letting others also know that we started. Okay, just give me a minute. I'm so sorry. We had to set up everything. Yes, done. Done. Hello, hello. Okay, let's start, students. Okay, awesome. Awesome. Let's not waste any time. Let's directly get into the class. Everything is perfect, right? Everything is perfect. Okay, perfect, ma'am. Pakka. Okay, share the link with all your friends, okay, who are writing NEET with you so that we can have a very good class. So, what are we going to do today? We are going to do human and plant physiology, which has more than 90 marks, okay. Sometimes 3 questions can come from all the human physiology chapters, sometimes 2, 3, like that, a mix of questions can come. So this entire unit is very important, okay. So what we are going to do, we are going to do an entire revision properly. Revision, that means, see, the pace of the class is going to be a little faster from the normal classes, but again, whenever you have a doubt, please let me know in the chat so that I can slow down and I can teach you once again. Okay? Yes, awesome, students. I hope all of you marked your attendance. Quickly smash the like button. That much we can do for our teachers who are going to stand here for 9 to 10 hours. Okay? Yes, very good. I want to see till the end of this class how many of them are going to stay, because those people are the real ones. Okay? You're going to fight with them tomorrow. Let's start with the first chapter, students, breathing and exchange. We will also discuss the weightage of this chapter. This chapter has a weightage of 3 questions. Okay? That is 3 questions can come. That means almost 12 marks. Okay, almost 12 marks from this chapter will be there. So very important. Let's directly get into the what is the major portions that from where the questions are going to come. First portion from where questions usually come is about a connection between animal kingdom and your breathing and exchange. What do you mean, ma'am? See, they ask you the organism and they expect you to write the respiratory organ. So it can come as a match the following. So whenever you know some answer, please continuously put it in the chat box. Take it as a revision, like you're learning with me, you're talking to me. Do not get distracted with anything else. Okay? So look here, students, the first question that can come to you, in lower invertebrates, what is the respiratory organ that we have? So in lower invertebrates like sponges or coelentrates, we can have respiratory organ that is by simple diffusion, because they are very lower organisms. So they will be like, okay, I do not want any complex structure. I will directly use, body surface to take in the exchange of gases. So simple diffusion is what happens in lower invertebrates. Okay, excellent. Okay, next one, students, earthworm. So earthworm is a little more advanced one because it's coming under Annelida. No parking here. Okay, Nereis, Pheretima. So keep continuously practicing that also. Hidden area. Okay, moist cuticle. Again, this people also is like, I do not want extremely something complicated because I am going to be under soil where the moisture content is going to be extremely high. I will use my skin again. I will use my cuticles to exactly observe the things. Okay, clear to all of you? Then students, insects. Insects are going to use tracheal system. So this I have explained you in cockroach, tracheal system. That is, tracheoles will be there, tracheal tube will be there. It will divide into tracheoles, which looks like a hand. All of you remember that. So insects are going to be tracheal system. Amphibians are going to be skin. Amphibians are going to be skin. Now in frog, we have studied 3 types of respiration. What are the 3 types of respiration? All of you quickly let me know in the chat box. So amphibian, students, it is going to be skin. At the same time, we'll also see lungs, we'll also see buccal cavity, right? Buccopharyngeal respiration, cutaneous respiration and pulmonary respiration, right? All of this is going to be present. Now, terrestrial animal, students, again they are going to use lungs as the respiratory organ, specialized vascular system, tube feet or book gills. Okay, tube feet or book gills. Now look here, students, depending on 2 different things, the entire respiration is going to be bothered. What is the first thing? Habitat. Where do you live? Now, if you are living underwater and I'm telling you, you have to respire with lungs, it will not make sense sometimes, right? They'll be like, no, I have gills, I would like to use my gills. And second is level of organization. If they were very primitive organization, there's no point of me giving alveoli, tracheae, bronchi, bronchioles to that primitive organisms. So for that, we will do what? Simple diffusion or so. Clear to all of you? Yes or no? As and when you're understanding a concept, please put thumbs up in the chat so that I also know that, okay, the pace is fine. Okay, I know that the pace is fine. Clear? Okay, now students, whenever a respiration is happening with the help of skin, it is called as cutaneous respiration, and whenever it is happening with the help of lungs, it is called pulmonary respiration. And please remember this in your head for the entire your NEET preparation. Whenever the word lungs come, we use the word pulmonary. Pulmonary. Remember, again, pulmonary circulation when we study in body fluids and circulation. Again, when we hear the word pulmonary vein, artery, please understand, okay, these people have some connection with the lungs. Okay, some connection with the lungs. Clear to all of you? Okay, now going into the NCRT. Okay, most important thing, students, how does the oxygen or carbon dioxide enter into our body and how do they exit our body? This is the thing, right? So if you look at here, if you look at your NCRT, all the green color things that you're seeing is the pathway of how the gases are going to go. Whatever you're seeing in yellow color and pink color are the important points that you have to remember for your, for your NEET examination. Now look here. The air is going to enter first through the external nostrils, okay, first through the external nostrils, opening above the upper lip. So you have your external nostrils. Now from the external nostrils, they are going to enter into a nasal chamber. This is your chamber. It is going to enter into your nasal chamber through the nasal passage, okay. There is a passage through that it is going to enter into the chamber. Now, after that, it is going to open into pharynx. From pharynx, where is it going to open, students? To larynx. Look at the green color only. Look at the green color only. From larynx, where is it going to enter into? It is going to enter into your trachea. Trachea is going to divide into right and left primary bronchi. From bronchi, it is going to enter into the bronchioles. From bronchioles, it is going to enter into the alveoli. Is the flowchart clear for all of you? Okay. Start with your external nostrils to nasal passage. Okay, there's the passage. Through the passage, it is going to enter into the nasal chamber. From nasal chamber, where is it going to enter? To pharynx. Pharynx to larynx, your sound box. Larynx to trachea. Trachea is going to partition to primary and, right and left bronchi. Bronchi to bronchioles. Bronchiole to alveoli. Flow of your air. Clear to all of you? Yes or no? So that is your entire flow. Now, what are the other most important things that you have to know from a PYQ point of view? The question has come, which is the common passage for food and air? Your answer will be what? Pharynx. Okay, your pharynx is going to be a common passage for food and air. Okay, so this is a PYQ question. I mean, potential MCQ question. So, the yellow color underlines are very important for you. Now, next question. Which is the cartilaginous structure? Which is the structure that is completely made up of cartilage? Your answer should be larynx. Larynx is a cartilaginous box which help as a sound box for you. Okay. How many cartilages are they made up of, students? Please let me know in the chat box. How many cartilages are they made up of? Let me also check if you all are marking your attendance as well. Okay, come on, quickly answer that question as well. Okay. Yes. Students, many of you haven't marked your attendance. Come on. Mark your attendance quickly. Okay. So, larynx is a cartilaginous box. Excellent. It is made up of 9 cartilage. Okay. 3 paired, 3 unpaired. In that, one of them is epiglottis, which we are going to. Glottis, epiglottis is there that you have to remember. Okay. Now, students, we call this larynx as the sound box. Now, I have the sound because of my larynx. Okay. That is a complete cartilaginous structure. Now, during swallowing, now when I am trying to swallow something, now look here, this bottle is a complete open bottle, right? This is an open bottle. Now, on another example, let's take this bottle. Okay, this is a perfume bottle. Okay, this is the perfume bottle. Now, let's say I am trying to take in something. Okay, I am trying to take in something. This is the cap that will protect it. This is the cap that will protect it. Same way, whenever I am trying to take in food or water, there is a covering on the glottis which is called as epiglottis, which is a flap-like structure, which is again an MCQ question. Okay. So look here, students. During swallowing, glottis can be covered by a thin elastic cartilaginous flap called as epiglottis. To prevent the entry of food into the larynx. Clear to all of you? That is sometimes when we talk, we choke. That means we start coughing. Why? Because the food has entered into the windpipe. Okay. That kind of mixing shouldn't happen. So what is happening? There is a flap-like structure. Now, Trachea is a straight tube extending up to the mid-thoracic cavity. Now the question comes, where exactly does the trachea split into or trachea divide into? That is the question. So the question answer is here, students. Potential MCQ again, that is, it divides in the fifth thoracic vertebrae, and what is that called? Carina. Okay, that region is called carina. Okay, now don't write carina and come. So on the fifth thoracic vertebrae, these people are going to divide into primary or left and right and left bronchi. Okay, now the bronchi are going to go for a repeated division to form secondary and tertiary bronchi. Again, they are going to divide to form what, students? Bronchioles. Okay, initial bronchioles are there, terminal bronchioles are there. We also say about respiratory bronchioles. Clear to all of you? Excellent, excellent, excellent. Yes. Now, one more important MCQ question. What is it? One more important MCQ question. Trachea and up to your initial bronchioles are covered by incomplete cartilaginous ring. So the question comes from here, students. See, the trachea, primary, secondary, tertiary and initial bronchioles are supported by incomplete cartilaginous ring. So why is the ring incomplete? Because the trachea shouldn't collapse. Because imagine something is entering and exiting, right? There are chances that where we need some compression and some relaxation. Now, if I put a proper ring around it, it is going to definitely break. So, I have to avoid that. So, what I am going to do? I am going to give an incomplete cartilaginous ring. Now, one more MCQ question that I can ask you is, the ring is incomplete in the dorsal or in the ventral side? All of you answer me. All of you answer me. Is it dorsal or ventral side? Faster, faster, faster. Yes, prevents collapsing. Excellent. Excellent. Up to initial bronchioles. Excellent. Okay. Trachea up to initial bronchioles, you will have incomplete cartilaginous ring. Now the question goes like this. Incomplete cartilaginous ring. Which side is it incomplete? Is it on the dorsal side or on the ventral side? Excellent question. Dorsal is the answer. Okay. Dorsal is the answer. So always remember, students, on the dorsal side, the ring is incomplete, and this is the kind of questions that they can ask you for NEET 2025, because they know everyone will study about incomplete cartilaginous ring. Students don't study dorsal and ventral. Okay. Yes. Now, each terminal bronchioles give rise to a number of very thin irregular walled, okay, irregular walled and vascularized bag-like structures called alveoli. So, alveoli is a sac-like structure, okay, sac-like structure where the actual exchange of gases is happening. Clear to all of you? Moving on to the next one. We have two lungs which are covered by double layered pleural membrane, okay. There is a proper pleural membrane. If you look at the diagram here, you will notice that if I take a section of the lungs, the first layer is going to be the parietal pleura, second layer is going to be visceral pleura. Whenever the word visceral comes, please understand it is something to do with internal. Okay, because we study visceral muscles, okay, visceral organs, visceral systems. So that means what? Something to do with the inner organ. So visceral is inside, pleural is outside. So lungs has two covering, parietal and visceral. Now parietal and visceral, students, between that what is there? Pleural fluid is there. What will pleural fluid do? It is going to act as a shock absorber. It is going to make sure to reduce the friction. Clear? Yes. Exactly. Very good. Very good. Okay. So, reduces friction on the lung surface. Outer pleural membrane. Question comes from here also. MCQ. Okay. MCQ comes. Outer pleural membrane is in close contact with dash. Okay. Is in close contact with what? The thoracic cavity. Outer will be, so just imagine, this portion is going to be in contact with the ribs. It is going to be in contact with the thoracic cavity. But the visceral one is going to be in contact with the lungs. So the question comes like that also. Clear to all of you? Yes? Yes, 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 yes. Can't see thumbs up. Okay. So the question goes like this. Inner pleural membrane is in contact with the dash surface. If they are mentioned in NCERT, then there is a potential chance that this comes as a question. Okay, I just hope all these questions which I am telling, please come, so my Vedantu NEET English students can score well. Okay, the part starting with the external nostrils, now comes the other portion. Two zones are there, okay, conducting zone and respiratory zone. Conducting zone means what? Now, up before we go into the flight, okay, entering into the flight, the entire airport way, there is an entire system going around, right, where we get the ticket I'm telling, boarding pass, where we get the boarding pass, checking of the luggage, the weight of the luggage will be checked, all that procedures happen. Actual flight is when you enter into the flight, okay, whichever Indigo, Air India, whichever you're entering into. So same way here also, conducting zone is the one who's going to do the moisturizing, filtration, humidification, and actual exchange of gases is going to happen in the respiratory zone. Again, a MCQ question, okay. So the question goes like this, students: external nostrils up to dash, okay, is the conducting part. So you will say external nostrils up to the terminal bronchioles. Do not include respiratory bronchioles. Respiratory bronchioles will fall into the part of respiration, okay, respiratory or exchange part. Clear? So up to the terminal bronchioles is going to be conducting part. Alveoli to their ducts and the respiratory, alveoli to their ducts and alveoli will be a part of the respiratory or the exchange part. Now the question again comes, what exactly happens in the conducting zone? Exactly what happens in the conducting zone, students? See, foreign particles will be removed, humidification will be done, temperature will be set, okay, conditioning will be done. Conditioning will be done. Okay, clear to all of you? Yes, thank you so much. Okay, very good, very good, very good. Now comes the next question for all of you. What is the thoracic cavity? What is the thoracic cavity? The lungs are situated in the thoracic chamber. So students, remember thoracic cavity like a four-walled system. Okay, four-walled system. That means, let's take, okay, my nose breathing, my respiration is not working properly. So this is a closed system, okay. So ventral, dorsal. So front side of the remote is ventral, back side is dorsal, side you have lateral, down you have one thing, okay. Now thoracic chamber is a tightly packed chamber which is going to help us in increasing the volume so that we can change the pressure, we can regulate the pressure, because if we do not regulate the pressure, then there is very less chances for the air to enter inside, okay. Clear to all of you? So now tell me, what is on the ventral side? On my ventral side, I have the sternum, okay, sternum. Now what is on my dorsal side? Dorsal side, I have my entire vertebral column or your thoracic vertebrae, okay. What do I have on my lateral side? I have my ribs. Touch and see. Can you feel? I have my ribs, okay. Yes or no? Yes or no? Clear? We have the ribs, okay. Lower portion, what do we have? We have the diaphragm. So this kind of makes the four box things, right? The entire airtight chamber is there. So look here, students. The thoracic chamber, dorsally you have the vertebral column, ventrally you have the sternum, laterally you have the ribs, and you have lower side a dome shaped diaphragm. I have only underlined the things that are important to you, okay, important to you. We will come to the lobes, okay, we will come to the lobes, don't worry, I have not skipped anything, okay. I have not skipped anything. I have also added my notes as well as NCERT for all of you, okay. Yes. So look here, students, we start from this portion, okay, we start from this portion. So very simple, what did we have? External nostrils were there, two openings were there, the air was taken inside. Now once the air is taken inside, it is going to come to the nasal cavity, to the internal nostrils and all of that. We understood, no? Nasal passage, nasal chamber, larynx, pharynx, all of that is happening. Now look here, the main three things that are done by your conducting zone is going to be, the hair is going to do the filtration of the air, conditioning and humidification. That means to regulate the temperature, to remove the foreign particle and also to humidify, because the air outside is going to be very dry air. So once that enters into our body, we are going to have some dust allergy or foreign particle has entered. No? So what we will do, we will add some humidify it. Humidify means what? A little like moisturizing. Okay, have you put moisturizer? Okay. Yes. Okay. Internal nostrils and air, students. Again here pharynx have three parts. Okay. This is there in most of your modules. So pharynx have three parts: nasopharynx, oropharynx and laryngopharynx. This is a common passage. So this is where your MCQ is going to come from. Okay, this is where your MCQ is going to come. Nasopharynx, that is air, oropharynx, air plus food, laryngopharynx, which is again near to your larynx. Now students, larynx, a 9 cartilaginous ring structure, okay, cartilaginous box, which is also called your voice box, where you have 3 paired and it is made up of 3 unpaired structures, okay. From larynx, trachea, primary, secondary, tertiary, initial bronchioles and alveoli. Any doubt in this? Any doubt in this? Yes or no? No, right? So look here, students, here you understand glottis is there, which is again a part of the larynx. It is coming under one of the cartilage. So on the glottis, there is a flap-like structure called epiglottis to prevent the food from entering. Okay. And if you see, there is incomplete cartilaginous ring circled completely. And one more question that can come is, where is the trachea dividing? It is dividing in the 5th thoracic vertebrae, that is also called as carina. Okay, that is also called as carina. Okay, yes, clear to all of you? Everything is done from here. Okay, this is again flowchart I have put for all of you, so you can see the diagrams clearly. See, this is the carina. That is the portion where the branching is going to happen into the right and left. Yes, lobes. Now your lungs have lobes. Okay, how did this lobe formation happen? Very simple, students. It's like an equatorial plane, no? On your earth, how your equator is running, dividing into longitudinal, maybe not. Okay, Arctic zone, Antarctic zone, something like that we had studied, no, all that. Okay, yeah, all the geography teachers, please leave the class. Okay, so on the earth, how the equator is there, like that we have fissures. Okay, like that we have fissures, students. So if you look at this lungs, no, if you look at the right and the left lungs, you will notice that the fissures are completely different, or one fissure is more, one fissure is less. So if you look at the right lung, students, you will notice that there are two fissures. One is the oblique fissure, which is common for both of them. Right also has, left also has oblique fissure. Correct? Yes. Now, horizontal fissure, students, horizontal fissure is only present in the right lungs. Horizontal fissure is only present in the right lungs. That is dividing the right lungs into superior, middle and inferior lobe. Okay? Superior, middle and inferior lobe. Clear to all of you? Yes or no? Clear to all of you? So, superior lobe, middle lobe and inferior lobe. Superior lobe, middle lobe and inferior lobe. Now students, on this side you only have oblique fissure dividing the lungs into superior and inferior. Superior and inferior. Clear to all of you? Yes or no? So left lungs, 2 lobes, right lungs, 3 lobes. Okay? 3 and 2. 3 and 2. Okay. Clear? Now, coming to the major portion of this class, that is, how does exactly exchange of gases happen? How does exactly exchange of gases happen? Okay, starting from the first. So far, any doubts, students? So far, any doubts? Please tell me. Please tell me any doubts so far. Yes. Come on, come on, come on, come on. Quickly, quickly. Ma'am, tracheal yes, yes, it is actually C-shaped hyaline cartilage. Okay, if you want to write it somewhere, please write and keep. Okay, if you feel that, ma'am, it might come as a question. That is, your trachea, okay, has C-shaped, okay, incomplete C-shaped hyaline cartilage. Okay, hyaline cartilage. Okay, PYQ question. So yes. Okay. Yes. Thank you, Lokesh. Yes. Moving forward, students. Clear so far? Can we go to the actual portion from where very tricky questions, or like sometimes very easy questions come? Okay. This is the 4 steps. Okay. 5 steps that are happening. Just think of it very simple, students. Breathing or pulmonary ventilation, by which what is going to happen? Atmospheric air is going to be drawn inside. Okay. Atmospheric air is going to enter inside, and the carbon dioxide is going to go out. So one is entering, one is exiting. Okay. Now all this you have studied in your smaller classes. O2 is entering inside, carbon dioxide is going out, is the first step. Second step, diffusion of gases across the alveolar membrane. That means we know that alveoli is there. Now we have to make sure that this gas which we have taken has to go in and has to come out. So here one diffusion is happening. Yes, ma'am, diffusion is happening. Transport of gases by blood. Now alveoli gave all the blood the oxygen that it required. Now it is important for the blood to transport it. Otherwise, just keeping it, it will not happen. Okay. Diffusion of oxygen and carbon dioxide between the blood and tissue. So now the blood is bringing, who is the person who is waiting for this? The person who is waiting for oxygen and is crying for oxygen is your tissues. So this fellow will give oxygen. Obviously something has to be given in return. So he will give carbon dioxide. Clear to all of you? Now last step, what happens, students? Cellular respiration. Inside the tissues, they are going to be very happy. They are going to use all the oxygen and perform all the functions for us. Okay? Perform all the functions for us. Clear to all of you? So these are the 5 most important steps. What is first? You are trying to take the air which is outside to inside your body, and whatever is inside, you are trying to give it outside. That first step is done. Clear to all of you? Pulmonary ventilation. Second step is what? You are trying to give whatever you took inside from the atmosphere, you're trying to give it to your alveoli, because your lungs is supposed to do the respiration, right? So you're giving it to the alveoli. So since you're giving oxygen, alveoli is wanting to take the carbon dioxide from there. Now, whatever you have given to the alveoli, alveoli is given to the blood, telling that I cannot go and transport it, so you do the job for me. So that transport is going to happen. Now, who is the one who is hungry for this oxygen? Your tissues. So we are going to give that oxygen to the tissues, telling that you now start doing your function, and ultimately they are going to do cellular respiration. Clear to all of you? Clear to all of you? Yes or no? So these are the 5 most important steps. Now you start with inspiration. That is, how do I take? First step. How do I do pulmonary ventilation? How do I take the air which is outside inside to my body? Now, this diagram has everything that is required. Only thing that we are going to focus on is the difference between the pressure and the volume. Okay. So, students, according to Boyle's law, we have to know that pressure is inversely proportional to volume. That means generally, if volume increases, pressure will automatically decrease. So, now my concern is I want to do inspiration. What I want to do? I don't want to inspire you people. I want to do inspiration. That means I want to do inhalation. Okay, I want to inhale. I want to inhale. Now, if I want to inhale, what is the first thing that I have to know? That anything, if I want to take it inside my body or do any kind of exchange, I should know that the exchange of gases is going to happen from a higher region to a lower region. Correct? Now, that means if I have to make a lower region, I should make sure my pressure goes down. If I want to make my pressure to go down, I have to make sure that my volume is high. Yes or no? Do you understand the cycle? Do you understand the cycle? Yes or no? Clear? Okay. Now, look here. If I want the gas to change, I need to come from higher to lower concentration. Then only air is going to enter inside my body. If I want to lower the pressure, okay, if I want to let my air come in, that means my pressure should be less. If I want my pressure to be less, I cannot go and regulate my pressure. What I can do, I can make the volume high, because according to Boyle's law, this proves right. Clear? Now, look here. If I want to increase my volume, there are 2 main people who are going to do that job for me. The 2 main muscles who are going to do that job for me. First one is going to be my diaphragm. My diaphragm is the first muscle who is going to help me to do all of this. My second muscle is going to be external intercostal muscle. Okay, external intercostal muscle. Okay, clear to all of you? This is going to be where? In the ribs. Okay, it is going to be present in the ribs like this. This is my external intercostal muscles. Now, the 2 things who are going to increase my volume is going to be, first, my diaphragm is going to contract. Now, many students have confusion. Ma'am, when diaphragm contracts, how do they flatten? Okay, so to remember that, students, remember, whenever you buy a flat means what? Like a house, okay, inside an apartment, whenever you buy a flat, you sign a contract. Yes or no? You sign a contract for the maintenance, okay, for the parking, blah, 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 okay. So, whenever you buy a flat, you sign a contract. Same way, remember, whenever the diaphragm contracted, it starts to get flattened, because my goal is to increase the volume, not decrease the volume. My diaphragm will flatten. Clear? Clear? Yes or no? I hope all of you are marking your attendance, okay. After all this hard work, the only thing that we ask you people is to mark your attendance, okay. Yes, it is not increasing. Come on, let's make it 600 likes quickly. Okay, it's not such a big thing for my students, I know. Those who haven't liked the video, quickly like the video. See, diaphragm contracted, so it got flattened. How will you remember? Whenever you buy a flat, you sign a contract. Clear? So, diaphragm contracts, flattens. First thing of my volume increase has happened. Now, students, understand, everything is happening inside the thoracic chamber that I told you. Sternum, thoracic vertebrae or vertebral column, ribs and diaphragm. That is the box that we are trying to increase the volume for. Second thing that is going to happen is, whatever diaphragm does, this fellow will copy exactly. External intercostal muscles also will contract. If they contract, what is the consequences, ma'am? The consequences is that the ribs and sternum are going to get raised. So, whenever you are breathing in, can you see the whole thing is coming up? Okay. The whole thing is coming up, right? So, ribs and sternum is going to get raised. Now, you tell me, did the volume increase or not? Yes or no in the chat box? Yes, Christina. Yes, Rihanna. Very good. Yes, come on, tell me. If all this happens, that is external intercostal muscles, diaphragm has contracted, both of them contracted, ribs and sternum got raised, this got flattened, has the volume increased? Yes or no question? Very good. Ma'am, volume increased. Ma'am, volume increased. If volume increased, I had one more task. What is the task? If volume increased, I know the pressure will go down. But why? How much the pressure will go down? Is it like how our BP goes down? Is it like our pressure goes down seeing NEET paper or seeing salary credited in the account? Not like salary getting cut from the account. Okay. So it is not like that kind of a pressure. It has a proper number. Now, when I'm speaking about pressure getting decreased, first of all, you should understand atmospheric pressure is 760 mmHg. 760 mmHg. Now, I want to make sure that this fellow is on the upper hand so that my air can come in. So, I will go by 2. That is, I will decrease my IPP. Now, don't ask me, ma'am, what is IPP? Intrapulmonary pressure. The pressure inside the lungs. Intrapulmonary pressure. Okay, intrapulmonary pressure. So, I am going to decrease it to 758 mmHg. Now, pressure decrease, automatically rush of air is going to happen, oxygen is going to enter into my body. Inspiration is clear to all of you. Inspiration is clear to all of you. This is the only thing that you have to remember in inspiration. No other questions are going to come from inspiration. Okay. Now we go to expiration. I want to remove the carbon dioxide from my body outside. Again, we follow the same simple rule. The rule goes like this. If I want carbon dioxide to go out, that means I have to again move it from a region of higher concentration to a region of lower concentration. But here, what is happening? My higher concentration is expected inside my body and lower concentration is expected inside atmosphere. Now, the problem is that I am no one to change the atmospheric pressure. I am no one to change the atmospheric pressure. Yes or no? So, if I cannot change the atmospheric pressure, what I am going to do? I will change my IPP. Correct. I will change my IPP. So, if I want to change my IPP, that means I need to increase the pressure. I need to increase the pressure. If I need to increase the pressure, again I have to play with the volume. That is, I have to decrease the volume. So, I will tell whose two people's help will I take. I will again take the help of diaphragm, external and internal intercostal muscles. Okay. I am going to take the help of diaphragm and I'll tell diaphragm, you please relax. You please relax. Because automatically when diaphragm relax, since it's a dome shaped structure, it is going to again go back to the dome shape. So, it is going to push the air outside. Correct. Now, if you come here, students, ribs and sternum return to original position. For that, who should help me? External intercostal muscles. External intercostal muscles should relax. Okay, relax. Students, in some PYQ question, I saw internal intercostal muscles. Internal intercostal muscles will contract. Okay. If the question comes, internal intercostal muscles will contract. Okay, clear? Contract. Now, diaphragm relaxed, this fellow also relaxed so that this came back to original position, making sure that the volume decreased. If the volume decreased, automatically can I increase the pressure? Yes, ma'am, you already made us understand. So, I want to increase it by 2. Again, I go 762 mmHg. 762 mmHg. Now, have I done the concentration? Yes, ma'am. Now, automatically carbon dioxide is going to go from a region of higher concentration to a region of lower concentration. Clear to all of you? Yes or no? Yes? Clear? Done and are done? Okay. So, this is what is going to happen. Automatically pressure increase. Now, students, during forced expiration, okay, during forced expiration, you can go up to increasing by 4, that is 764 mmHg. 764 mmHg. Got it? Yes? Okay. Okay, clear? Done, done. Pakka? Okay, now let's go to the most important question from where lot of questions come, students, your volumes and capacities. Students, volumes and capacities, you have to write and learn. There is no other option to it. Okay, so far good. Can I get a lot of thumbs up in the chat box if so far everything is good? So far everything is good. Can I get thumbs up in the chat box? Fast. Done. Okay. Okay. Yes, today midnight it should be released, your admit card, or by tomorrow morning max to max, because May 1st is what they told. Okay, May 1st is what they told. Mostly today midnight they will release it. Okay. Yes, come on. Come on, come on, come on, come on, come on. Okay, let's do volumes and capacity. Students, only 4 volumes you have to study, which is not a difficult task. Okay, it's not a difficult task. You people study all your best friends Instagram ID, which is more complicated than encyclopedia names. Okay, so look here, starting with tidal volume. Tidal volume is the amount of air that I'm normally taking in and normally giving out. So normal expiration and inspiration. Now your tidal volume is happening, right? Normally you're breathing in, going out, breathing in, going out. You're not going to take a stress about it. So normal tidal volume is going to be how much? 500 ml. Okay, 500 ml. That is normal inspiration, expiration, normal inspiration, expiration. But suddenly you see the NEET paper. What will happen? You do that is forceful inspiration. How much air that you can forcefully take it in. That goes to your inspiratory reserve volume, which is the highest, student, 2500 to 3000 ml. 2500 to 3000 ml. Clear to all of you? Yes? Okay, clear? Okay, clear, done. So 200 to 3000, 2500 to 3000 ml is your inspiratory reserve volume. Again, this is forced. Okay, you're forcefully taking it in. Now, after forcefully taking it in, if you hold like that, then forcefully you have to go up. Okay, you have to leave it also. So that will become your expiratory reserve volume. Okay, expiratory reserve volume, there is a forced expiration. How much maximum air that can go out? You have done forced expiration, which is around 1100 to 1200. No. 1000 to, even this value is correct, 1000 to 1100. Okay, 1000 to 1100 ml. Correct? Yes or no? Yes or no? Done. Okay. Now, after that, students, how many ever times you expire, how many ever times you try to do this air out, there is one amount of volume that will not come out of your lungs. This is to make sure, since your alveoli is a sac-like structure, there are chances that alveoli will collapse. To avoid collapsing of alveoli, we have a amount of air that is going to remain as such, which is called the residual volume. Residue means what? Residue means leftover. No, residual volume, which is around 1200 ml. Lot of times this question keeps coming. Clear to all of you? Yes or no? So this is your volumes. With the volumes, you have to calculate the capacity. Capacity means what? Addition of 1 or 2 volumes together or 3 volumes together makes your capacities. Now, let's start with the first capacity, which is called as inspiratory capacity. Okay, inspiratory capacity, students. Remember, for expiratory and inspiratory capacity, you have to take the tidal volume. Okay, so inspiratory capacity means what? I am trying to see how much I can breathe in during normal and forced. If I want normal, I'll take tidal volume. If I want forced, I'll take inspiratory reserve volume. Correct or not? So my IC, okay, inspiratory formula only they will ask you. Okay, you can do the maths basically. Inspiratory capacity is going to be IRV plus TV. IRV plus TV. Correct? Yes, ma'am. IRV plus TV. I agree to that. Now, how much air can I give out? How much air can I expire out? Normal, forced. What will be that? Expiratory capacity. That is going to be ERV plus TV. Okay, ERV plus TV. Correct? ERV plus TV. Done. Okay. Now, functional residual capacity. What do you think functional residual capacity, students? Again, after expiration, how much air I have, how much I have expired out. So, I am going to take what? I am going to take my ERV. Okay, ERV plus RV. Correct? It's very simple. If you try to understand the concept, no, you will automatically understand, okay, how do I do the calculation? Okay, so is this clear to all of you? Functional residual capacity, excellent. ERV plus RV. Okay, ERV plus RV. Now, vital capacity. Vital capacity is how much I can normally expire, forcefully expire, but it does not include, okay, it does not include what is remaining in your lungs. Okay, that means we are going to remove the RV, everything else we are going to include in vital capacity. So vital capacity will be what, students? It is going to be TV plus IRV plus ERV. Okay, they will ask you to do the calculation of this. You can easily do the calculation. Total lung capacity means what? You calculate everything. TV plus IRV plus ERV plus RV. Okay, don't, please make sure in MCQ question they will give you like this: TV plus ERV plus IRV minus RV. Don't do that. Don't do this. If there is an option which does not have this, does not have RV, you select that option. Don't go for this minus option, because when you do maths, right, this value is going to be different, right? When you do totaling minus, it might be different. So that is why do not go for this, go for this. Clear? Clear to all of you? Yes? Done? So don't make such mistake. They will try to trick you. Don't fall for that trick. Done, done, done. Okay, very good. So that is your volumes and capacities. Okay. Now students, we are going to understand how does the exchange of gases exactly happen. Okay. Now before that, you should know what is a partial pressure. Partial pressure is actually the pressure exerted by an individual gas in a mixture of gas. Okay, now you people are, this many of you are there. There is one student who is continuously asking some question. What is that? What is this? What is this? That fellow is putting pressure on me. So that means the pressure exerted by an individual gas in a mixture of gas. Clear? That is the partial pressure. So from now on, you are always going to see this symbol, right? PO2. That means partial pressure of oxygen, partial pressure of carbon dioxide. So don't get confused. What is the meaning of it? Partial pressure is different, pressure is different. Okay. Now let's try to understand this. So in this box, you have to know by heart. You should know it by heart. You don't have to by heart it if you already have learned it 2-3 times with me itself. Okay. With me itself. Now look here. PO2 and PCO2. That means partial pressure of oxygen, partial pressure of carbon dioxide. Atmospheric air. Okay, in atmosphere, oxygen should be more, otherwise it will not come inside. Okay, so 159 is out. So remember what I told you, higher concentration to lower concentration, higher concentration to lower concentration. Okay, so let's only think of this one. Let's not think about carbon dioxide now. Now I want to take in oxygen. That means I want to do inhalation. Okay, I want to do inhalation. If I want to do inhalation, what will I do? From a higher concentration, it's going to come to a lower concentration. Where is the lower concentration? My second step is diffusion in the alveoli. So second step, alveoli will have 104. Alveoli will have 104. Clear? Now, 104 to where it is going, students? It is going into your deoxygenated blood. Why is it going to your deoxygenated blood? So I can make it oxygenated. I can make it oxygenated. So please understand, here diffusion is happening, not exact exchange is not happening. Okay, diffusion is happening. That means what? Let's say I'm continuously giving you some, think of something. Okay, something I'm giving you. It's diffusing. I'm not giving you as quantities. Let's say I'm not giving you, I'm pouring you curry. Okay, I'm not giving in ml. I'm just pouring the curry. I'm just pouring the curry. Curry is increasing. Okay, but let's say I'm giving you one chocolate piece. I know the exact number, 1, 2, 3. Here addition is not happening, but continuous diffusion is happening. Clear to all of you? So here what is going to happen? Okay. Okay, here what is going to happen? Deoxygenated and oxygenated blood. So students, from 40, here you are trying to make it oxygenated. So continuously oxygen is adding. That is from 104 to up to 95. Continuous diffusion is happening. So do not calculate 104 plus 40 should not be 95. Okay, it is not proper addition, it is diffusion. Okay, now from 95 it has to go to 40. That is your tissues, it has to go, because only oxygenated blood we can give. No, we can't give deoxygenated blood. Clear to all of you? So that is how your oxygen is going to enter inside. Now let's try to remove carbon dioxide outside. You start from the tissues. Tissues have all used up the oxygen and they have converted it. So 45 to 40, we are giving it to the oxygenated blood. Now the oxygenated blood is automatically going to become deoxygenated. Why? Carbon dioxide amount is more in the blood. Okay. So from 40, it is going to go to 45. That is how, again, here also diffusion is happening. Okay. Here the carbon dioxide is going to increase. Okay, here the oxygen is going to increase. Now from 45, students, it is going to the alveoli, which is at 40, 40 to atmosphere at 0.3. Like this, you can give out the carbon dioxide. Simple dimple. Okay, simple dimple, humble. Okay, now the same thing here also. Same thing here also. Look here, the alveolar air is there, 104 and 40 is there. From there, you can see how the gaseous exchange is happening. It is coming all the way through the pulmonary vein, going into the heart, and it is going into the tissues. Okay, going into the tissues. Now from here you see, from the tissues it is taking systemic veins, again taking it to the heart, from the heart to the lungs, lungs is removing it out. That's all. That's all. You just have to know this number. Okay, and one more PYQ question that can come is, which among this have the same numbers? Your answer should be tissues and, okay, tissue and tissue and deoxygenated blood. Okay, tissue and deoxygenated one will have the same value. What is the same value, students? 40 and 45. Okay, 40 and 45. Correct, no? Look here. Deoxygenated blood 40-45, tissue 40-45. Correct? So this question will come. Okay, this question will come. Okay, now this is done, this is done. Okay, students, again, very important MCQ question. How does the exchange happen? What is the respiratory membranes? Okay, what is the respiratory membranes? Look here, there are three major layers. Okay, first one is a thin squamous epithelium of the alveoli, then you have endothelium, and then you have basement membrane. So all of you look here, first is going to be squamous epithelium. The question has come in structural organization also. Where is squamous epithelium present, or which is the epithelium that lines the blood capillaries and the air sacs? Your question is epithelium. Answer is epithelium. So, squamous epithelium, then you have endothelium of the capillaries. Between this, there is a non-cellular layer which is called the basement membrane. And students, one most important factor or characteristics of the respiratory membrane, it is that the thickness should be less than 1 millimeter. Okay, MCQ question. Less than 1 millimeter. Clear to all of you? Yes or no? Okay, perfect, perfect, perfect. Okay, so endothelium, epithelium, endothelium and the basement membrane. Now talking about the transport of gases. Okay, transport of gases, students, oxygen, basically if you calculate 100%, 3% will be dissolved in plasma, rest 97% will go with hemoglobin. What will it form? All of you tell me in the chat box. What will it form? Like hemoglobin plus oxygen. What is the combination called as? Yes, faster, faster, faster. Faster. Yes, less than 1 mm. Excellent. Epithelium. Squamous epithelium of the alveoli, middle one non-cellular layer which is the basement membrane, and third you have the endothelium. Okay, epithelium, basement membrane, endothelium. Do not forget this. Now students, what did I tell you? Oxygen, students, 97% will go off with whom? Will go with hemoglobin. That will form oxyhemoglobin. Excellent. Now, many students have confusion in this. Please do understand that whenever oxygen transport, when we talk about, we should also talk about carbon dioxide and carbon monoxide. Why? Because oxygen to carbon dioxide, students, understand these people have 20 to 25 times more affinity. Who? Carbon dioxide has more affinity with hemoglobin. Okay, and when you compare it with carbon monoxide, they have 200 to 250 times affinity. 200 to 250 times affinity. Who? Carbon monoxide have with hemoglobin. Now the question goes, when when hemoglobin combines with carbon monoxide, what does it form? What does it form? Please remember, it is carboxy. It is forming carboxy hemoglobin, not carbaminohemoglobin. When it combines with carbon dioxide, it will form carbaminohemoglobin. Carbaminohemoglobin. Done, done, done. Now, when you talk about carbon dioxide, the transport is a little more complex. Only 20 to 25% is going to go with hemoglobin, 7% is going as dissolved plasma, and the rest 70% is going as bicarbonate. Okay, 70% will go as bicarbonate. Clear to all of you? Now you have to study how is the bicarbonate formation. That is a very, very important question for all of you. Okay, very, very important question for all of you. Okay, very good. Now, students, this is also very important MCQ question. Every 100 ml of oxygenated blood can deliver around 5 ml of oxygen to tissues under normal condition. When we are running, when we are doing marathon, we can go up to 15 ml. But normally, after the atrial and venous blood calculation, 19.4 minus 14.4, you are going to get 5 ml of oxygen will be given to the tissues. Clear? Yes? Okay. Now, here comes the oxygen dissociation curve. Very, very, very important. I am going to tell a very, very silly story here to make you understand this. But please understand, the story is just to make you understand. It does not have anything to do with my knowledge, educational content or anything like that. Okay? Clear to all of you? Yes. Now look here, students. What I am trying to say is, whenever the oxygen, okay, hemoglobin and oxygen is going to have an affinity, notice that it's very simple. When you are starting to eat biryani, okay, when you are at 0, okay, you are full hungry, you are at 0 food, biryani is going to taste better. You are going to have more affinity, liking towards the biryani. But let's say when you reach by the 50%, you have eaten half of the biryani, your stomach also is half full. Will you still have that much liking to biryani? No, you would be almost heading to saturation. Same thing happens here also. When we talk about oxygen dissociation curve, students, understand that once the oxygen, okay, what is this dissociation meaning? Remember, alveoli, okay, in the alveoli, okay, and in the blood, In the alveoli, students, we are trying to make oxygen get combined here. So here association is going to happen. That means we are trying to give this oxygen to the blood and telling that blood, you please make HbO2. Okay, here association is happening. We are trying to associate them. But the same thing when the blood is coming to the tissues. After this association, here he has to give the oxygen. So here dissociation will happen. Here dissociation will happen. So oxygen dissociation curve means what? When we are going to give this oxygen to the tissues. When we are going to give this oxygen to the tissues. Now look here, students. Two graphs are there. Right shift and left shift is there. So right shift what is happening, left shift what is happening, you have to know. Look here, first of all. When three people are there, oxygen's life is very miserable. When three people are there, oxygen's life is very miserable. Why do I say that? I'll tell you why. See, whenever the partial pressure of carbon dioxide is high, automatically there is two more chamchas we tell, no, or like nanbans, okay, H plus and temperature. These people are also high. Whenever partial pressure of carbon dioxide is high, these two people also are high. Now this will lead oxygen to actually break up with hemoglobin and do the right choice. So think like this. Think like this. There are three toxic friends for hemoglobin. Okay. Now listen to the story carefully. Listen to the story carefully. No one is going to explain you this way. Let's say there are three toxic friends for hemoglobin. And he is very, very close to this one fellow. That is your carbon dioxide. Now oxygen is this fellow's, who? Hemoglobin's wife. Okay. Wife. She's very frustrated. When these three friends are extremely at a higher pitch, they're coming to home every time. That means oxygen is very, very, feeling very bad. So oxygen will do the job. That is, oxygen will take a right decision of going to her home. So right shift happens when carbon dioxide is high, H plus is high, temperature is high. Understood? The graph will shift to the right when oxygen and hemoglobin has reduced affinity. Why do oxygen and hemoglobin have reduced affinity? Why do they not have love? Because these three friends are coming and sitting in their house. Understood? Clear to all of you? I told you I'm going to tell you a very simple example, like very lame example. Okay. So students, right shift is going to happen when they have reduced affinity, increased temperature, increased H plus concentration. That means oxygen has taken the right decision to go to her home. Okay, and take some break. Understood? Understood? Yes. Vedant not in NEET channel. Okay, here no spam. Here very serious students are there. We will not let you do any kind of spam here. Okay, no other conversations. Clear? Now, on the other side, will you forget this? Right shift is going to happen. When are you going to take the right decision? When your husband is always with his friends, or when your wife is always with his friends. Who are the three friends? Carbon dioxide, H plus and temperature. Clear? When will you take the left shift? When these people are in, that means hemoglobin and oxygen is happy. Why? Because the three friends have gone to Thailand. Okay, three friends have gone to Thailand. So obviously hemoglobin and oxygen is going to have association. That means they are going to be happy. They are going to have high affinity towards each other. Correct? So oxygen, the graph is going to shift to the left when these people are low and oxygen is high. Okay, that means hemoglobin has not reached the saturation. Hemoglobin can take more oxygen and make the thing. Okay, clear? Clear, all of you? Yes, exactly. So no breakup will happen in the left shift. Breakup will happen in the right shift. That means oxygen takes the right decision to leave the house to break up with hemoglobin because carbon dioxide, H plus and temperature is partying in the home. Okay, when these three people are going for trip, when carbon dioxide goes, these two nonbans also will go. Temperature and H plus will go. If they are going, automatically hemoglobin's affinity towards oxygen is actually more because he doesn't have anyone else. He'll be like, okay, I'm lonely. So he'll look at his wife and he'll be like, okay, I have her. I'll talk to her. So affinity is more. That means this is where association is starting to happen. Okay, this is where your dissociation is starting to happen. That means hemoglobin is giving the oxygen to the tissues. Clear? Yes. Okay, done. Okay, this is the example. So don't forget this, students. Please remember what is exactly happening. Okay, last topic. Last topic of this chapter, most important topic of this chapter as well. Done? Pakka? No one will forget. No, when does the breakup happen? All of you once again revise with me. When does the breakup happen? When does the right shift happens? All of you tell me. Right shift happens in 4 conditions. When partial pressure of carbon dioxide is extremely low, partial pressure of oxygen is extremely low, carbon dioxide is high, H plus is high, temperature is high. Okay? Yes. Which background? Some normal background, I don't know. Yes, tell me students, quickly. Okay, when does right shift happen? I want to see answers. Dissociation, extremely good. High temperature, carbon dioxide is high, H plus is high, very good. When does left shift happen? Okay. Students, you are in oxygen side. So you're going to think like this. Okay, oxygen took the right decision when these 4 people were partying. So think like that. Okay, don't be on the husband side, don't be on the wife's side. Okay, you can change your genders according to your genders. Okay, I mean, you can change, yeah. Understood, right? Okay. Now, going forward, students, I told you that carbon dioxide transport happens. 7% is dissolved in the plasma because obviously carbon dioxide can dissolve in water more than oxygen. Okay. And 20 to 25% will go with hemoglobin. Okay, we'll go with hemoglobin, that is carbaminohemoglobin. Don't write carboxyhemoglobin. Okay. And 70% will be stored as bicarbonate. Done. Now let's see how the bicarbonate formation happens with this help. Okay, with this help. Yes, I have rhinovirus. Okay. Now look here, students. Carbon dioxide was in the plasma. So this is the plasma. This white color thing is the plasma. This is your RBC. I hope this much you're understanding. Okay. Now carbon dioxide in the plasma, students, will start entering into the RBC. Why? Because hemoglobin is there. Okay. Now that carbon dioxide plus H2O will form H2CO3. It will form carbonic acid. Okay. Now please do understand, one most potential question that can come from is, what is this carbonic anhydrase? Where is it present at maximum? Please remember, CA, chartered accountant, is more there in RCB office. Okay, like your cricket office, not like that. It is more in RBC office. Okay, and chartered accountant is less in plasma. Very traces of amount is there in the plasma. So this is a potential MCQ question. So here, students, carbon dioxide plus water will form H2CO3, that is carbonic acid, with the help of carbonic anhydrase. Why? Otherwise the process will go very slow. Okay. Now this H2CO3 will break down. That is, they are going to dissociate to form what? H plus and HCO3. They are going to form what, students? H plus and HCO3. This H plus will not chumma sit. It will go and combine with hemoglobin to form HHB. The function of HHB, they will ask you, it acts as a buffer. Buffer means what? Someone that can maintain the pH. Clear? Yes or no? Done. Very good. Now, this HCO3, what he will do, student? This HCO3 will go out of the plasma. Sorry, out of the RBC. He'll be like, no, I want to go outside. I'm so bored inside. So, he'll just go outside. Now, the problem here is, since this fellow is a negatively charged ion, there is an ionization difference between these two. Okay, there is an ionization difference between these two. So, this negatively charged ion went outside. Now, who is complaining, you tell? RBC is complaining to plasma, telling that one negative ion went outside. I am going to have ionization problem. So, do something. So, immediately plasma will tell, okay, don't worry. I have salts in me, right? NaCl. I will break down that to Na plus Cl minus, and I will give you the Cl, which is called as what? Hamburger shift or your chlorine shift. Okay, chloride shift. So, this chloride shift will just be done so that the ionization can be balanced. Okay, one negative came out, one negative we gave inside. So, RBC is like, okay, fine, you gave me chlorine, it is fine. Chloride shift is helping me to balance the ionization. Clear to all of you? This is how, you know, exactly the bicarbonate formation happens, students. So, with this Na, who is going to combine? Your HCO3 can combine to form NaHCO3, which is a bicarbonate, and they can be stored here in the body. Now, when the moment, okay, our respiratory rhythm centers, pneumotactic centers, chemosynthetic centers get to know that the carbon dioxide is very high, the entire process will go in a reverse pattern. Entire process will go in a reverse pattern. Clear to all of you? Yes or no? Okay, Likita is telling 800 likes. Come on, let's make it. Okay, so this HCO3 will break down. HCO3 will enter inside. Chlorine will come outside. HCO3 will combine with H plus after breaking this HHB. This two will combine to form this acid. This will again break down to H2 and CO2, and this CO2 will come outside. Clear to all of you? Yes? Done? Pakka? This is your entire thing. Okay, so I've just given a more clarity picture that to understand, make you understand that from plasma how it is entering inside, what is the formation and how it is going outside. Okay, yes, all of you understood? Yes. Now the last topic of this chapter, students, look here. A specialized center is present in the medulla. So questions are going to come not from what is the specialized center, question is going to come about the location. Okay, because they know in the exam you are going to forget the location in the tension. Okay, so location they will ask you. Location they will ask you. Okay, so a specialized center called medulla region is present in the brain, called as the respiratory rhythm center. It's primarily responsible for the regulation of what? Your inspiration and expiration. Okay, another center which is present in the pons. Many students get confused. Okay, they think that the next center also is in the medulla. No, one center is in the medulla, that is RH, respiratory rhythm center, RRC. Okay, is in the medulla. Your next center is in the pons. Okay, next center is in the pons. Okay, revision class, no detailed class. Yes. So next one is in the pons, students, and this is the pneumotactic center that can moderate the functions of the respiratory rhythm center. Understand? This pneumotactic center is acting as a moderator. All the words I have highlighted, you cannot skip it. Okay, you cannot skip it. It is acting as a moderator to function of like control the respiratory rhythm center. Now, students, other than that, there is again centers called as the chemosensitive areas, which are present where is situated adjacent to the rhythm center. That means location is where? Location is medulla. Location is medulla. Okay, so now what and all did you study? Medulla. First you studied respiratory rhythm center. Now you are studying chemosensitive areas. Okay, pons mein what is there? Pneumotactic center. P for pons, P for pneumotactic center. If you don't know spelling, then I cannot help it. Okay, so yes, which is highly sensitive to carbon dioxide and hydrogen ion. So automatically the body will get to know that okay, H plus and carbon dioxide is high. That means it is time to break down the bicarbonates and send all the carbon dioxide outside. Okay, now increase in this substance can activate the centers, which in turn can signal the rhythm center to make the necessary adjustments, okay, in the respiratory process. Now students, look here, again very important, receptors associated with aortic arch and carotid artery, which is in your heart, also recognizes carbon dioxide and H plus concentration. They also will signal the rhythm center, telling that you know what, there is a problem, we have less oxygen, we have more carbon dioxide, so please start inspiration. Okay, please start inspiration. Okay, thank you. Yes, clear to all of you? RRC. Okay, done. So the role of oxygen in regulation of respiratory rhythm center is quite insignificant. Why, students? Because here try to understand that we are not bothered about how much oxygen is there. We are bothered about how many carbon dioxide is there. Done. Yes, Ammu. Okay, perfect. Okay, I'm not going to do the disorders because we have done a separate video for disorders. Now answer this question, all of you. Select the favorable conditions required for the formation of oxyhemoglobin at alveoli. They are talking about association. They are talking about association. You can also think about your left shift. Answer quickly in the chat box. Fast. We have to start the next chapter. Vega. Okay, I'll take a Vicks. Okay, I'm doing ads for Vicks students, but no one is paying me. I don't love rhinovirus. Rhinovirus loves me. C. High PO2, low carbon dioxide, less. Okay, excellent. Yes. What's the answer? Yes. Okay, very good. Very good. Next question. The partial pressure of oxygen and carbon dioxide at alveoli. Okay, they're asking at alveoli what is the exact pressure of, partial pressure of oxygen and carbon dioxide. Come on. Oh, 11, 11. Faster. C. This also is C. Very good. Very good. 140 and 40. Okay. Students, do this question for me. No time to draw full thing. Answer this A and B. A and B. Yes. Tell me faster, students. A and B. What is the cells on A and what is the cells of B? Okay. No time to draw the whole thing. So just assume and tell me the answer. Faster. A is squamous. Excellent. What's squamous? Squamous epithelium. B is? B, I can't see. Faster, 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 faster. Okay, let me see how many of you marked your attendance. Yes, 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 yes. Done. Very good, very good. Endothelium. Okay, endothelium. Good job. Next question. Okay, let me see. Yeah. Select the correct events that occur during inspiration. Yes, very nice question. Answer this question as well. Contraction of diaphragm, contraction of external intercostal muscle, pulmonary volume decreases, intrapulmonary pressure increases. Okay, Lokesh. Next time I'll give you a tour on what backgrounds and music. Now answer this question. Faster. Faster. They're asking you the flow. Flow, 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 flow. Just do the authentication if you have used your PAN card. C, you're telling 1 and 2. Okay, select the correct events. Okay, contraction of the diaphragm, agreed. Which one are you telling? 2. Contraction of the, yes. Pulmonary volume decreases, wrong. Intrapulmonary pressure increases, wrong. Okay, IPP does not increase. Very good. Excellent. Okay, now students, instead of body fluids and circulation, I'll be doing locomotion now. Because the body fluids and circulation is a little more big chapter. So, I'll take little more time. So, we will borrow some time from Basavaraj. Okay. Now, we will do locomotion and movement. Ready? All of you? You're not feeling tired, no? 1 hour I'm taking for 1 chapters. Okay. Some chapters can go that side and this side. But otherwise, you just imagine 1 hour, 1 whole chapter, every single details I've gone into. Okay, let's put some fire emojis and let's start locomotion and movement. Very simple chapter, nothing to worry. All the questions directly you can answer. I 100% guarantee you. Come on. Yes. Let the energy be as high as possible. Okay. Yes. Yes, yes, I'll be taking revision. I'm taking revision, no? Pavam Basavaraj sir, it seems. Oh. He has 3 chapters. I have 6 chapters. So there is no power. Okay, I will beg and borrow. Steal also. I'll tell, break. I will break you people. No, I'll break your bones in locomotion and movement. Break, it seems, after 1 hour. I've become so violent, no, students. Last year also like this only I was very violent after, April 25th. Only scoldings, only hit word used to come from my mouth. Okay, because you are also confident I cannot hit you. I am also confident I cannot hit you. No, so only I can show my frustration by telling the word hit. Okay, yeah. Hey, we are coming on May 6th to Chennai. Okay. So, Pakalam. Got it? Come on, locomotion and movement. Okay. Yes. Okay, let's start with the first thing. Okay. First thing. So, look here, students. This types of movement, see, it's not that complicated and all. You don't have to break your head. It's kuti things. Okay. Yes. Internal intercostal muscles will contract for expiration. Okay. Okay, come on. Now look here, amoeboid movement, okay, amoeboid movement, ciliary movement, muscular movement. So, movement and locomotion, what is the difference, students? Movement does not, see, this is a movement, okay, this is a movement, okay. Locomotion means this is a locomotion, okay, this is a locomotion, I can do moonwalk, okay, yes. So, here what is happening, students? Please try to understand that when I say movement, it's not necessary that I locomote. That means from one point to another point, displacement is not necessary. I can stand and also do aerobics. This is movement. But locomotion means what? I am doing displacement. I am moving from one place to another place. Clear? Done. Now look here. Amoeboid movement, ciliary movement and muscular movements happen in our body. Let's try to learn which are that. Amoeboid movement, students, it is done mainly by your amoeba because of their false feet, pseudopodia. But where does it happen in our body? It happens when WBCs are trying to do diapedesis, macrophages are trying to do work for our immune system. Okay? Done? Yes. Okay. Now, who is asking break? Should I tell again? Huh? Excretion and body fluid we will do. I am not going to skip any chapter. Don't worry. I am just doing different order. I am taking a heavy chapter with a light chapter, heavy chapter with a light chapter. Otherwise, you'll all only ask break. Okay? You will become then light-headed. Head only won't be there. Okay? So, that is why I am doing a heavy chapter, light chapter, heavy chapter, light chapter. So, that way we will go. Okay? Now, look here, students. Amoeboid movement is done by your amoeba because of the pseudopodia. But where does it happen in our body? Question is going to come there. WBCs and macrophages. Exactly where WBC does it? Diapedesis. That is, they are looking at, okay, there is one problem there. They are going to squeeze out of the pores of the capillaries, go and phagocytis. Okay, clear? Clear, 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 clear. Clear, clear, clear. Okay. Okay. Students, one time if you tell, I can read. I can understand. You don't have to spam. Okay, next is going to be ciliary movement, students. Lining of the fallopian tube and lining of the trachea and bronchi. We have already studied this in structural organization. Okay, structural organization. Muscular movement, there's nothing much to learn. Just understand your limbs and jaws. Okay, and 639 muscles are there. And flagellated movement is only shown in sperm. Okay, shown in sperm. Clear to all of you? Yes or no? Yes or no? Yes. Okay. Okay. Very good. Very good. Next one. Properties of muscle, EEC, EEEC, okay, E for excitability, that means the ability for them to take the stimulus, to take the impulse from the neural system and get excited, okay. Second one is extensibility, the ability for them to extend and also come back to their original position, okay. Contractibility, the ability to contract, and elasticity, again, to stretch, to be flexible, at the same time come back to their original length. Come back to their original length is the properties of the muscles. Now students, please understand, nervous system, skeletal system and muscular system should work in coordination, or else it will be like how these actresses are doing ramp walk. Have you seen? Leg and hand will not be coordinated. They'll be doing like this and then they'll realize, why am I not able to walk? Why? Because these three systems are not in coordination. Nervous system is telling, why are you doing this? You are not even a model. Okay. Skeletal system is telling, I am voluntary, I can walk, but you are not walking like a model. Then muscular system is like, okay, I am contracting and relaxing, but it is not coming in proper movements. So these three have to be coordinated for you to walk like a model. Clear to all of you? Yes or no? Very good. Now, okay. Now look here, students, three types of muscles are there. Okay, three types of muscles are there. Which are they? Cardiac muscle. Okay, cardiac muscle, smooth muscle and skeletal muscle. Cardiac muscle means what? Ma'am, we know, ma'am, it is in the heart. Okay, and my heart is not in control. That means it's involuntary. I cannot control it. Ma'am, and these people are branched. Okay, these people are branched. Right. And these people also have striations, or they might not have striations also. Okay. So, striations are present. This is the characteristics of your cardiac muscle. They will give you the muscle diagram. They will ask you the characteristics, or you have to ask the characteristics. Clear? Okay. Yes. Now students, look at it, smooth muscles. Smooth muscles means, first of all, you should know these people are fusiform shaped. Fusiform shape means very fancy word, that means tapering at both the ends. Tapering means what, like the sharp ends, okay? Tapering at both the ends. These people are present in the visceral organs. Visceral organs means what, in the lining of your stomach and all of this. So this can also be called as your visceral muscles, okay? These people are also involuntary. Okay, these people are also involuntary. They are not branched and they are not striated, okay? We can tell not striated or not striped. Clear? Done, done, done? Yes or no? Why are you telling Ada? I didn't even ask you a question. Huh? Okay, next one here is the one that you are going to study right now, your skeletal muscles. These people are the only one who are voluntary. They are the only one who are voluntary. These people have striations. They are not branched. They are cylindrical in shape. Even the cardiac muscle is cylindrical in shape. Please remember, these people are not branched, okay? And they are multinucleated, or synthesional word we can use, okay? Done, done, done. Okay, ho gaya. Very good. Very good, okay? Yes, intercalated disc is present there, but again, it's not directly mentioned anywhere in your NCERT. So, intercalated disc also you can write. Now, coming to the exact bundles, okay? Bundles. These are from my backlog series notes, okay? Because there's no time to write notes, okay? Now, students, look here. The bone is there. To the bone, the muscle will be attached. When a bone attaches to the muscle, when the muscle attaches to the bone, that is called a tendon. Now, tell me one thing that you people know about tendon. Ma'am, I know that tendon is a dense regular tissue. So, when you are studying something, try to connect it with other things as well, so that you don't have to revise every chapter. Dense regular tissue. Remember all of you, dense regular tissue, ladri saw a snake fight. Ladri saw a snake fight. So, it is a part of the dense regular tissue. Now, the entire muscle will have a protective cover called as epimysin. Not very difficult to remember. Why? Because epi means outermost layer. Okay. Now, when I take the entire muscle and take a cross section and see the muscle, I notice that inside the muscle there are muscle bundles. What are these muscle bundles called? All of you tell me quickly in the chat box. What are these muscle bundles called? Yes, dense regular tissue. Dense regular tissue. Now it's easier than before. Very good. Okay. Yes. So, entire epimysin is there, muscle is there. Now I take a cross section of the muscle. I notice that inside that muscle bundles are there. Okay. Muscle bundles are there. Okay. Tell me. Yes. These muscle bundles are called as fascicles. Okay. What are these muscle bundles called as? Fascicles. Okay. Now students, I gave muscle a covering. Fascicles will complain to me. So, I will give fascicles also one covering. Got it? Excellent. Now, fascicles also have a covering. That covering is called as perimysium. Peri, between, okay? Epimysium, perimysium, endomysium. Epimysium, perimysium, endomysium, okay? So, I gave that. Now, muscle bundles, no students. Many people come together, they fight and all that. So, we want to make sure that these people are all in position. No problems happen in the future. So, we have a collagenous sheath which is running between all the muscle bundles, which are called as fascia. Okay, fascia. That is collagenous sheath which are connecting all the muscle bundles together. Now, when I try to pull one muscle out of it, okay, I notice that my muscles are called as muscle fibers. Why? Because these people are slender and long. Okay, because they are long and slender, I call it a muscle fiber instead of a muscle cell. Okay, so when I pulled it out, I pulled one more thing out of it, which is like a structural unit of it, which is called your myofibrils. And in the myofibrils, when I go deep into it, I understand that these people are made up of contractile protein called myosin and actin. Done, 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 myosin and actin. Done, 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 that is your entire muscle. So, if you look here also students, this entire diagram will show you exactly that. So, look here, this is your entire muscle. Muscle has an epimysium, and if you go inside, you can see many bundles, and if I try to pull out one thing out of the bundle, I see that, okay, the entire thing that is coming out is a muscle fiber. When I go again pull it out, I notice that there is a myofibril, and if I look into the myofibril, I will see actin and myosin filament. Okay? Okay? Yes or no? Yes. Okay. Now, students, the endoplasmic reticulum of muscle is called as sarcoplasmic reticulum. Sarcolemma is the plasma membrane. Sarcoplasm is the cytoplasm of the muscle. Clear? This much words you can understand. Yes. Come on, Teju. Let's make the target. Okay, very good. Understood? Now, sarcomere. Okay, sarcomere. Yes. Students, what is sarcomere, students? If you go back to this structure, you will notice that in the myofibril, there are proper, like, sections are there like this. Okay, sections are there. And no one is understanding what is the section. Then we get to know that this is the actual unit of contraction. That means contraction is happening because of these sarcomeres. So, what did we do? We took a small portion of the sarcomere and now I'll show you magic. Okay, and we put it here. Clear? Okay, put it here. Now look here, all of you. What is happening? The Z line, okay, the pink color line which is going is called as the Z line, and these two are your actin filaments. Many actin filaments will be there. Now we have only taken 2 actin filaments to show you, and this line which is running in between is called the Z line or zigzag line. What is it called? Z line or zigzag line. Okay. Now students, this Z line is going to make sure that actin remains in place. Now, if you come to the middle, you will be, ma'am, actin only is there, myosin is not there. Myosin is, okay, my God. Myosin also is there. This myosin is a thick filament. Myosin is a thick filament. Clear? Yes. So, this thick filament is going to be in the center. Now, to keep this thick filament in place, I have an M line. What line I have? M line. M for myosin, M for M line. Z for actin, A for actin. Okay. Z line which will hold the actin in place. Now, listen to me carefully. You already know all of this, so it is going to be very easy for you to understand. Now, when I am thinking of contracting my muscles, my Z line, these two Z lines are going to move inwards. Okay, these two Z lines are going to move inwards. But there are some bands and zones in the sarcomere. Okay, first band that you have to study is isotropic band, where only actin is present. Iso means same. So, only actin is present. So, can I call it isotropic band? Yes, ma'am, you can call it. But I noticed there is another region next to it where actin and myosin is slightly present. So, that means I am going to call it anisotropic band because they are not of the same type. Okay. So, this portion will be anisotropic band. Clear? Where actin and myosin is there. This exact center portion. Okay. Now, students, A band. A people, that means A class people do we tell? Like VIP people we tell, no. VIP people are like the A, like how we get A in our classes also. Oh my God, topper. Like that, this fellow is a little VIP. Okay, remember this fellow. I will ask you later question. Now, look here, students. Other than 2 bands, isotropic and anisotropic band, we have zones. Which are the zones? H zone, O zone. Okay. Now look here. O zone is the region which is, from the name itself, overlapping zone. That means where actin and myosin are overlapping on each other. Okay. Now this is my elbow is the Z line. This is also my Z line. Okay. Now just imagine I want to contract. My Z line comes forward. Will my overlapping zone increase or decrease? Tell me this, all of you. Will my overlapping zone increase or decrease? Ma'am, obviously, ma'am, if someone push from both the sides, your overlapping zone is going to increase. Correct? Right? Because actin is going to slide over myosin and move forward. So, the only zone that increases due to contraction is your O zone. Okay? Okay? Yes? Okay. Now, look here. O zone, overlapping zone. H zone, students, myosin also will feel bad. No. That for actin, you people give isotropic band. For me, you're not giving any place. So, we are going to give him H zone, which is called the Henson zone, the region where only myosin is going to be present. The region where only myosin is going to be present. Clear to all of you? Yes or no? So, A band, anisotropic band, I band, isotropic band. Isotropic band is where only actin is present. That is your thin filament. Okay. Now, look here, students. Contraction. When a contraction happens, this is your Z line, this is your Z line. When contraction happens, what is going to happen? Z line is going to move inwards. A band is going to be constant because A band is VIP. VIP will be like, hey, don't push me, yaar. Huh? Go that side. I will not move. So, A band is going to be constant. M line also is constant. Because he is staying with A band, no? He got influenced. Okay, these people are like, I am an MA. Okay, I am an MA. I will not move a little bit also. So, A band is going to be constant. Okay, I am doing so much of acting to make you remember everything. A band is constant, M line is constant, Z line reduces. Okay, Z zones will reduce. Why? because actin will start coming and taking over myosin's place. Okay, ozone is the only one who is increasing, and I band also will decrease. Clear to all of you? Huh? Clear? We don't have to tell which band key inside zones are coming. Okay, let's take them as entities. Okay, separate entities. Huh? Clear? Clear? Oh, Ninja doesn't have phone right now, but he is on YouTube. Wow. If anyone can decode this question, okay, 5 marks guaranteed. Come on. Ninja doesn't have phone, but he is right now on YouTube. How come? Okay. Yes, I understand he doesn't have phone. He's watching on PS, but he's telling that he cannot Google something also. So again, that becomes the confusion. Okay. I'm not good at acting, students. I'm really horrible at acting. Okay. Like if you ask me to act, I feel like crying. Yes. Janani laptop use pannuva. But Ana on Google use panna maate. Edukku? Tell me. Edukku? I did not clear the question. Now the question, second part of the question is coming. He is telling I'm not going to Google because I don't have phone. Okay. So that is the question actually. Okay. Now listen here. I band isotropic, A band anisotropic, H is Henson zone, O is overlapping zone. Now one more question that can come for all of you is what is one sarcomere? Sarcomere is what? Sarcomere is between two Z lines. Okay. Two consecutive Z lines, a sarcomere is formed. Okay. Two consecutive Z lines, a sarcomere is formed. Clear? Yes? Now, look here students, one sarcomere has half I band. I am not giving the full I band. Why? Because sarcomere starts from one Z line to another Z line. So, this much only I can give. So, this is one half I band, this is one half I band, but I can give one full A band. I can give one full A band. So, half I band plus half I band will give me an entire sarcomere, that is one I band plus one A band. Okay? One I band plus one A band. Clear? Clear to all of you? Yes. Fine. Got it. Very good. Functional unit, or we can say a contracting unit. Okay. The unit of the muscle where the exact contraction is going to happen is the sarcomere unit. Okay. From one Z line to another Z line. Okay. So what you have to remember? You have to remember which of the zones are getting reduced, which are constant, which is not, which is increasing. This much only you have to remember. Now let's go to myosin. Okay, let's go to myosin. Myosin, students, is made up of monomers of meromyosin. Monomers of meromyosin. That meromyosin is made up of 6 polypeptide chain. 6 polypeptide chain means 2 long chains, 4 short chains. 2 long chains, 4 short chains. Okay. Myosin has a head and a tail. Just like you people also, they have head and tail. Okay. Now, okay, you people don't have tail. Sorry. Head you have. No, yeah. So, the head is a globular head. Actin binding site will be present. ATP binding site will be present. Please do not think that this ATP binding site and ATP is different. Imagine enzyme and substrate. Enzyme is different, substrate is different. If substrate comes and binds only, the whole thing will happen. No, same way, ATP binding site and ATP is different thing. Many places they will give you ATP and many students will answer ATP, but that's not wrong. That's wrong. ATP binding site is present. Clear? Yes. Okay. So, look at the chain, students. This is your entire tail. Okay. 2 long polypeptide chain. This is the 2 long polypeptide chain. And the short chains are what you're seeing here, the white color. White color are the 4 short chains. So, tail, head. Tail, head. Head is globular. On the head, actin binding site is there, ATP binding site is there. Okay. Now, this like how we have neck, like that one small region called as the short arm is there. So, question has come majority time question has come, where does the heavy meromyosin is present? Students, heavy and light meromyosin is just like your chemistry here. They are based on molecular weight. Okay. Tail will have you seen any dog's tail that is heavier than their head? Of course not, no. So, tail will be light meromyosin, head and the short arm, like your neck, I am telling you to remember, okay. Head and short arm is going to be heavy meromyosin. Okay. Yes. Clear to all of you? Wow, you people are self-trolling at this point. Huh? I don't even have to do my job. Done. This is your myosin. Now going to actin. Clear to all of you? Thumbs up. Thumbs up. Okay, now look here students, structure of actin. There are some globular actins which were present. They did polymerization. They were like, our structure is not actually helpful, so let's do polymerization with the help of magnesium ions. Many students ask me, ma'am, which ion, which ion? With the help of magnesium ion, they are going to form like an infinity structure, okay? That is, this infinity structure will have these globular actins, and this will ultimately form the F actin. This also question has come. Fibrous actin or F actin, they are helically coiled. Okay. This also students, PYQ question. See, everything I write in my slides, okay? You people only don't download. Students, if you want to join my Telegram channel, you can join my Telegram channel, Gopika Biology Official, okay? Where you will get to talk to me, meet, like, have some motivation, okay? Random facts, notifications. Most of the time, I'll give you motivation at the right time that you need, okay? So, yes, you can follow me on Gopika Biology Official channel. And, yes, let's be connected, okay, even after NEET. So, look here, students, globular actin forming F actin. F actin ke baad you should notice that on the actin there are two villains. Okay, the story starts right now. Okay, the interesting story starts right now. Two villains are present. Again, we are going into a marriage story. Okay, I am basically serial director. Okay, to make you teach, I'll tell any kind of example. Okay, so look here. F actin, two villains have come here. Here we have one villain. Who is the one villain? ATP binding site is the one villain. Okay, this is the father of myosin. Okay, here actin is the girl. Actin has two brothers. One is called as troponin and one is called as tropomyosin. Ma'am, where are they present? This tropomyosin is that rope-like structure which is going. Troponin is like three-head structure. Okay, that is, one structure will be TPC, one will be TPT, one will be TPI. You remember TPC. Most importantly, you remember TPC. Okay, yes. Hi Kavita. Okay, look here students. Now, this troponin and tropomyosin are the brothers of actin. I'll tell you why. We are going into that. Okay. So, I am going to call them the regulatory proteins. Please do not get confused with contractile protein and regulatory proteins. Contractile protein, who is going to contract? Regulatory protein, who is going to regulate? Right? Simple English. Contractile protein is actin and myosin. Regulatory proteins are who is going to regulate actin. Should you go to college? Should you not go to college? Should you study? Should you not study? So, troponin and tropomyosin. Okay. Now, students, these people are actin. See, this TPI, TPC, TPT or troponin and tropomyosin have masked myosin binding site. Actin likes myosin, myosin likes actin. But they have made sure that these people are not masked. These people are masked. That means they cannot see each other. Okay. Now, we are entering into the story. Okay, we are a set of people with me, Gopika ma'am army. Okay, Gopika ma'am army and Gopika ma'am has entered into the entire scene. Now, look here. First, I'll tell you the stories. Then, I'll tell you the scientific part of it. Look here. Neuromuscular junction. Sorry, first I'll tell you the scientific part, then the story. Neuromuscular junction. I told you, one of the properties of the muscles is to get excited. Correct. One of the properties of the muscle is to get excited. So, when they are getting excited, that region where the muscle and the nerve is there, that is called your neuromuscular junction. So, your axon is there, muscle is there. They are attached like this. This is called a neuromuscular junction. Neuromuscular junction. Okay. Thank you, Veena and friends. So, now look here, students. The entire story goes like this, where we are going to make sure that the brothers of actin is going to get distracted so myosin can come and bind. This is the whole story. For that, we need a stimulus to come, an impulse to come. Now, one stimulus is carried by the axon and it is coming here. Obviously, space, they are going to have neurotransmitters, they are going to have chemical, they are going to do chemical synapse. In the neuro, that inside the synaptic vesicle, there is acetylcholine. That acetylcholine will be given to who? To the muscles. On the muscles, receptors are there. This acetylcholine will be taken up by the receptors. The moment, you know, even there also, when impulse is coming, sodium influx will happen. Here also, sodium influx is happening. But on where it is happening exactly? It is happening on the T tubules. T tubules. These are the structures which are present inside the muscle. Okay. To the T tubules, there is an influx of sodium. because of the continuous influx of sodium, sarcoplasmic reticulum, endoplasmic reticulum of muscle, sarcoplasmic reticulum is a storehouse of calcium. Storehouse of calcium. That calcium will be released, which will go and bind with TPC and do a conformational change. Now let's come to the story. Story is that we have all come. We are this. Okay, we are the acetylcholins. We have all come. We have all got into the house. Okay, this is Actin's house. We have got into the house. After we enter into the house, we decide to create a fake fight. That is the influx of sodium. That means full. We are all running and we are going to fight. Seeing our fight, one girl, okay, whom I have assigned. Let's say Suvra is whom I assigned, okay, to go and call TPC. So, the moment the fight is happening, the moment the influx is happening, Suvra will run and go and tell one of Actin's brother, that is troponin TPC, that you know what, inside your home big fight is happening. The people who came here, the friends of Actin, is creating a big scene. What will happen? Troponin and tropomyosin will get distracted. Will do a conformational change. Will get deviated. We can say all that words. No. They will deviate, and when they deviate, the myosin binding site will be visible on actin. That means actin is ready. Actin has the guts to go and meet myosin. All of this we only did. Okay? We only did. Okay? Yes? Clear to all of you? Huh? Clear? Yes or no? One second. Has the guts to go and meet myosin. Okay. Yes. So here, students, what has happened? Suvra ran and told troponin that you know what, in your home such a big scene is happening, why don't you come? So they both will run to solve the issue which we have created, fake issue. Okay. We might get belted after this, but that is the secondary story. We don't have the climax of the story. All my serials end in the mid part. Okay. So students, that leads to a conformational change, unmasking the myosin binding site. Done? Clear? Now, second task, to make sure that myosin's father leaves the place. Otherwise, these both are not going to bind, right? So, this one is clear to all of you, yes or no? So, axon is bringing the impulse, neurotransmitters are there, synaptic, that is the synaptic vesicles will open, acetylcholine will be released, they will be taken up by the receptors, influx of sodium will happen, influx of sodium will lead to the release of calcium from sarcoplasmic reticulum, calcium will go and bind with the TPC on the tropomyosin, doing a conformational change, automatically leading to the unmasking of myosin. Done. Done. Clear? Now, second task. We need to remove myosin's father from myosin's head. Right? Now, for that, what can we do? Main problem is that ATP binding site has ATP. So, we have to break ATP. So, again we go for myosin's home and we break ATP's legs. Okay, we break ATP, that is called as hydrolysis of ATP. We break ATP into ADP and PI. Okay, we break his legs. Okay, and then students, what will happen? Automatically actin and myosin will meet. Okay, climax of the story, actin and myosin will meet. That is called as cross bridge. Okay, because they are from different states, so it is cross country marriage, no? Like a cross bridge formation, it will happen. Now, after the cross bridge formation, what will happen, students? We broke their legs, no, ADP and PI. He'll get frustrated. So he'll be like, okay, whatever, he will walk out. Just like that, from the head, from the myosin head, ADP and PI will move out, leading to a power stroke. Now, let's say actin and myosin was attached like this. Okay. The moment this ADP and PI lives, myosin head will move like this. So, there is a space created between them. That will lead to actin sliding and going. Okay, actin is Papa, one light-weighted girl, students, one small actress. She will slide and go off. Okay, so that will lead to power stroke, breaking the cross bridge, and automatically when cross bridge breaks, the father will find myosin. Father will be like, come, let's go home. So, new ATP will come and bind. Clear? New ATP will come and bind. This is your sliding filament theory. If someone listens to all this story, they are going to be like, what is this ma'am telling? Okay. Okay. Yes. So, actin filament, we already did conformational change on actin. Now, what did we do? We broke myosin's leg into, that is ATP2, ADP and PI. Hydrolysis happened. Now, that will lead to myosin head, that is going to lead to cross bridge formation. Now, the cross bridge formation, students, what is going to happen? After some time, ADP and PI will be forced to go outside, because of which breaking of cross bridge will happen, and actin is a thin person, it just slide and just go. Okay, so here sliding and rotation will happen. The moment sliding and rotation happens, there is a space created between them, due to which a new ATP will come and join, and this will lead to again the entire cycle continuing. Your muscles have now contracted and it is going back to relaxation. Clear? Interesting story, ma'am. If you don't write an exam and come now, then it is there for you people. Okay? Clear? Yes? Yes. Oh ho ho ho. Kolaveri song these people are making, creating. Classes, I mean not class. This is done? Yes? Okay. Perfect. So sliding filament theory also is done. Okay. This is given by okay. Okay. Now, see students, this is what happens. Okay. So this Z line will contract. How? When the troponin, tropomyosin all contraction happen, this Z line will move forward. This will lead to contraction. Can you see how the actin and myosin is binding? And then they will slide over. Okay. That time you can see calcium ions are mentioned here, because calcium ions are the most important. Only if calcium ions go and bind with TPC, then only the entire conformational change will happen. Okay. Yes. Thank you, Siddhappa. Okay. Yes. Okay. Done. Done. Okay. Now, students, we'll go to skeletal system. Skeletal system, that much only in locomotion, entire muscle thing. Okay. Very simple chapter. Very easy questions will come from the chapter. Okay. Yes. Correct or not? Correct or not? Yes. Okay. Yes. Now, look here. Axial skeleton, students. Axial skeleton means what? If you take your body, this, I'll break the glass now. Wait, 1 second. This is your axial skeleton. Okay. This is your appendicular skeleton. Okay. All of you do aerobics with me. Okay. Axial. Okay. Axial. Okay. Appendicular. Okay. That means your girdles and your forearms and your legs. Okay. And axial skeleton, skull, sternum, cranium, vertebral column, all this is coming under axial. Okay. Now look here, students, you only have to know the number here, nothing to take tension. Okay, don't worry anything. Look here. Axial skeleton total is 80. Okay, how many bones all of you have? Before your parents break it, tell me the exact number of bones that you have. Sorry. Faster. How many bones total? Total bones. Aerobics, no, not exercise. 206. Correct. 206 bones are there. Okay. In that 80 bones are going to be axial students. So look here. Skull, vertebral column, sternum, ribs. This much you have to remember. Okay. SVSR. Okay. SVSR. Got it? Yes. SVSR is your skull, vertebral column, sternum and ribs. So whatever we learned in the thoracic chamber, no, that same thing we have to remember here also. Okay. Now look here. Axial skeleton, students, skull, cranium, facial bone, ear ossicles. That day only I told you people, hyoid is not a part of the skull. But now they corrected in the new NCRT that hyoid is a bone which is present in the base of the buccal cavity. It is not to be considered a part of the skull, because it is coming here. How do we connect it with skull? But everyone were teaching skull, so I used to write it there, but I used to not write it under skull. Okay. So students, look here. Cranium, 8 bones are there. The brain box, the skull. Okay. Cranium is your brain box. Okay. 8 bones are there. Facial bones are 14, but luckily you do not have to study the facial bones, because only one image is given for facial bones. Okay. Only one image is given for facial bones. There is nowhere written all the 14 bones clearly. Okay. Then ear ossicles, hyoid bone, sternum, vertebral column and ribs. Now, the trick to remember all of this, I'll tell you people. Look here, students. To remember the cranium bones, what's the trick? Yes, very good. Excellent. Step off. Cranium, you remember like this. Step off. That is when someone is eating your head. Okay, what will you tell? What will you tell? Step off. Like, don't eat my head, just stop it. Okay, so same way here also. S for sphenoid bone, which is also called the keystone bone because it articulates with all other bone. Okay, T for temporal bone. Okay, temporal bone. E for ethmoid bone. P for parietal. O for occipital. F for frontal. Got it? Frontal. Clear? So, step off. Okay? Step off. So, look here students. You have temporal, parietal, occipital. Now, here, remember the only people who are going to be paired. The only people who are going to be paired in your school also you would have noticed PT teachers always come in pair. So, only the PT teachers are going to be paired. That is parietal and temporal are going to be paired. So, parietal to temporal to frontal, sphenoid, ethmoid. Sphenoid, please remember they have the sella turcica, that is the part of the chemical coordination chapter. Sella turcica is a small depression that holds the pituitary gland, and sphenoid bone articulates with all other bone. So that's why it's called a keystone bone. Okay, now look here students, cranium is done. Now facial bones you don't have to study, don't waste your time trying to learn all the facial bones. Okay, facial bones you don't waste your time. Facial bones are 14 of them. Okay, 14 facial bones are there. Next is ear ossicles. Ear ossicles, you have to know the name. So, remember the trick, Miss has. Okay, Miss has. Miss has means with the shape and the name. Okay, M for malleus. Okay, malleus, incus, stapes. Okay, stapes is the smallest bone and the shape is going to be has. Hammer shape, anvil shape and stirrup shape. Okay, yes, butterfly shape bones. Clear? Okay, so ear ossicles also you know. And students, hyoid bone is a tongue bone which is present at the base of the buccal cavity. This time they might ask you for location because it changed from the old NCERT to new NCERT. There are chances that they catch hold of that. Okay, please remember it is present at the base of the buccal cavity. So here you see skull 8 bones, step off, cranial, facial bones 14, ear ossicle 6, 3, 3, malleus, incus, stapes, hammer shape, anvil shape, stirrup shape, Miss has. Hyoid bone, tongue shaped bone which is only a single bone which will not articulate anywhere and it is also present at the base of the buccal cavity. Now sternum, vertebral column and ribs we will learn. Students, sternum is this. Okay, I've put the facial bones for you people but don't waste your time, okay, today. Students, sternum is dagger shaped. What shape? Dagger shaped. For me, it looks like a tile shape, but these people are telling dagger shape. So, we have to also take it as dagger shape. Okay? Yes, very good. Yes. So, dagger shape, students, actually there is a prosternum, mesosternum and a sphenoid process. You only remember if you can. They will not like as such ask you a question because again, nowhere it is stressed. Okay. Student, this is sternum is the place where the ribs are going to come and attach. Okay. Sternum is present ventrally. Vertebral column is present dorsally. Ow. And ribs are going to come and connect. Clear? Okay. Now, vertebral column. Okay. Vertebral column. I know that the slide might not be that clear. But here only focus on this atlas and axis. C1. How is the formula going? C7, C7, T12, L5, F5, C4. They will ask you the formula. What do I mean? 7 cervical bone, 12 thoracic bone, 5 lumbar bone, 5 sacral bone, 4 coccygeal bone, sacrum and coccygeal, so my god, okay, 1 second student, my tongue is twisting. Sacrum and coccygeal. Why am I not able to tell? Sacrum and okay, sacrum and this coccygeal vertebrates are fused. Okay, because I'm talking continuously, my brain is gone. Okay. Yes, Nizam is a VIP mammal. Oh wow, so nice you remember. Okay, so nice you remember. Students, I had given a trick for this. Okay, I think one of the student actually gave the trick. Nizam is a VIP mammal. We don't even know what is the meaning of the sentence, but yeah, we were able to remember all the words here. Okay. Okay. N for nasal bone. Okay. Inferior nasal. Okay. Then Z for zygomatic bone. A for what did you? I think Nizam ka M is there. Okay. So if you want, you can remember, students. Okay. Not brain. My tongue is twisting. Okay. C1 is atlas. C2 is axis. Okay. That's the only thing you have to remember. C1 to C7, you write C1, C2, C3, C4, C5, C6, C7. C1 is atlas, C2 is axis. Okay. These two will articulate with each other, and please remember, students, here your condyle is there. Okay. Condyle. That condyle bone will have articulations. Okay. Like this. A skull is dicondyle, we tell. No, that is because it has two condyles like this, which will articulate with these bones. Which will articulate with these two bones, so that they will continue. The entire thing has to continue, no, from the skull vertebral column. So for that, articulation is there. Okay. Now look here, students. Thoracic vertebrae T12. Lumbar is the strongest. 5 bones are there. Sacrum and coccygeal are going to fuse together to form your tailbone. Tailbone. That is why we put bracket. So we write like this. Okay. We write like this. C7. Okay. T12. L5. S5 fused, okay, C5 fused. That means like how you write your, how you write your, this one, plant families formula. Okay, plant families formula. Okay, done. Now, sternum. I already told you about sternum. Flat bone located in the middle of the chest, which is also called the breastbone. And students, this is dagger shaped, okay. Dagger shaped, but for me, it looks like a tie shape more. That's all you have to remember here. Articulates with clavicle and upper ribs. Okay. Now, ribs are 24 bones. Now, ribs, students, remember, true ribs. True ribs means there is only two things that you have to remember here. If a rib is supposed to be called true, okay, then there is one thing that you have to remember is it has to be attached to the sternum ventrally, and it has to be attached to the thoracic vertebrae dorsally. Okay. If they don't follow this two, I will not call it the true ribs. So, true ribs are first to the 7th ribs connected directly to the sternum. Done. These can also be called as vertebrosternal. Vertebrosternal, because they are attaching to the sternum. Okay. False ribs, students, indirectly connected to the sternum. That means they are not attaching to the sternum. Instead, they are taking the help of the 7th vertebrae to attach. Sorry, 7th rib to attach. So, I will call it what? False ribs. It can also be called as vertebrochondral ribs. Okay. Vertebrochondral ribs. Got it? Yes, very good. Now, floating ribs, students, they are not at all connected to the sternum. They are not connected to the 7th rib, 8th rib, 9th rib or 10th rib. They are not at all connected to the sternum. They are free floating. Okay, free floating. Clear to all of you? So, we can call this vertebral ribs. Okay, vertebral. So, remember, they will not directly give you false ribs, true ribs and floating ribs. They will use these words to confuse you. Clear to all of you? So, look at the structure here. This is the manubrium or the prosternum. Okay, body and sphenoid process. I told you, no, the sternum. Now, look here, students. Here, true ribs are there. 1 to 7, which are attached ventrally to the sternum, dorsally to the vertebral column. 8, 9 and 10, if you see, they are attached to the 7th rib instead of directly attaching to the sternum. Okay, and if you see the last two ribs, T11 and T12, you will notice they are just floating. They are not attached anywhere. This is the tip of them. Clear to all of you? Yes or no? Yes or no? Pakka? Okay, so costal cartilage, that is why again we tell vertebrochondral. Okay, they are attached to the costal cartilage. Then hyaline cartilage also is there, but here. Now, coming to the appendicular skeleton. How many students appendicular skeleton? Appendicular skeleton, you have 126. Okay, appendicular skeleton, you have 126. And what and all will you remember? You have to remember your hands, okay, bones in your hands. legs or forelimbs, hind limbs, and your girdles. This is the only thing who come in your appendicular skeletons. Okay. Now look here. How are you, CM Pandu? This is how you will remember the bones. How are you, CM Pandu? Humerus, radius, ulna, carpals, metacarpals, and phalanges. How are you, CM Pandu? That is humerus, radius, ulna, carpals, metacarpals, and phalanges. Clear to all of you? Okay, clear to all of you? Yes or no? Okay, yes, we'll give you a break. Okay, so humerus, radius, ulna, carpals, and metacarpals. Now look at the number, students. Humerus, only one is there. All of you touch and there'll be, yes ma'am, one is there. Radius, ulna, one each. Correct? If you go here, you cannot feel much, but carpals are 8 of them because they'll be like this. Small, like this, small pieces like this, 8 of them will be there. Metacarpal, students, you can exactly feel your metacarpals. Touch and see, 5 metacarpals are there. Phalanges, how do I remember, ma'am? Can you see the cut on your fingers? Can you see this strikes? So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. 14 phalanges. Okay. So how are you, CM Pandu? That is your 14 phalanges. So that makes a total of 30 bones. That makes a total of 30 bones. Clear? So, forelimbs done. Now, can we go into hind limbs? Yes. So, here students, fly Tarzan, mountains and planets. This is Gopika ma'am production. So, he can go outside also. Okay. Tarzan, I only made, no? So, I can do anything with that. Fly Tarzan, mountains and planet. F for femur, the longest bone, your thigh bone. Okay. Tibia, just like radius and ulna, you have tibia and fibula. Okay, tibia and fibula is this portion. I cannot lift my leg and show, but you understood, no? Then tarsals, which is the portion again, where how we have here, there on the leg also we have. That is our, now remember the number, here it is 7. 7. So that means this entire thing constitute to 29 bones, and we will include the knee bone, which is the patella. Okay, knee bone. So that makes a total of 30 bones. Okay, so fly Tarzan, mountains. M for metatarsals, P for phalanges. Done. Yes or no? Yes. Carpals are 8 in hand, tarsals are 7 in legs. Okay, tarsals are 7 in leg. You have to include patella together, which makes you a total of 30 bones. Okay, 30, that is again 60. Now, 60, pectoral and pelvic girdle. Remember students, girdles are something that is like a circle, like a loop, which is connecting. Okay, so if I want to connect my hands, I will use my pectoral girdle. That is, I am going to use the clavicle, your collarbone, and your scapula, which is here, a triangular structure. So all of you touch your scapula and feel it, that is your collarbone. Okay, there is no beauty bone, there is nothing beauty like that. Okay, collarbone and scapula. This two has attached. Now I need to attach my humerus to this. So in Barbie and all of you remember, when you remove the Barbie's hand, you will notice a ball and socket kind of structure like this. That is your glenoid cavity. Glenoid cavity, like this, when you pull out, you will notice that it has a circular structure. So glenoid cavity will be present to attach your humerus. Okay. So look here students, pectoral and pelvic girdle bones help in articulating the upper and the lower limbs. 2 halves, they are made up of 2 halves. That means obviously body is like that, no? 1 half here, 1 half here. Okay. Now, each half of pectoral girdle consists of a clavicle and scapula. So, clavicle is your collarbone, scapula is your triangular structure, and they have a like a ridge. Okay, triangular flat bone situated in the dorsal part of the thorax between the 2nd and the 7th rib. So, here location they might ask you. Okay, question, location. Where is it located? Where is it located? 2nd to 7th. Okay, between the 2nd and the 7th thoracic vertebrae. Okay. The dorsal flat triangular bone will have a ridge, which is called the spine. Okay, which is called the spine. Now look here students, there is a slightly elevated ridge called the spine, which projects as a flat expanded process called acromion. Okay, acromion. So the clavicle articulates with this below the acromion. So students remember, scapula should get attached. Okay, scapula and clavicle should be articulated. Yes ma'am. So for that I will use what? Acromion. Okay, acromion. Okay, now scapula and clavicle got attached. My pelvic girdle is done and dusted. Now I have to make sure that my humerus is getting attached to these people. So to attach my humerus, we have a glenoid cavity. Understood all of you? Glenoid cavity. Clear? Glenoid cavity. So, below the acromion is a depression called glenoid cavity which articulates with the head of the humerus. So, whenever you're thinking about Barbie, okay, I want you to remember glenoid cavity. So, hand, you remove the Barbie's hand. So, there will be a round structure, so think of it glenoid cavity. Okay. Now, students, if you go further and try to understand, this is clear, nothing is there. Scapula is there, triangular structure which is present in the dorsal side, clavicle is there which is the collarbone. These two will articulate with the help of acromion process, and below the acromion is the glenoid cavity to attach the humerus to it. That's it. That's it. Break I'll give you. Break I'll give you. Okay, I am the one who is continuously talking. Why do you people need break? People are there no. Now, pelvic girdle consists of two coxal bone. Students, you remember this trick, okay? I play in class. I play in class. Okay. I play in class. Yes, ma'am. We all play in class. Whenever teacher is teaching, we all talk in class. Online class, offline class, everything we talk. Okay. So, students, how you are going to remember? What is the trick for this? See, I play in class. I for ilium, P for pubis, I for ischium. Okay. And C for coxal bone. Coxal bone. That means these three bones together forms the coxal bone. Okay, coxal bone. I play in class. Tell. Okay, I play in class. So, ilium, ischium and pubis, at the point of fusion of the above bones, there is a cavity called acetabulum. Acetabulum. Here, only word that you will learn is acetabulum. Clear? Acetabulum, students, to which the thigh bone articulates. So, I want to attach my femur. For my femur to attach, I have an acetabulum. Here I had glenoid cavity and acromion process. This two attachment, this one attachment. Here I have only acetabulum. Okay, acetabulum. That is, the two halves of the pelvic girdle meet ventrally to form the pubic symphysis containing fibrous cartilage. Okay, clear to all of you? Yes or no? I play in class. Repeat after me. Otherwise, for trick I have to make another trick. Now, students, from here lot of questions come, but exactly questions come from synovial. Okay, because synovial is too difficult to remember. So, they think like that, but we are going to make sure that it is not difficult. Now, there are joints in our bodies, right? To keep everything together, there are joints in our bodies. And the first joint here is your fibrous joint. What joint? Fibrous. Students, fibrous joint is like the very strict principle. He does not let any movement. No one can move in the class, no one can breathe in the class, nothing will happen. No movement at all. Fibrous joint is no movement. An example for that is sutures. Where is sutures present? In your skull. Sutures are present in your skull. Not at all movable portions. Okay. Only you remember the example. Sutures, fibrous joint. Now we got one more. Let's say 3 phases of our life. First we met a more strict principle. It's like don't move, don't talk, don't breathe, don't do that. Fibrous joint. Then next time we got one more better principle which is cartilaginous joints. Okay. Cartilaginous joints. This fellow will only allow at least some movement. Okay. Like limited movement. Okay. Limited movement. That means we can breathe. Okay. We can move a little. But again, not extreme movement but limited movement. This example is going to be the cartilage which is present between the vertebral column, which is allowing you to do the bending, moving, everything like this. Like this. Can you see the cartilage which is present between the vertebral column? That is why you are able to bend, you are able to lift your leg, all that you are able to do. Okay. Then last, students, after praying a lot, before we were about to get graduated, one nice principle came who allows more flexible movement, which is your synovial joint. Have you always noticed when we leave the college or school, no, suddenly school will start becoming one 5-star hotel. Okay, swimming pool will come, badminton court will come, volleyball court will come. It's because we are not a bad luck. It is just that they have money now. Okay. Who is that fellow? NEET without biology is heaven, it seems. I'll take and throw you. Okay. Papa, students are passing only because of biology. You don't want me to have job, huh? You have a problem that I'm teaching biology, huh? We will boycott him, students, after some time. Now we will finish synovial joints. Okay, synovial joint is the last principle that came, who is very, very good man. Okay, very, very good man. Why, I'll tell you. But he has multiple, subheads. Okay, so synovial joints are fluid-filled synovial cavities between the articulating surface of bone. This allow considerable movement, like he is considerate. Okay, good Pavam principle, considerate. Now what you have to learn? What you have to learn is that first joint that is there, students, is ball and socket joint. Where did I tell you ball and socket joint? This one. Think of the Barbie. Ball and socket joint is between humerus and the pectoral girdle. Okay, same thing goes with pelvic girdle and femur bone as well. Okay, yes. Now look here. Then students, hinge joint, that is your knee joint. Okay, hinge joint is your knee joint. Yes. Students, come sir.
You're doing footballer?
Yeah, I'm actually for full aerobics I've done.
Full aerobics, sir?
Yeah, full.
Nice, nice. Ma'am, how's it going, ma'am?
It's going good. Sir, but they want a 5 minutes break.
Yeah, we'll give them, ma'am. Otherwise, they'll go mental.
Yeah.
They'll not understand a single thing. They'll write that answer here. Okay.
Yes.
So we'll give them a small break, ma'am.
Yes.
Yeah. Now, how much of time?
Over. This is the second chapter, is last topic. Yeah.
Okay, ma'am. Okay. Cool.
Okay. Yes. I have maintained my time, students. I told sir that I'll finish. Okay. Yes. Thank you. Now, later you tell hello, bye, sir, and all. Sir will come again. That time you will tell sir, please go and bring Gopika ma'am here. Okay. So many fans, you know. So I tell sir, okay. I tell sir that students are going to ask for me. Okay. So don't tell hi and all. Now come. NEET without physics, chemistry, biology is heaven. Exam only won't exist. Okay, then you will be in heaven. Oh, no exam only. Okay. Now come back to class. Come back to class. Okay, so ball and socket joint between humerus and pectoral girdle, pelvic and femur bone. Hinge joint. Student, hinge means what? In door and all, have you seen when we open the door, the side plate is there, no? That is hinge joint. So like that only a knee joint is. You bend and you do this, you bend and you do this. Understood? You bend and do this, you bend and do this. So that is your hinge joint. Okay? Pivot joint. Pivot joint, atlas and axis. First cervical and second cervical. Atlas and axis, you have pivot joint. Okay? Pivot joint. Now, gliding joint. Gliding means carpals. Gliding. Okay? When carpals and metacarpals talk to each other, they feel sad. So, it is saddle joint. Okay? Saddle joint. Clear? Yes? Okay? Done? Pakka? So, saddle joint, it is between carpals and metacarpals. Gliding joint. Okay? Gliding joint is carpal. Hinge joint is knee joint. Okay? Pivot joint is atlas and axis. And your ball and socket is humerus and pectoral girdle. Clear to all of you? Done, done, done? Done, done, done? Any doubts? Any doubts? Students, I want to show you people questions, but I don't know where is the questions. 1 second. Okay, listen here. So, I want to attach, but that's a very big file. Let me try, students. Just give me a minute. Just give me a minute. Okay, clear to all of you? Yes, for sure. For sure. Done. Locomotion is done. Okay, locomotion is done. 1 second. I'm trying to open the file, students, where we had all the questions. Just give me a minute. Okay, not this. Not this. Yeah. Since here, let's go to locomotion and you do some questions, okay? These are the NEET PYQs. So, we can do it from here only. 1 second, let me check where is the questions. Breathing and exchange. Circulation. Okay. Yes, 1 second. Excretion. Locomotion. Okay, students, first question. Oh, no. Yeah. Yeah. First question on the screen for all of you. Okay, answer the first question. Can you all see the question? 1 second. Yeah. Yes, answer quickly. 5. If they ask you how many bones make up the tailbone, that is sacrum plus coccygeal. Okay, 5 plus 4. Okay, but if they are asking you only sacrum, it is 5 bones. Only coccygeal is 4 bones. They together makes the tailbone. Clear? Yes, answer the question. List cartilaginous joint, ball and socket joint, fibrous joint and saddle joint. Like this questions will come. So you have to remember, SAD is when carpal and metacarpals talk to each other. Carpal alone is there because they're kuti, they will glide. Okay, gliding joint. So remember like that and answer. All of you are telling A, is it? Cartilaginous joint, it is between adjacent vertebrae in the vertebral column. A2. Okay. Ball and socket is between humerus. B4. Okay. Fibrous joint, it is between the flat bones. C1. Okay. Yes. Excellent. It is option number A. Very good. Very good. Okay. Option number A. Next question I'll show you. 1 second. Yeah. Which of the following is a correct match of diseases? Okay, we didn't do diseases, but you will remember because I did disorders very recently. Arthritis, inflamed joints, tetany, high calcium level, myasthenia gravis, genetic disorder, muscular dystrophy, autoimmune disorder. Do fast. Yes. Tamil theriyum. Edukku? Why you people asking me? Students, we are coming to Chennai on May 6th. Okay, again, I'm reminding you, we will be there in Anna Nagar Center. So, whoever is in Chennai, whoever can travel to Anna Nagar Center, do not fail to come because I'll be more than happy to see all of you. More than happy to see all of you. Okay? Yes. A, this also. Arthritis, inflamed joint. Very good. Why? Because tetany is low calcium levels. If calcium level is low, then contraction and relaxation won't happen fast. Rapid spasm will come. That is because calcium has to go and bind with TPC. Myasthenia gravis is the autoimmune disorder which is actually going to kill the cell. Muscular dystrophy is the genetic disorder. Exchange of these two people. Okay. Yes. Hyderabad mostly on 17th. Okay, 17th. But you people all should be coming to Chennai. Okay, please do come to Chennai. Do not fail to come. Don't be like, oh, who will travel? Okay, you should always meet your teachers, no? When will you meet otherwise? Which of the following is present between adjacent bones of the vertebral column? See, easy questions only will come. Hyderabad 11th. Sorry, 17th. Okay, Hyderabad is 17th after my birthday. Okay, yes. Hey, Emperor, that's all for you. You go with them only. To Thunderbolt, you go and ask them, please teach me NEET. Where in Chennai? Anna Nagar Center in Chennai. Just come to Anna Nagar Center. You can bring your parents, sister, brother, whoever you want to bring, you bring, okay? Yes, cartilage, very good. Second question, chronic autoimmune, we just did the same thing, okay? This we did. Students, one good question is coming. Answer this also, last question. Okay, after this I'll give you a break. Okay? Scapula, cranium, sternum, vertebral column. Cartilaginous joint, ventral part of the rib cage, part of the skull, triangular flat bone. Okay? Yes. Rani, yes. What will you give me if you come to Anna Nagar Center? You're asking me what will I give you? You can see me. Okay? Scapula, cranium, sternum, vertebral column. Answer fast. A. Chumma don't tell me. Scapula, triangular, flat bone. A4. Okay, two options we have, A and B. Cranium, part of the skull. B3. Option number B. Yes, very good. Option number A, why are you telling? Option number B. Okay. Yes, very good. Okay, done. Okay, I'll come and see you again for body fluids and circulation, and maybe we will do neural control and coordination. Okay. After that, we'll do excretion and chemical coordination. Done. Don't go anywhere. Okay. Don't go anywhere. Be here only. Okay. Real warriors are people who stay back even when things are not going, like they planned. So resume back. Students, only 5-10 minutes break, okay? 5 minutes is enough actually. Because more the time we delay for you people only, it will become a problem. 10, 12, okay? 10, 20, sorry, what am I telling? 12:25. Okay, because Basavaraj sir has another class. So he has to finish one chapter, okay? Yes.
Hello students, quickly tell me in the chat right now if I'm audible and visible to all of you students. Quickly tell me in the chat right now if I'm audible and visible to each and every one of you students. Yes? Hello, 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 hello. Hello students, hello, hello. Quickly tell me in the chat right now if I'm audible and visible to each and every one of you students. Now again, plant physiology will be starting right now students. We'll be starting off with the plant physiology. I'm waiting for the presentation to download, that's all. Yes? A minute. Done, 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 done. Perfect, perfect students. Import, downloads. Huh? It is not there. Students, where is it going? Cancel. File, import, new. What? It's going somewhere else. I don't know where is it going now. Where is it going? One second. Let me just check the file location here. Let me just check the file location. Where is the downloads even going? Someone changed. Student, this is why coming to the office is such a big task. 1 second, students. Students, tell me what is your preparation level in the plant physiology right now? What is your preparation level in the case of plant physiology? Hello, hello students, hello. Thank you so much students, thank you so much again. Yes, no sir, students, I'll eat my lunch later on. Students. Students, what is your confidence level in the case of your entire plant physiology students? What is your confidence level in plant physiology students? I am from home, huh? Okay, nice. Nice. Let it import. Let the file import students. Bird rate, I'll give students. Bird rate, I'll give you. What is there? If you, all of you score really well, obviously if you come to Bangalore or if you are coming to your city, we'll give you a treat. What is there in that? Okay. What is there? I'll give you bird rate. 90%, 50%, moderate, 0. Bro, 0 little difficult now. 0 to 100. Okay. Not good, sir. 70, 75. Okay, it's fine, students. If you are anywhere between 70 above or out of 5, 3 above, this is the video for you students, because in today's lecture, we will be going chapter wise, every single chapter of plant physiology. We will be starting off with the first chapter, that is photosynthesis in higher plants. Again, students, I will be telling you the concepts. I will be teaching you the concepts, and then after that, I will make you solve PYQs also. So students, are you ready, students? Are you ready? Because it's a matter of brain thing now. All you need to do is concentrate in my class, listen to my words, listen to the points which I'm telling you, and listen to the questions which I'm asking you. So there's a very simple rule in my class. There is a very simple rule in my class, students. Every single time I ask a question, you're supposed to tell the answer, students. You're supposed to tell the answer, students. Okay, students, let's go. All of you. Students, shall we start off? You ready? Can I get some fire emoji in the chat to let us go? Let's completely start the entire plant physiology now, students. Ready, all of you? Ready? Everyone, students, quickly fire emoji in the chat, and we will start with the first chapter of your plant physiology, that is photosynthesis in higher plants. Again, there is no deleted syllabus here. The entire chapter is as such. And out of the 3 chapters, this chapter has the highest weightage among your entire plant physiology, students. Entire plant physiology. Now, students, how many of you cook? Students, how many of you cook in the house? Raise your hands. See, I love cooking. I love making food in the house. I love cooking in the house. Right? So every single time you need to cook something, do we need ingredients? For example, let's say I'm trying to make chicken biryani. I need chicken, I need rice, I need spices, I need salt, I need everything possible to make a chicken biryani. If you're trying to make chapati, we need obviously wheat flour, water, salt, and everything. So to make any type of food item, we need ingredients. And we always know that plants make their own food. So if a plant is able to make its own food, does the plant also require certain ingredients, students? Tell me yes or no. Students, do you think that plants also require certain ingredients? With the help of these ingredients, they can make their own food? Yes, they can. Yes, they can, students. So here, let's say we go to plant also goes to shopping market, and plant is able to pick certain substances. Plant is able to pick some ingredients. For example, students, the first and the foremost ingredient is the solar energy, students. Tell me yes or no in the chat. Plants will require solar energy. They will require, students. Okay. Now, students, let's take one plant cell here. Let's just say this is a plant cell right now. This is a plant cell here. Okay. Now students, can anyone remind me again? Do plants take in CO2 or do plants take in oxygen in general? Plants take in, students. Plants take in what? Plants take in always, in general, plants take in CO2. All of you agree with me. That is, all of you and me, all of us are, we are giving CO2 to the plants and plants are happily taking the CO2 inside. Okay. Now students, inside the plant what happens? This CO2 is getting reduced, right? This CO2 is getting converted to a form of sugar called as, students, a form of carbohydrate called as, this CO2 inside the plant cell is converted to C6H12O6. That right here is the glucose molecule. It is going to be the glucose molecule, students. It's going to be the glucose molecule right here. Now students, you tell me in the chat right now, all of you, what is the major difference between this CO2 and this C6H12O6? See, here we have carbon atom, oxygen, okay. Then what is the missing element here, students? The missing element here is the water. All of you agree with me, students? So let's just add some water in it. Let's say you have a garden. In your garden you add water. We add water, students. We add water. In a garden we add water. So let's say we added water here, H2O. Okay, full chemistry. Chemist we are today. Chemist. Okay, chemist we are. Okay, so we add water inside now. Now what happens, students? Inside the cell there is a divorce happening now. Who's getting divorced here? The person who's getting divorced here is water. There is husband and wife are splitting here. That is, wife is leaving the house, students. So oxygen is leaving the cell, and this is called as your byproduct here. Byproduct of divorce here. Okay, byproduct of your divorce. That is, oxygen has been removed here. It has been removed now. So what is remaining is, students? Remaining is the your hydrogen here. Now this hydrogen will join this entire process and will help in the glucose formation. And can I say, students, can I say here what is happening here? There is nothing but addition. There is nothing but, students, addition of hydrogen. All of you agree with me, students? There is addition of hydrogen here, and this process of addition of hydrogen is called as what, students? What do you call that? Addition of hydrogen is called what, students? Addition of hydrogen is called as reduction. So reduction process happening here. Reduction is happening here, students. Reduction is happening. Now, students, addition of hydrogen is reduction. So CO2 is reduced to C6H12O6. That is, CO2 is reduced. CO2 is reduced here. It reduced to C6H12O6. Your CO2 is being reduced to C6H12O6 here. Reduction process happening here. Now, students, if this is undergoing reduction, if I call gaining of hydrogen is called as reduction. But here, water lost the hydrogen, right? Water is losing the hydrogen. So, can I tell water is technically undergoing oxidation? All of you agree with me? So, technically speaking here, water is undergoing oxidation. Is undergoing oxidation. Undergoing oxidation right now. Oxidation right now, students. Oxidation right now. Now, students, in this entire reaction right now, it is called as photosynthesis right now. Now, in the process of photosynthesis, plant is using sunlight. So here is the sunlight here. Plant is able to synthesize something. What is the synthesize, students? It is synthesizing nothing but the glucose. So glucose is being synthesized in the process of your photosynthesis, and this entire reaction is separate here. Now, as a matter of fact, students, the entire photosynthesis is divided broadly into two categories. One is called as light reaction, one is called as dark reaction. In light reaction, what happens? In dark reaction, what happens? That is the location where the question will be asked from. What is light reaction and what is dark reaction, students? So I hope till here there is no doubt to any one of you, students. I hope till here there is no doubt. This entire process of photosynthesis is called as it is called as physico-chemical process. Physico-chemical process here. Why, students? Because there is the exchange of gases here, physical process, breaking of bond here, chemical process. So it is a physico-chemical process happening here. Now, all of you tell me in the chat right now, students, Is this an anabolic process or a catabolic process here? Now here what are we doing is from inorganic substances, inorganic substances, we are making organic things here. That is, it is a anabolic process. Now students, you tell me the chart right now. Is there students absorption of heat here? So if there is an absorption of sunlight here, should I call it as endothermic reaction or should I call it as exothermic reaction, students? I should be calling it endothermic reaction. Entire thing is called as endothermic reaction. Okay, endothermic reaction, students. Now till here, is the basics clear to all of you, students? Addition of hydrogen is called as reduction. Loss of hydrogen is called as oxidation here. Now students, all of you should know very well right now, here hydrogen right now is nothing but the water is nothing but the H plus donor here. That is, hydrogen donor is the water here. Okay, and this particular oxygen, students, this particular oxygen in the glucose is coming from which location, students? This particular oxygen in the case of your glucose, where is it even coming from? It is coming from nowhere. Is it coming from water, students? Is it coming from water? No. This oxygen is actually coming from the CO2 here, from the CO2. This is the same oxygen coming here. While the water oxygen is removed outside the cell. It is removed outside the cell. So I hope till here, this is called as basics of your photosynthesis in higher plants chapter. If you understand the background knowledge, this is the framework which you need to understand. If you're able to understand this concept right now, this chapter is going to be very easy. That is my guarantee, students. That is my guarantee. I hope this basic concept of what exactly is happening in a plant cell is clear to all of you. All of you, students, it's a redox reaction. Then, students, can we go to the next concept now, students? Till here, any doubts? Starting part, I had to go slow because this is the basics of this chapter. If you don't understand this basics here, this chapter becomes meaningless to understand. There's no point in by-hearting anything further. Okay, students? Now, before we go, students, this is the entire reaction here, physical-chemical process here. Carbon dioxide plus water in the presence of sunlight and chlorophyll, glucose plus oxygen. This is what we learned here. CO2 plus water will give you glucose in the presence of sunlight and chlorophyll, and oxygen is a byproduct here. Same thing here, students. Same thing here. Now, importance of photosynthesis. Understand, there is something called as importance here. Obviously, students, obviously. Can you think of a life without plants? Students, can you think of a life on earth without plants? Not possible. For a matter of fact, students, the primary producers, the first trophic level in the entire hierarchy, it is nothing but the plants, right? The first trophic level, the producers are plants here. Without plants, we consumers will not exist, right? So, primary source of food on earth, responsible for the release of oxygen also. It is responsible for the release of oxygen, students. Now, again, can anyone remind me, students, this release of oxygen is mostly done by algae or mostly done by normal plants or your angiosperms? Tell me, students. Remember there was a meme? Remember I showed you? Right? I remember, students, some of you, I think it was Megan who sent me the meme. Students, I appreciate all of you sending me memes, okay? Please send me botany memes. I'll be very happy because it's a time pass, it happens for me also, right? So without algae, right? Without algae, there is no, most of the oxygen fixation will not happen, right? The algae does that. Now, students, now we come to certain type of early experiments, right? We come to certain types of early experiments right now, okay? Now, in these early experiments, we will focus on certain scientists and what was their contribution. All of you ready, students? All of you ready for the scientists and their contribution? All of you? It is algae, not angiosperms. It's algae. Okay. Now, the first scientist is called as, all the scientists here, there is an experiment called as variegated leaf experiment. Now, what is the meaning of variegated leaf? This right here is called as variegated leaf. The meaning of variegated is that variety of leaf. For example, in one leaf itself, we will have one region which is green color, one non-green color, students. So, in one leaf right now, we have two types of your pigments here. Two types of pigments in one leaf right now. Now, students, now this experiment, it is called as variegated leaf experiment. Now, in variegated leaf experiment, students, we understand the importance of who, students? we understand the importance of chlorophyll. Right. Now, for example, here we have a variegated leaf here. Now, here students, we have certain regions here. Here we have certain regions where we have dark green color here. Can all of you see here? Dark green color is here. Dark green color is here. Then, students, there are certain regions here. There are certain regions here where we do not have green color here, light green color. Then we keep this, we take this leaf from a plant which is already in the sunlight for a very long time. Now we take this leaf and I take it home. Let's say Naina takes this leaf home now. Now Naina and as well as Likita are doing certain experiment here. Both of them are adding certain types of your, what is that? Friends, that paneer test, paneer also you do a fake paneer test. That is nothing but your iodine. So once you add iodine, we notice that wherever we had the dark regions, we get blue-black color here. We get blue-black color here, students. We get blue-black color here. So this indicates that, students, what does it indicate? It indicates that, students, in the blue-black region, there was production of glucose. Glucose was here, students. And how do we get glucose? Glucose is a byproduct of your photosynthesis. So it tells you, students, it tells you that in the wherever we find chlorophyll, in that location photosynthesis is happening, and in that location glucose is being produced, and this shows the importance of what? Importance of chlorophyll in the entire process of photosynthesis. Students, I know all of you students, all of you, right? All of you are like, listen to a podcast, don't listen to a podcast, type also in the chat here and there. I feel all of you are listening to a podcast now again. Okay, so I believe all of you understood the first topic done. Okay, chlorophyll is necessary for photosynthesis. Now, students, next experiment is called as Moll's half-leaf experiment. Who wants to do this, students? Who wants to do this experiment? Who wants to take part in the experiment? I want one volunteer. We will make one scientist on the board. Okay, let's take Roshni right now. Let's take Roshni. Students. Now, I'll give a plant. I'll give a plant to Roshni right now. I will give a plant to Roshni, and I will tell the Roshni to cover this half of this leaf, half of the leaf here in black color paper, students. In black color paper right now. Okay, half of the leaf is covered in black color paper here. Now, in this region right now, there is no sunlight. There is no sunlight here. No sunlight right now. Okay, no sunlight. Now, what do we do, students? What do we do? Oh, this is different. One second, students. This is KOH experiment. Oh, this is one, students. There are two experiments like this. One is for sunlight and one is for KOH. We'll do the KOH one right now. One second, students. We'll change our experiment only now. Okay. Now, what we need to do here, students, is that here, this jar, right? In this part here, we are adding something called as KOH, students. We are adding something called as KOH. Okay, KOH. Okay. Now, what does KOH will do, students? This KOH here, it will remove all the CO2. All the CO2 present here will be removed by KOH. So, KOH will come, it will eat all the CO2. But in this, students, in the top region we can find CO2 is present. And after some time, students, if you do the experiment of your iodine again, we will notice that wherever we find CO2, in such case starch formation is happening. Wherever we add KOH, there was no CO2. Since there was no CO2, there was no conversion to glucose, and there was no glucose here. No glucose here. So glucose was completely absent here, and that is why it does not turn blue-black. It does not turn blue-black, students. Done? All of you? That is, CO2 is essential for the photosynthesis here. CO2 is essential for photosynthesis here. Done. Now, for the next experiment here. Next experiment is called as bell jar experiment. Next experiment is called as bell jar experiment. It was done by your scientist called as Joseph Priestley. Okay, Joseph Priestley. Now, what was the hypothesis here, students? Hypothesis is very simple here. Here we have jar A. Students, here we have jar A here, jar A. Now, what happens here? There is a candle burning here and there is a rat burning here, students. We will call this rat as ratty. Okay. Now, what happens is that after certain, let's say 2 hours maximum, okay, I'm being generous here. After 2 hours, the ratty is dying here. So, RIP in the chat. all of you. So a rat is dying here, as well as the candle is stopped to burn here. That is, candle is extinguished here. Now it tells you that, what does it tell you? It tells you that, students, something is present in the air. Something was present in the air. It got completely consumed. The burning of candle and the respiration by the rat completely consumed something in the air which was previously present here. Now, students, can anyone tell me what is the something here missing? Now, Joseph Priestley gave a hypothesis. Later on, students, in your second jar, in jar B, he added a mint plant. Let's say he added a mint plant here. Okay. Now, once he added a mint plant here, students, he told us that plant is restoring to the air whatever breathing and animals and burning of candle removed. That is, burning of candle as well as the respiration, it completely removed something called as oxygen. Oxygen was completely consumed by these two processes. So, but when you keep a mint plant inside, a mint plant is able to release what, students? Able to release oxygen, and that is why the candle is burning and the rat is alive here, students. And the rat is alive here, students. Done? All of you? Everyone happy now? Ratty happy, you are happy, everyone is happy. Okay? Everyone is happy right now. All of you. Okay? All of you are happy right now. Now, Joseph Priestley experiment. Okay? Now, students, then we have Jan Engine House. This person, we say that, right? Don't copy the homework. Okay, you will tell to your bro, ma'am will get to know, sir will get to know. Don't copy my homework completely. You do some small change here. You do a small change before you copy my homework. This person is copying the exact setup, almost the exact setup of your Joseph Priestley with a small change here. With a small change, students. What is a small change, students? Understand, friends. Jan Engine House did an experiment where he kept a plant in a bottle. Students, have you seen this anywhere, students? Have you seen this anywhere? Like a plant kept inside a bottle, glass bottle? Yes, we have. Joseph Priestley only. Okay. But the difference was, students, what is the difference here? He did the experiment in the daytime, wherever we have sunlight, and he did the experiment in the shade or in the night time. He did the experiment here. Now he noticed that, students, he noticed that in the daytime I am getting bubbles here. In the night time, no, I'm not getting any bubbles. So bubbles is missing. Okay, not the power of bubbles. This is different bubbles here. Okay, actual bubbles. Now it tells you that in the daytime what is happening? In the daytime, bubbles means oxygen is being released here. Oxygen is being released means what? In the daytime, photosynthesis is happening. It is occurring here. But in the night time, no bubbles means no oxygen here. And in the night time, it means that there is no photosynthesis is not happening here. It is not happening here. So it tells you that the importance of what? It tells you the importance of sunlight. That's all. Importance of sunlight. Friends, done? Now, see here. Oxygen is important. Sunlight is essential. Bubbles is equal to oxygen. Green plants. Now, students, then we have Julius von Sachs, students. Okay, provided evidence for the production of glucose by the plants. He told us that glucose is produced by the plants. His studies showed that the chlorophyll is located inside the chloroplast. In the case of plant cell, there is a special type of structure here. Called as your chloroplast, and inside the chloroplast, we can get something called as chlorophyll. Sir, for this, students, exactly, don't do Dabba experiment. Back then it was not a Dabba experiment, it was a very good experiment that time, not Dabba. Students, all of you know what is the meaning of Dabba? The meaning of Dabba is waste. So in, I don't know, students, in South India we generally say, Dabba means waste. Okay, waste person. Okay, so he also told students, leaves synthesize glucose, which is later stored as starch in the case of plants. So basically it is stored as starch in the case of plants again, students. Can anyone tell me again, students, is starch a homopolymer or heteropolymer, students? Homopolymer or heteropolymer? Quick, students, quick. Now, then we have T.W. Engelmann experiment, students. T.W. Engelmann experiment. Now, his experiment was very important because he did a small experiment, students. He did a small experiment here, right? I know box in Hindi, but in South India, Dabba means waste person. Okay, like a donkey. Now, students, here, but donkey also is very important, students. Donkey is very important. It is doing a lot of work, so we can't compare all of it with donkey. Donkey is very great. Okay, now, students, what did T.W. Engelmann do? T.W. Engelmann did a very simple experiment, students. He used physics in biology. physics and biology, students. Okay, students, how many of you? Okay, small secret. If my friend gets to know this, he'll probably throw me somewhere. I still remember when we were in class 11th, we had experiment, class 11th or 12th, we had experiment on prism. My friend, after school got over, I still remember, he asked me to stand near the door to check if someone is coming. I thought he's going to do something great. Okay. At the end of it, end of students, after 10 minutes, he's coming out and he's like, Baswa, come here, come here. I'm like, what, bro, what? He's opening his pocket and he's showing me, look at this prism. From physics lab, shamelessly, he went and he picked up prism, escape off. Okay. And he still has a prism in his house. Oh my God. Students, now all of you know what is the prism. All of you know what is the prism, students. In prism, what happens is that when the white light is entering inside, the white light undergoes scattering here. The white light undergoes scattering right now, scattering. Okay. Now, students, when the scattering happens here, that and he took this entire scattered light and he completely illuminated the scattered light on a particular test tube here. This is a test tube here. Now, inside this test tube, he had something called as a green algae called as cladophora. He had an algae called as, this is a cladophora algae here. So he kept this algae inside here, and he also added something called as aerobic bacteria. Aerobic bacteria. Aerobic bacteria, students. He also added something called as aerobic bacteria, students. Have some shame. Who flicks glass rod from the chemistry lab? Have some shame. Even if you're trying to get something, don't get glass rod. Glass rod, what will you use to do with the glass rod? Can't use it as a straw, as a straw also. Now, students, when he noticed that when he illuminated all the spectrum of light on this particular algae here, he noticed that, students, in a specific regions, there was more aerobic bacteria in these two regions. Okay. If more aerobic bacteria is present in these two regions, what is the meaning of that? It means that, students, in this region and this region, there is more oxygen here. That is why we have more aerobic bacteria here. Common sense. For example, I come to your class and I tell, I am giving you chocolates, my birthday treat. All of you, where are you sitting? You will start running and coming towards me. You're like, free chocolate. Same thing. All the aerobic bacteria here, like, oh, free oxygen, free oxygen, free oxygen. It is going mental now. All the oxygen is losing it. They're like, free oxygen. So all the aerobic bacteria is losing it now. So all the aerobic bacteria will accumulate in two regions. Why? Because there is more oxygen here. Now, what is the meaning of free oxygen here, students? The meaning of more oxygen here is that there is more photosynthesis happening in these two regions. In these two regions. And these two regions are nothing but red region and blue region, students. In red region and blue region, there is more photosynthesis happening, and this became a precursor or the first type of action spectrum. Action spectrum. Okay, action spectrum. So action spectrum tells you, students, action spectrum tells you that at what wavelength of light photosynthesis is happening. Photosynthesis is occurring here. So at these two wavelengths here, photosynthesis is happening. So this is called as nothing but action spectrum right now, students. Action spectrum. Done, students? Okay. Done, students? Done? Done? Now, students, cladophora done. Aerobic bacteria done. This is what we learned till now. Okay. There is one more last scientist called as Cornelius Van experiment, students. Very simple again, nothing much here. All he told us what? All he told us that, students, in the case of your entire photosynthesis, the hydrogen donor is water. That's all. That's all, students. Nothing more than that. Hydrogen donor is water. That's all, students. That's all. Okay. How did he do the experiment? He did the experiment on something called as your purple and green sulfur bacteria. And in the, students, in these bacteria, here water is not the hydrogen donor here. Water is not the hydrogen donor, students. In these bacteria, students, in this bacteria, the hydrogen donor is your H2S. It is going to be, students, H2S is going to be the hydrogen donor here, students. Now, students, all of you tell me in the chat right now. In the case of plants, what happens? In the case of plants, we add water, and in the case of bacteria, we have H2S. Now, students, when this water undergoes divorce, this hydrogen is given there to your glucose formation and oxygen is released. Now, what happens here, students? What do you think released here? What do you think is released here, students? Here, hydrogen is given again for the making the food here. Nice food. And remaining sulfur is released as a byproduct here. So sulfur is released in the case of this certain bacteria. Okay, and imagine the smell, students. Imagine the smell here. Imagine the smell, students. Okay, now students, that's it. Nothing more difficult here. Okay, so oxidation of water results in the liberation of O2. Oxygen evolved from the green plants comes from the water and not from CO2. From the water, the oxygen is being evolved. O2 is being evolved here. Done, students. Done. All of you? Yes. Done, students. Okay, why is the board stuck now, students? It reminds me of home now. The board getting stuck reminds me of my home. Okay. Now, students, now we learn something called as site. Now we learn something called as site of photosynthesis. Where does the photosynthesis takes place? Students, does the photosynthesis happens in the case of xylem and phloem? No. It happens in a structure called as chloroplast. There is a structure called as chloroplast where the entire photosynthesis takes place here. Now, this is a structure of a typical chloroplast here. I will tell you what you need to remember here and how question can come from here. Listen to me, students. Now, all of you, this is a double membrane structure. Double membrane structure. Outer membrane, inner membrane. It is basically wearing two jackets here. In summer, it is wearing two jackets. One jacket, one more jacket. Okay, two jackets here. Now, inside the chloroplast, students, we have certain coin-like structures here called as thylakoid. Coin. Each single coin is called as thylakoid here. And entire stack, students, the entire stack is called as grana. Okay. So, the entire stack is called as grana. Each one coin is called as thylakoid. It is called as thylakoid, students. Done, all of you? Done. Absolute drama. Now, students, every single grana is connected by a pipe here. Not a pipe, there is something else. Okay. Now, it looks like a pipe. Okay, it looks like a pipe here. Now, this pipe is called as what? Stromal lamellae or stromal lamella. Stromal lamella is a pipe between two grana. For example, let's say this is one building, one more apartment. In large apartment here, we have one houses here. So one apartment and one more apartment are connected with the help of one pipeline. The pipeline is called as stromal lamellae. Now, students, in this apartments here, what is happening? We have a large area here. This large area is called as stroma. Matrix is called as stroma. Matrix. Students, one of my favorite movies, Matrix. Okay. Now, matrix is called as stroma here. Now, students, all of you need to understand, in this stroma, all the dark reaction happens, not dark magic. Okay, dark reaction happens here, not black magic or dark magic. This is different here. Okay, dark reaction happens in the stroma. Okay, and in this thylakoid, grana, and stromal lamellae, all three of them, students, all three of them here. In all three of them here, light reaction happens. Okay, light reaction, puja happens here. Okay, light reaction happens here. Now, students, what is more important for examination point of view? From examination point of view, I want every single one of you to please remember at any cost which photosystem is present where. Let me teach you. If you look very closely here, students, you will realize that on the thylakoid we have both. Students, doesn't it look like a caterpillar? No, looks like a caterpillar with two eyes. Okay. Now here, students, on this thylakoid we have both PS1 and PS2. Both are present on the thylakoid. Both are present on thylakoid. And at the same time on the stromal lamellae, students, what do we have? On the stromal lamellae, what do we have, students? We have the PS1 only. So on stromal lamellae, on the pipeline we have what? On the pipeline we have PS1 only. And on the on the thylakoid we have PS2 and PS1. Both the photosystem are present on both of them. Then, students, any doubt till now? Then, students, all of you, done? Only PS1, both are present here. Students, I realize I am talking from last 30 minutes without taking a break, without even inhaling once. Continuously I spoke so much. Okay. Students, yesterday also told you. I will tell you all the time, students. I will tell you all the time. Students, my college, Christ University. Christ. Now, students, done. Now, pigments involved in photosynthesis. Which pigments are involved, students? Which pigments are involved in photosynthesis right now? Okay. Now, first of all, there is a small experiment here for you to do here. How can we know which pigment is present? How can we know which pigment is present here, students? First of all, students, all of you know how to make chutney. Raise your hands. Students, all of you know how to make chutney. Students, have you seen ma'am's bottle? Students, have you seen ma'am? Look at the fancy bottle which ma'am has. This is ma'am's bottle, students. You know what is my water bottle? One 10 rupees ka this Pepsi bottle, old yesterday's Pepsi bottle. I was just showing them a fancy bottle, ma'am. Yeah. And this is my water right now, ma'am. Thank you, ma'am. Now, students. No, it's not. Now, students, all of you know how to make chutney now? Yes, we'll make some chutney now. Okay, understand, all of you. How to make chutney? So, you need to take one leaf here. Take any leaf. Okay, don't take any random leaf also. Take a normal leaf on the roadside. Then you like, sir, I took a dry leaf, nothing came. Don't take a dry leaf. Take a normal fresh leaf. Okay, let's take mint leaf only. Let's take mint leaf, students. Mint leaf. That is what tumbler. I don't know, students. I know only water bottle. Tumbler. I don't know. Okay. Now you take one leaf here. You add certain type of your solvent here. All of you know what is solvent. No chemistry people. If you do not know what is solvent, I cannot help you now. Okay. Exam is in 3-4 days. You don't know what is solvent then. Okay. You add a solvent here. Okay. And nicely make a chutney out of it. Now once you make a chutney, you need to do something called as filtration here. Why? Because we don't want extra leaf of part here. We need only the bottom part, that is the extract here. Now this is very concentrated. This is very concentrated right now. Very concentrated. Now students, this entire thing needs to be added drop by drop on this point. Drop by drop on this point, students. And remember, students, if you are doing this experiment in future, please be careful of your slide, because there are many thieves and robbers in a class who will take your slide and run away. So be very careful with the marker. Write your name also here. Let's say I'll write Baswa here. I will write my name here, students. I don't want anyone to flick my slide also. I'll write BAS here. Okay. So write your name also somewhere in the background. Now, students, after adding your drop by drop here, you take this slide and keep it in the test tube here. Keep it in test tube. And in the test tube also, students, we have a solvent here. Now what happens? Capillary action happens. Now, for example, students, all of you know what is capillary action? Right? Physics, little bit physics. For example, students, if there is water here, if I keep the pipe down, because of the capillary action, the water will go upwards automatically. You might have observed. So, with the help of capillary action, what happens, students? With the help of capillary action, what happens? As the solvent is going up, as the solvent is able to move up, right? Solvent is moving up. All the different types of pigments which are soluble in the solvent are also getting separated here. Based on the solubility, they're getting separated here. They're separated, students. So as you can see, we get a certain bands here, and we can identify it, students. We can identify here chlorophyll A, chlorophyll B, xanthophyll, carotenoids. So all of these are called as your pigments in your plants. Okay, pigment in the case of your plant, students. Students, if I drop the bottle, ma'am will only drop me from first floor or second floor. That's how costly I feel the bottle is. I feel even if I touch the bottle also, she'll ask 10 rupees from me. Okay. So if I drop the bottle, gone then. Now, students, what is a pigment, understand, students? What is a pigment you understand? All of you. What is the meaning of pigment you understand right now? Pigment is, students, pigment, all of you might tell me, students, pigment is chlorophyll. That is an example of pigment. But what exactly is a pigment? Pigment is chemical. It is something which can give color. It can give color, obviously. Color, which color? Okay. Now, understand what is happening here, but what is a pigment actually? Pigment is nothing but, students, it is a chemical that converts light energy into chemical energy. This is the definition of pigment, not just color. Not just color here. Okay, color. It is actually it is converting the chemical energy into, light energy into chemical energy. For example, students, how the plants make their own food? Plants make their own food by using the pigment called as chlorophyll. It is able to trap the sunlight and convert it to chemical energy. Okay. Now, let's say here we have a pigment here. Students, let's say we have a pigment here, chlorophyll A. Chlorophyll A is the pigment right now, students. Chlorophyll A is the pigment right now. Now, what happens here? Now, let's say we have sunlight here. Sunlight is coming. Students, do you realize pigment are like a Superman? See, Superman also, how does he get power? Superman gets power from the sun. All of you know that the more the sun solar radiation, stronger the Superman. So your plant is also like a Superman now, because it is able to absorb light, use the light energy. So this is the secret of Superman also. Who knows, Superman might have something like, some type of cells similar to chlorophyll. Who knows, right? Now, students, here it is able to absorb the solar radiation, and when it is absorbing the light here, all the electrons get excited here. Now, this excitation of electrons will happen at a fixed wavelength. So, all of you should know something called as PAR. Called as, students, PAR. Not PAR. PAR. photosynthetically active radiation. At this wavelength right now, students, plants can absorb the light here. Okay, there is something called as specific wavelength. I'll tell you that term later. I'll tell you later on that. Okay, now electrons are getting excited now, students. Now these electrons will come back to the ground state. These electrons will come back to the ground state. It has to come down. Physics again, physics. I don't know what will the physics be, but okay, some physics is here. Now they'll come back to the ground state here. When they're coming back to the ground state, what happens is that they can emit the light here. Yes, they can. All of you might have observed, right? There is a experiment called as salt analysis here. All of you might be aware that certain types of salts can provide different color, right? Some can give green color, some can give red color, brick red color, some can give purple color, bright red color. Something is there. I don't know the rest. All of you think that you know. Okay. Now what is happening is that when the electrons are coming back to the ground state, it can emit light here. It can emit light here. It can emit heat here. Or. This is very important. It can transfer the energy to another pigment here. This is very important when you want to learn something called as light harvesting complex. If you really want to understand light harvesting complex or non-cyclic or cyclic photophosphorylation, this point is very important because sometimes what happens is that, spoiler alert, sometimes what happens is that electrons or energy is passed from your PS2 to PS1. Students, which reaction, students? Students, which reaction? Students, which one? Tell me, students. Which of the following reaction where what happens from PS2 the energy is going to PS1, electrons are going to PS1? That will happen. Okay. That will teach you later. So non-cyclic, I'll teach you non-cyclic later on. This happens in non-cyclic. This is a spoiler for non-cyclic right now. Or we can use the energy to do a reaction. That is basically your photochemical reaction here. Okay. Now students, now we will understand something called as action spectrum as well as absorption spectrum. Students, all of you are nice. So all of you are understanding the concept right now. So all of you understood what exactly happens in the case of non-cyclic. Pigments are exciting the electrons, energy transfer to the next pigment here. Right. Now students, non-cyclic, it happens in non-cyclic, I'll teach you later. Now let's understand what is called as your action and absorption spectrum. Ready all of you? Let me take a breath, student. Let me take a small breath. Continuously I'm talking from past 20-30 minutes. Water feels so good, student. Water feels so good. All of you drink some water. And all of you give your brain some 1-2 minutes to think also, because I feel I am forcefully feeding all the information to all of you, and all of you just eating the information. All the information is going from this, is going inside. Okay. So let's take a breather, right? Let's take a breather right now and process all the information you learned till now. Process it. Okay. Feels so good. So good, students. Students, didn't you feel like you were watching the lecture in 2x speed or 1.5x speed? Yes. Students, didn't you feel that? It felt like the lecture was going in 1.5. It's good. Otherwise, some of you are like, sir, I don't have time. Then they'll comment. Legends, one night before the exam. That time you don't have to watch in 1.5x. You can watch my lecture in normal condition. It will feel like 1.5x in either way. It will feel like 1.5x either way, students. Okay. Now we come to something called as action spectrum as well as, students. One second, there's a poll coming up. Students, all of you like the video first of all? Gopika ma'am has added a new poll, so I want all of you to quickly smash the like button. Okay. Now students, now. What exactly is action spectrum and absorption spectrum? Understand first of all. What is absorption spectrum? Students, in which there are certain classes where you can absorb the information very fast. Sometimes you cannot absorb very fast. For example, you are fresh into school, you went to school right now, first period, okay, and you're sitting there, all the information is going to your head. But you have a class right now, let's say I'm taking a class after lunch break now. All of you nicely ate curd rice, all of you ate chapati, all of you ate biryani, you're coming and sitting in my class. I will also feel sleepy looking at your face. Tell me yes or no. So in that condition, you might not be able to absorb all the information I'm telling you, because your brain is not active that time. Your brain wants to sleep. Looking at you, even I'll sleep off. Okay. Now students, what is happening here is same thing is happening. Same thing is happening in the case of your plant also. Of all the solar radiation, plants sometimes can absorb certain wavelength. So other wavelength plant does not want to absorb. Like how you have a favorite food item, right? If I'm giving you, let's say I am giving, let me make you jealous, students. Let's say I give you a protein bar to all of you. I will give you a protein bar, okay? Or I will give you, let's say, one eclairs, 1 rupee eclairs. Which one do you want, students? Which one do you want? All of you will want what? All of you want the protein bar because very tasty. Okay, some of you will not want, but okay. So like how you have a favorite food item, right? Similarly, plant also have a favorite wavelength of light, and plant will absorb that wavelength of light. Okay. Now, what is absorption spectrum? Absorption spectrum tells you that. Students, it tells you that at which wavelength, which pigment is getting absorbed. For example, at this wavelength right here, chlorophyll A is getting absorbed. At this wavelength, chlorophyll B is getting absorbed. At this wavelength, carotenoids is getting absorbed. So every single wavelength, something is getting absorbed. Tell me yes or no. It is getting absorbed. So at which wavelength, at this wavelength here, a pigment is getting absorbed. That is called as absorption spectrum. At which wavelength, which pigment is getting absorbed? Absorption spectrum. Now students, what do you mean by action spectrum? Action spectrum is that, students. What do I mean by action here? Not a movie action. The action here defines, the meaning of action here is nothing but photosynthesis. The action here is nothing but, students, the action here is photosynthesis is the action here. Okay. So action spectrum is telling you, students, it shows that at what wavelength photosynthesis is happening. At this point here and this point here, photosynthesis is happening. That is called as action spectrum. Students, do all of you remember this experiment which we did 10 minutes back? Students, this experiment right here, it tells you that at this wavelength of light right here and this wavelength of light here, and these two regions, in red region and blue region, what is happening? Photosynthesis is happening. That is called as action spectrum. Action is happening here. What is happening, students? Action is happening here. Done. Action is happening here. That is called as action spectrum. Now this is your, if you can notice here, students, this is your absorption, action. Now students, if you overlap these two, if you overlap the absorption, that is your light blue, black color action. Now you might notice here, students, is that at this region absorption is happening, action is happening. Absorption is happening, action is happening. But what is the but here, students? The but here is that at 500 to 600 nanometer, there is no absorption, but action is still happening. Why? Because students, because at this region, chlorophyll A is not getting absorbed. Only chlorophyll A is not getting absorbed. At this time, at this region, chlorophyll B might be getting absorbed. Chlorophyll C might be getting absorbed. Carotenoids might be getting absorbed. Xanthophyll might be getting absorbed. Yes or no? So all of these pigments are getting absorbed here. They're getting absorbed. All of you tell me, students, in the chat right now, sometimes you forget your lunch, because I used to forget my lunch, right? So let's say I go to school, I forgot my lunch, I kept it at my sofa and I came out to my school. Now once I go to school, lunch break is here. I'm like, oh my god, I forgot my lunch box. Do I simply keep quiet like this in corner? Do I sit in corner like this? No. What do? I go, without even asking, I will go, put my hand in my friend's tiffin, nicely take it and eat and run off. Yes or no? So even though there is no lunch box, even though I don't have any lunch box, what is happening? Even though there is no lunch box, there is, I am still eating. I am still eating. That is the same concept here. Even though chlorophyll A is not getting absorbed here, chlorophyll A is not getting absorbed here, right? Chlorophyll A is not getting absorbed. Photosynthesis is still happening here. Okay, photosynthesis is still happening. Now, that tells you that, student, that tells you that. What does it tell you, student? What does it tell you? It tells you that, student, there are two types of pigments here. One is called as chief pigment. Chief pigment means the boss here, the ultimate boss, your school principal. Yes, student, your school principal is boss only. Okay, this is your school principal here. School principal here is called as chlorophyll A. Chlorophyll A, and this is called as chief pigment or reaction center. Now, in your school, student, in your school, do we have other teachers here? Yes, we have other teachers here. Now, the other teachers are called as auxiliary pigment here. Okay, we have chlorophyll B, carotenoids, xanthophyll. All of them are called as your teachers of your school. They are called as teachers of your school. Students, again, this color also is very important here. Blue, green, yellow to yellow, orange, yellow. Again, very important. Again, very important. Okay. Now, students, all of you tell me the chart right now. Can this principal manage every single class on his own or on her own? Not possible. A principal requires teachers who can go to every single class and teach. That's how school works. Here also, students, here also, chlorophyll A cannot do all the work. It cannot do all the work. It needs other pigments to help the chlorophyll. So these are called as your other pigments here. Now, students, all of you tell me the chart right now. In school, what is to happen? Whenever you keep dancing in the class, or if you keep shouting in the class, what is that one dialogue? What are the two dialogues of your teacher? First dialogue is this a fish market? First dialogue. Second dialogue is worst batch ever. Second dialogue. The third dialogue is that. Students, your teacher will be like, I will send to principal now. I will send you to principal right now. Go to principal's office. Why? Because principal has all the authority. A principal has all the power to give you something called as TC. Students, TC is what, students? TC is nothing but transfer certificate. It is nothing but transfer certificate. Good only, no, you go to different school. Okay. Now, why am I telling that, students? Because here, this chlorophyll B, carotenoids, xanthophyll, it may be pigment, but it cannot do any work. These carotenoids, xanthophyll, and chlorophyll B, it cannot give you what? It cannot do photosynthesis. Why? Because it does not have the power. Just like teachers cannot give you TC, this chlorophyll B, carotenoids, xanthophyll cannot give you TC here. They cannot do photosynthesis. All they can do, students, all they can do is transfer the energy. Transfer all of you to, transfer the energy to chlorophyll A. Students, all of you understood? Students, all of you understood? All of you understood? Tell me yes or no. Friends, tell me yes or no. I have gone to principal's office, so I don't even know how many times I've gone. Principal's office. That used to be my second classroom only. Second classroom. Okay, done, students. Done. That is called as chlorophyll A. This is what happens here. Okay, this is what happens in your entire pigment here. Now, students, all of you tell me the chart right now. What will happen if all the teachers are absent in one school? Apart from party. Tell me, students. Apart from party, what will happen? If all the teachers are absent in your school one day, what will happen? Can the principal go to every single class and take all the classes? If principal go to every single class and he takes all the classes, principal next day will become that ratty. Okay, principal become ratty then. Remember, principal will become ratty. Okay, not possible. So similarly, students, imagine all the light is getting absorbed by your chlorophyll A. What will happen? The chlorophyll A will become ratty now. What is the meaning of that? Chlorophyll A undergoing photooxidation. That is, absorb light, protect the chlorophyll A from photooxidation. So if all of these pigments are not present, chlorophyll A undergoes photooxidation. To prevent that, we have other pigments here. Now, similarly, how a principal knows what is happening in every classroom. Why? Because of the teachers. So teachers are doing what, students? Teachers are expanding the reach here. Similarly, students, all the pigments here are able to expand the range of absorption of light. Understand, students? Understand here. See here. See, this is your principal here. Right, chlorophyll A. Chlorophyll A will absorb light at 680 nanometer. But we have chlorophyll B absorb at 6 to 680. Xanthophyll 550 to 600. Carotenoids 500 to 550. So do you realize all the pigments are actually increasing the range. They're like antennas. Students, can I call all of these pigment as antenna now? Like how an antenna can increase the range. Antenna can increase the range here. Similarly, all of these pigments can act as the antenna. So these are also called as what? These are also called as what, friends? Antenna molecules or light harvesting system. They are part of your light harvesting systems. They are called as antennas here. Like how an antenna can, send all the signal to one receiver. This is the one receiver. Students, who is the receiver, students? The receiver is chlorophyll A here. Chlorophyll A is called as the reaction center or the receiver or the chief pigment here. Remaining all the different types of pigments which are called as your carotenoids, xanthophyll, chlorophyll B are antennas. They will capture the light and transfer all the light to chlorophyll A or the reaction center. Students, understood? Tell me, understood or not? This much I talk, this much I spoke, all of you. All of you understood how the functioning of reaction center happens? How does the reaction center works? How does the entire antennas work? All of you understood, students? Yappa. My mouth only, students, mouth is hurting. I feel my mouth became 2 centimeter bigger by screaming so much. All of you understood, students? All of you understood? Yes? So here we have now chlorophyll B, carotenoids, xanthophyll, chlorophyll A, reaction center. Okay, reaction center. Now, students, this is called as light harvesting complex. This entire thing is called as what, students? Light harvesting complex. LHC, light harvesting complex. It will do what, students? So what is antenna? Antenna is this one. These are called as antenna molecules. These are called as your antennas. These are called as what, students? Antennas right now. Okay, they are called as antenna. Okay, okay, students. So this entire thing with reaction center as well as your antenna together they are called as LHC, light harvesting complex. Light harvesting complex is able to harvest the light for the plants. It is able to harvest the light for the plants. Then, students, then all of you, it is harvesting light. See, nicely it capturing the light and sending to there. Now, so all of you know what is LHC now. LHC has reaction center and antennas. That both are part of LHC. Now, students, understand what is the meaning of photosystem now. Understand what is a photosystem right now. Okay. In a photosystem, we have something called as LHC, which has antenna and reaction center. Electron carriers and enzymes and proteins are also present here. So this entire thing is called as what? This entire thing is called as photosystem. So LHC is part of photosystem. So all of you should remember LHC is a part of photosystem. Students, my life only went away explaining this now again. Okay, this is your entire photosystem. Entire thing photosystem. LHC is a part of photosystem. It is a small part of photosystem. Now see here. Here we have two types, PS1 and PS2. PS1 works at P700. Students, why 700 is coming first and 680 is coming later? Because, students, 680, this 700 was discovered first. That is why it's called as PS1. Not based on number. It is based on the discovery, year of discovery. Okay, discovery. Now this 680 is because of discovered later on. Okay, PS2, 680. Okay, now it is found in both grana and stromal lamellae. I remember PS1 present on both, while PS2 is present where? It is present only on, students. It is present only on where? On the pipe here. Sorry, it is present only on the your apartments. That is your coins. It is present only on that. Okay, PS is present on only on that. While PS1 is present on this also, PS1 we can find, and on the pipe also we can find PS1. On the pipe also we can find PS1. Now, friends, now we come to the main part of the chapter. Now, students, now we come to the main part of the chapter. Shall we go, Shan? Students, shall we start? Students, shall we start? All of you. Oops, student, the pen broke. Oh my god. Shall we start? Students, shall we start now? The main part of the chapter. This is the main part of the chapter. Now, this is where you understand more important concepts actually. Thank you so much. Thank you so much, students. Now, students. Now, Now we come to something called as the phases of photosynthesis. One is called as light dependent reaction, one is called as light independent reaction, also called as dark reaction. It is called as dark reaction not because it is dark in color, it is called as dark reaction because students it does not require light. It does not require light, students. You will get a lunch break after my class, don't worry. Lunch break, students, you will have lunch break all the time in the world for lunch break. Okay. Okay. Now, it is called as light independent reaction or dark reaction, also called as biosynthetic reaction until later on. Now, what exactly happens in light reaction? In light reaction, students, what is happening is trapping of light energy by pigment happens here. Okay. Now, under light reaction, we have two types. Under light reaction, students, we have two types here. Okay. Here we have two types. Non-cyclic photophosphorylation as well as cyclic photophosphorylation. So cyclic and non-cyclic again. Now again, students, I will give you my own, like this is the best thing which you can do, right? This is the best thing which you can do, students. Just learn this and you're good to go. Just learn this and you're good to go, students. More than this, they'll never ask you anything, students. More than this, they will never ask you anything. More than this, they'll never ask you anything, students. This right here is a non-cyclic photophosphorylation. Now, where does it start from, students? Where does it start from? Understand, students, understand. It is starting from something called as PS2. PS2 working at 680 nanometer here. Now, here, students, can all of you see this is called as your photosystem here. This is your LHC. LHC has your antennas here, antennas on top border here. Along with antennas, we have the in the middle reaction center. Solar energy is trapped by all of these antennas and energy is transferred to chlorophyll A. And this is the main pigment here where electrons get excited here. Now here, students, the primary electron acceptor. The primary electron acceptor in the case of non-cyclic photophosphorylation is none other than the pheophytin, students, pheophytin. Now pheophytin energy electrons sent to PQC by PC. All of you repeat after me, students, repeat after me. PQC by PC. All of you type in the chat PQC by PC. PQC by PC. PQ is nothing but plastoquinone. CY is nothing but cytochrome BCF complex. PC is nothing but plastocyanin. Three different things you have to remember here. Three different things you have to remember, students. PQC by PC here. Done, students? Done. Now what happens here, students? Now what happens is that from PC, the electrons is now sent to something called as PS1. PS1. So from PS2, we are going to PS1. PS1. Now again, what happens is, students, here also we have antenna molecules, antenna molecules. In the right in the middle, we have the reaction center here. Now here also energy is captured here. Now what happens again, students? Electrons are excited here. Electrons are excited here. Now here, students, the primary electron acceptor, the PEA, the primary electron acceptor in the case of your PS1 is nothing but iron sulfur centers, FES, iron sulfur centers. Now from here, electrons are transferred to FD, ferredoxin, ferredoxin. Now, students, who is the final electron acceptor, students? The final electron acceptor in the case of non-cyclic is going to be NAD+. So NAD+ is the final electron acceptor. Now this final electron acceptor will accept the electrons and it will also take 2H plus from the stroma. Now students, you tell me in the chat. Gain of electrons and gain of protons, what do you call them? Gain of electrons and gain of protons is called as what? Reduction. Reduction. So what is happening here is that, students, this NAD plus is undergoing reduction to form NADPH2 or NADPH or NADPH. And now in this entire reaction is enzyme mediated. Here we have FNR enzyme. FNR enzyme. Ferredoxin NADPH reductase enzyme. Ferredoxin NADPH reductase enzyme. Okay. Ferredoxin NADPH reductase enzyme is present here and final electron acceptor and we get NADPH2 in the end. NADPH2 and students. Done, all of you? Done. Here, now students, can anyone tell me, students, here electrons are getting consumed here. So do we need unlimited supply of electrons? Yes, we need continuous supply of electrons. The continuous supply of electrons is coming from, students, it is coming from the splitting of water. So water here is being split here. Water is undergoing divorce. All of you know divorce, right? So water is when it is undergoing divorce here. 2H plus is released here. This H plus, 2H plus are sent for ATP synthesis. 2 electrons are sent here, right? And oxygen is released outside. Oxygen is released outside. And this is what we learned in the first slide. This is what we learned in the first slide here. water is being split, oxygen is being removed out. Oxygen is being removed out here, students. Oxygen is being removed out right now. Okay, and these electrons are sent here. It is happening on the lumen side. It is happening on the lumen side right now. Lumen side. Okay. Now, students, all of you tell me in the chat right now. In this entire reaction, right now, O2 is released. O2 is released. Here in this entire reaction, students, in this step right here, you can see between PQ and CY, ATP is formed. Yes, ATP is formed here. ATP is formed. Do we have NADPH here? Yes, we have NADPH here. NADPH formation. Now, students, in this entire reaction, water is being split here. Yes, water is being split here. Water is undergoing splitting here. That is called as photolysis of water. Photolysis or oxidation of water. Okay. Now, students, in this entire reaction, is it called as, is it present in the Z way? Z way? Yes, it is called as Z scheme. It is called as Z scheme, and it is completely non-cyclic in nature. Completely non-cyclic in nature also, students. It is called as non-cyclic in nature, and that right here, students, is your non-cyclic photophosphorylation. All of you done? Done, students? Friends, I want all of you to learn this image itself, because in examination they can ask you any of the following questions. They can ask you from anywhere. They can ask you from anywhere right now, non-cyclic in nature. They can ask you this also, PQ, CY, PC from here. This entire thing can also be asked in examination. They can ask you this. They can ask you ATP or NADPH form. They can ask you all of these. They can be asked. Okay. Now, students, here is your cyclic. Again, very simple. Cyclic, what happens, students? Here we only have PS1. Now, all of you tell me in the chat right now, students. Here, if we have only PS1, what is the location here? Where is it even happening? Students, do you think cyclic is happening on the thylakoids? Do you think cyclic is happening on thylakoids, or is it happening on the stromal lamellae? Here. Where do you think it is happening? Here or here? Cyclic. Where do you think it's happening, students? Cyclic is happening right here. Cyclic is happening right here, students. Right here, cyclic is happening, because we find only PS1 here. We find only PS1 is present here. Only PS1 is present here. PlayStation 1. We have only PlayStation 1 here, students. Okay. So this is what happening. Chlorophyll A, PS1, 700 nanometer. 700. Electrons getting excited here. Excited here. Primary electron acceptor is FES here. FES. Then we have what? FD, PQ, CYP. Same thing. Same thing you write. FD you write here. Then PQ, CYPC. Between PQ and CY, ATP formation. FES and FD on ATP formation. Electrons are again sent back here because it is cyclic in nature. Now, students, here do we have O2 evolve? No. Do we have NADPH here? No. Do we have ATP here? Yes, ATP formation is happening here. ATP formation is happening here, students. ATP formation is happening here. Done, students? Done? All of you? Done? Cyclic. It is called as cyclic, students, here. See the difference here, students. Electrons move in a cyclic manner. Electrons in a linear pattern. Only PS1, both PS1, PS2. Expelled from PS1 first, expelled from PS1 first, and they expelled from PS2. Electrons return back to P700. They don't return back. They do not return back here. Synthesize ATP during the light reaction of photosynthesis by cyclic, here non-cyclic. Occurs in isolated chloroplast and photosynthetic bacteria. Occurs in higher plants and algae here. Occurs in without oxygen. It occurs in oxygenic condition. Oxygen is being evolved here. Oxygen is being evolved here, students. Final electron is P700 again because cyclic nature. Final electron acceptor is NADP plus here. Photolysis does not occur here. Photolysis that happens here. That is, water is being split here. Water is being split here, students. Water is being split here. Done, students? Done, all of you? All of you understood the entire cyclic and non-cyclic again? Students, all of you understood? Cyclic and non-cyclic. Students, again, if you want all of these notes, they will be available right here. If you want these notes itself, I want these notes only. I want to write this. This is the way I want to learn in the last few days also. Today only I will upload it here. Today only I will upload here, students. Okay. Now, students, we understand something called as chemiosmotic hypothesis. Students, how many of you think this is difficult to understand? Raise your hands. Students, how many of you think this is difficult to understand? While you reply to me, let me just take a bite of protein bar because I need energy. Ready? Ready? Students, understand, students. Students, I think I have to postpone my class of 2 p.m. I guess, probably. Let me just check the team. Okay. I think we'll take it tomorrow. Okay. Or some other time. Okay. Now, students, no. Biscuit, nice. Now, students, understand what is chemiosmotic hypothesis. Students, you tell me the chart right now. In your cyclic and non-cyclic, did we get ATP anywhere? Students, did we get ATP anywhere, students? Did we get ATP anywhere? Students, tell me the chart right now. Did we find ATP formation in the case of your cyclic and non-cyclic? Yes, there was ATP formation happening there. There was ATP formation happening in the case of cyclic and non-cyclic. ATP formation was happening there. It was happening. You can see, students. Here we can see here. If you go back, ATP formation. Here we had ATP formation. So, chemiosmotic hypothesis. Chemiosmotic hypothesis, it explains. What does it explain? It explains how the ATP was formed. That's all. That's all is chemiosmotic hypothesis. Students, that's all is chemiosmotic hypothesis. Chemiosmotic hypothesis explains how the ATP is formed. That's all. Nothing more than that, students. Nothing more than that it explains. It explains how the ATP is being formed there. That's all. Now, if you want to understand in detail, let me just teach one small extra point here. Understand, students. For a chemiosmotic hypothesis, first of all, I want a perspective you to understand what exactly is this and where is it even happening. Students, all of you ready? All of you ready, students? Ready? Okay. Where is this exactly happening? Students, in a chloroplast, there is a coin-like structure, right? What do you call that? In a chloroplast, students, we have a coin-like structure called as thylakoid. Okay, thylakoid. Now, students, if you enlarge a thylakoid, if you take this one thylakoid and if you zoom it, this is what we get. This is what we get, students. This is what we get if you zoom in one thylakoid. This is what we get if you zoom in one thylakoid. This right here, students, this right here is the thylakoid membrane. Thylakoid membrane. Thylakoid. This is one thylakoid. And on this one thylakoid, what is happening, students? On this thylakoid, what is happening? On this thylakoid, PS2 photolizes PQCYPC. Then we have PS1, then we have FD here, and then we have FNR here. Then we have your NAD plus taking here, and we are getting final electron acceptor, and then we get NADPH here, students. Can anyone tell me what exactly is this happening here? Did we learn this, student? Did we learn this? Did we just learn this entire thing somewhere right now? Can anyone tell me? What is this exactly on top? What is exactly on top, students? Tell me, students. What exactly written on top? That is nothing but non-cyclic. That is nothing but non-cyclic. And do you remember, students? Do you remember in the starting I told you? In the starting I told you, students, on the thylakoid, what do we have? If you go to my starting of the class, I told you right now here itself. Here itself I told you, students. Here itself I told you on the chloroplast. If you look at a structure of chloroplast, here we can find PS2 and PS1. Tell me yes or no. Here I told you, students, here we can find PS2, PS1, I told you. Now, students, you tell me the chart right now. Does it make sense now? Does it make sense why I told you here we can find both PS2 and PS1? It means that this entire process of non-cyclic photophosphorylation, it is happening, students. It is happening on none other than the thylakoid membrane. It is happening none other than on the thylakoid membrane, nowhere else. It is happening right there. It is happening right there. Entire non-cyclic is happening here, non-cyclic. Okay. Now, this is where the ATP formation happened. Okay. Now, students, for a chemiosmotic hypothesis to work, we need four main things. The first one is called as intact membrane. Then students, then we need something called as proton pump. Then we need something called as ATP synthase. And the final product which we need, or final thing which we need, is called as proton gradient. Proton gradient. Proton gradient we require. Okay, we need proton gradient. Now, what is the meaning of proton gradient? Understand, all of you understand. The meaning of proton gradient. The meaning of proton gradient is that, students, there is a difference in proton level. What we need to do here, students, see, this is obtained already. All three are obtained. These three are naturally present here. These three are naturally present right now. These three are naturally present right now. What we need is a proton gradient right now. How do we create that? How do we create it? There are three ways to create it. What we need to do here, students, inside the lumen, we need high proton gradient, and outside the lumen, we need lower. Outside is the stroma, we need low H plus. Inside, we need high H plus. Inside, we need high H plus, students. Okay. Now, students, all of you need to understand here. What is happening here is that, students, first of all, how is the H plus concentration increasing inside? Understand, all of you. Now, all of you remember, students, here I told you, when the water is undergoing divorce here, I told you oxygen is released as such, and I told you electrons are sent up here. Remaining H plus is sent inside the lumen, I told you. H plus is sent inside the lumen, I told you. It is sent inside the lumen. Now, all of you understood, right? So, when this water is being split here, can I tell H plus concentration is increasing inside from the splitting of water here? From the splitting of water here, H plus is increasing inside? Yes. H plus is increasing inside. Now, second one you understand, students. Second one. In the last step I told you, this NADPH will take one of the H plus from the stroma. If you go back here, students, I told you here, in the last step here, see this last step here, students. This H plus is coming from stroma. I told you right here, I told you. This H plus is coming from stroma here. So, can I tell, right now, students, can I tell in the stroma what is happening is H plus is reducing in the stroma. H plus is reducing in the stroma here. H plus is reducing the stroma. It is reducing the stroma right now. So, inside is increasing, outside is decreasing here. Yes, it is decreasing. At the same time, students, this particular structure here called as your plastoquinone will also act as a hydrogen donor, or it will act as a hydrogen carrier here. To be precise, it will act as a hydrogen carrier here. So, this particular plastoquinone will take one H plus from the stroma, pull it inside the lumen, and again inside H plus is increasing, outside H plus is decreasing. So, three ways. First one is splitting of water, increasing. Here H plus decreasing. Here again H plus increasing because plastoquinone is pulling it. So, by doing this method, can I tell inside the lumen right now here, inside the lumen of thylakoid, inside the lumen, ultimately the H plus concentration is more here inside. Ultimately H plus concentration is more here. It is more. Okay. H plus concentration is more right now here. It is more. Now what happens? Understand, all of you. Understand what happens. Now every single time this ATP synthase rotates. Every single time 4H plus is transferred outside. Whenever every single time 4H plus is used to rotate this, what happens? 1 ADP gets converted to 1 ATP. So every single time 4H plus from the lumen is transferred here. Can you all of you see 4H plus is being transferred here? 3,4 rotating the ring here. 3,4 rotating the ring here. And 1 to bring this phosphate here. And ultimately we get ATP here. And that is how ATP is formed in your cyclic and non-cyclic. This is the concept right now. Students. This is the concept right now. How ATP is formed right now here. That is called as not 2, 4H plus, not 2, 4H plus always written. 4H plus. So every single time 4H plus transferred, we get 1 ATP. 4H plus 1 ATP. That's all. Tell me, is it difficult to understand? This is so easy. 4 things here. How the proton gradient happens here, that's all you have to remember. That's all. Students, solve this question. Students, all of you solve this question, all of you right now. Solve this question quickly. Solve this question right now. Phase is light reaction. Photochemical phase is light reaction. Done, students, done. Done, students. See here. Do we get oxygen here? Yes. NADPH? Yes. ATP is used here. Students, do you think ATP is used here or ATP is formed? Or ATP is formed here? ATP is not used. What is being splitting here? Carboxylation, no. What is the answer, students? What is the answer? ABD. ABD. Option number B is the right answer, students. Here ATP is not used. ATP is produced here. Okay. This is a NEET 2024 question. 2024 question. Quick. The reaction center of PS2 is absorption maximum at. PS2 is 680. Is 680. 680. Incorrect. Grana lamellae has both PS2 and PS1. Grana has both, yes. No, wrong. Because cyclic will have only what? Cyclic will have only PS1. Only have PS1. What are the products of light reaction? Products of light reaction. What are the products here? Products. What are the products of a light reaction, students? ATP, NADPH, oxygen. ATP, NADPH and oxygen. One is the answer. See, water is not produced. Water is consumed. Same mistake you're doing, all of you. Some of you doing the same mistake. Here, water is not produced. Water is consumed here. Students, is my voice coming right now? Sound check, students. Am I audible right now, students? Am I audible? Students, am I audible right now? Sound check, students, sound check. Sound check. Voice is there or not? Voice is there? Yes? Okay. So here, water is not produced here. Water is not produced here. Remove that. Okay. This is an easy question. Skip this. Oh, this is also easy for students. Easy questions, all these are. Quick answer. Easy question again. All of the NEET PYQ students. All of these are NEET PYQ itself. Again, all of them are very easy. Matrix. See here. The first electron acceptor, iron sulfur centers, photosystem 1. Okay. So I'll give you this PDF, students. You can solve the questions very easy. There are many questions. You can solve all of them very easily. Okay. You can solve all of them. Okay, now students. Now we come to dark reaction, students. What exactly happens in the case of dark reaction, students? Understand. What exactly happens, students, in the case of dark reaction? All of you need to understand here. Okay. Understand. Dark reaction is also called as biosynthetic phase or Calvin cycle. Why? Because in this biosynthetic phase, the entire glucose which we eat is produced in a dark reaction. In light reaction, only energy is made. Like you make the money in your light reaction. The money is spent to make the food here, to buy the food. Okay, now students, what exactly happens in the case of Kelvin cycle? Understand, Kelvin cycle. This Kelvin cycle has three main steps: carboxylation, reduction, regeneration. Here are the three steps. Here are the three steps of your dark reaction, students. Three steps of your dark reaction. Okay. The first one is called as carboxylation. First one is called as carboxylation. Students, understand it is called as Kelvin cycle, C3 cycle, or PCI cycle. Okay. Photosynthetic carbon reductase cycle. Photosynthetic carbon reductase cycle, students. Okay. Okay. Now, students, here we have RUBP called as ribulose 1,5 bisphosphate. Now, this RUBP is 5 carbon. Now, this 5 carbon will take one more carbon from the CO2. So, 5 plus 1 carbon, we get PGA. PGA is what? Phosphoglycerate here. PGA is 3 carbon. 3 into 2 is 6 carbon. So, 6 carbon here. 5 plus 1, 6 carbon here. So, RUBP plus CO2. RUBP plus CO2 will give you 3 PGA. Phosphoglycerate. And this step is called as carboxylation. This step is called as carboxylation, students. In carboxylation step, we have Rubisco is the enzyme here. Rubisco is the enzyme here, students. Now, phosphoglycolate, right? Phosphoglycerate will get converted to triose phosphate right now. Now, this second step is called as reduction. Now, in this step, students, 2 ATP is getting consumed here and 2 NADH is consumed here, students. Can anyone tell me which are right or students? This ATP and this NADP is coming from where? Light reaction. They're coming from light reaction right now. Okay. Now, this triose phosphate is 3 carbon, 3 carbon. It gets converted to glucose eventually. Glucose. Okay. Now, the triose phosphate now gets converted back to RUBP and it is called as regeneration. So, carboxylation, reduction, regeneration. And again, in this step, 1 ATP is getting consumed here. So, in total, students, it is called as what? It is called as 3-2-1-0. 3 means what? 3 ATP consumed here, 3 steps here. 2. Why 2, students? It because 2 NADP is consumed here. What is 1 here, students? 1 is nothing but here. This entire cycle, this entire C3 cycle is called as this entire C3 cycle. It is happening for 1 CO2. For 1 CO2, we need 3 ATP, 2 NADH for 1 CO2. Now, students, what is the 0 here? The 0 means here, students, 0 energy is produced here. No energy is produced in your C3 cycle. Now, you might ask me, why is it called a C3 cycle? It is called a C3 cycle because the first stable product here, the first SP here is 3 carbon. That is why it's called a C3 cycle. The first stable product here is 3 carbon, C3 cycle here. Okay. Why it's called as Kelvin cycle? Because the discovery was done by Melvin Kelvin. Melvin Kelvin discovered it, so Kelvin cycle. Okay, done, students. Done. Now, students, this is for 1 CO2, 3 ATP, 2 NADH. Enzyme, Rubisco enzyme. Okay. Now, students, in the examination, students, if they ask you by chance, students, all of you understood, right? 3-2-1-0 method. All of you understood? 3-2-1-0 method. 3 for 3 ATP, 2 for 2 NADH, 1 for 1 CO2. 0 means 0 ATP produced here. Okay. Now, for 1 glucose, what happens? For 1 glucose, all you need to do is into 6. See, all of you know, for 1 CO2, it is 3 ATP and 2 NADH. Just multiply into 6. Why? Because in 1 glucose, if we have C6H12O6, 6 carbon in 1 glucose. So into 6, because for 1 CO2 we need this much. For 6 carbon into 6, 18 ATP and 12 NADH. This right here is a PYQ. This is a PYQ right here. This right here, students, is a PYQ. Students, I'll give you break after my class. Okay. So this is a PYQ right here. 18 ATP and 12 NADH is required. Okay. Examples of Kelvin cycle, students: wheat, soybean, and oats. Example. Okay. Now, students, now we understand what is called as. Now, students, we understand called as what is photorespiration. Now, understand. Now, students, in life, what happens that there are some people who will be with you whenever they think you're beneficial. Whenever they think they can take advantage of you, they're with you. As soon as you do not have any use for them, they will leave you and go. There are many people like that. You will encounter, you might have encountered, you will encounter such people in your life. First encounter, when was the first encounter for me? Whenever birthday used to come, my birthday used to come near, one of the few people in my gang, they used to become extra friendly with me. Why? Because If I have to go give chocolate to every single classroom on my birthday, I have to take one friend with me. I have to take best friend. So all the friends will become best friends that time. They're like, because they want to come with me to give chocolate to every single classroom. Okay, every single classroom students. Now same way what happens in the case of your plant also it happens. In the case of plant, what is happening is that students, this Rubisco enzyme is at friend here. Rubisco enzyme is the friend students. Rubisco enzyme is the friend here students. This Rubisco enzyme, it can form a bond with oxygen. It can form a bond with CO2 based on the preference. Now, in general what happens? In general, this Rubisco forms a bond with CO2 and we have Kelvin cycle. Then we have a Kelvin cycle, normal Kelvin cycle. Here we learned, right? Here we learned. In normal Kelvin cycle, RUBP is forming a bond with CO2 and this cycle happens. This cycle takes place. But what is the but here students? The but here is that whenever there is less CO2, what happens? Or whenever there is less CO2 or whenever there is more oxygen, whenever there is less CO2 or whenever there is more oxygen, what happens? This Rubisco will leave CO2. This Rubisco now will form a bond with oxygen. Full scam, students? Full scam, right? Tell me yes or no? Full scam. This particular Rubisco will form a bond with oxygen right now, oxygen. And when that happens, when RUBP plus CO2 happens now, and your enzyme is Rubisco as usual, Rubisco enzyme, the particular RUBP undergoes something called as photorespiration, also called as C2 cycle. Why? Because the first stable product here is PG. This PG is 2 carbon. That's why C2 cycle. And we get phosphoglycolate plus PGA. 3 carbon plus 2 carbon is 5 carbon. Here we have 5 carbon here because oxygen is 0 carbon. So in the case of photorespiration, what happens is that we get phosphoglycolate. Phosphoglycolate is obtained here and one PGA molecule. 5 carbon, 5 carbon. That's what answer. Here there is no sugar formation. There is no ATP formation. ATP is used here. CO2 is used here. So, can I call this entire process, if there is no energy, nothing, no sugar, it is a complete wasteful process. Complete, utter waste it is. Complete zero waste. No use only. It has no use only. It is complete wasteful process for the plant. And students, remember, this entire C2 cycle happens in your C3 plants. It happens in your C3 plants, but it will never happen in the case of C4, students. It will never happen in the case of C4. Is that point clear to all of you, students? Is that point clear to all of you? CO2 is used. CO2 is used in this process. Again, CO2 is not used, it is released. If CO2 is not used technically, it is completely released as such. See, usually what happens is CO2 is very important for the plants. Because of this reaction, this CO2 is getting released as such only without being used. Right? So very difficult. Very difficult. Complete waste. Complete waste, students. Complete waste. Okay. When my mother calls me, sometimes my mother calls me C2 cycle, students. Sometimes my mother calls me C2 cycle. All of you understood? They say no waste body, sitting in the house, not doing anything. Okay. So this is what my mother calls me, C2 cycle. Okay. Now students. Now when does C2 cycle occurs here? Whenever there is more oxygen, whenever there is very less CO2 or very high light and temperature. Whenever there is very high light and temperature, it can happen. It can happen. Okay. Then students, then all of you. Now students, what exactly happens in the case of C4? Students, we have 15 more minutes, we will be done. We have C4 and then we have chapters done after C4. After C4, we have factors affecting 15 minutes will be done. 15 minutes will be done, students. Now understand what exactly happens in the case of C4. Okay. Now students, what exactly happens in the case of C4, you understand all of you. C4. Okay. Imagine there is one room here. Imagine there is a room here. And in this room, I will trap all the CO2 here. All the CO2 is trapped in this room. And in the same room, students, in the same room, I will trap Rubisco also. I will trap Rubisco in the same room, students. Okay. I'll extend 15 minutes. That's fine. And then I'll tell later on. Then that's all. All students are here only. Okay. Now students, in this room, let's say I will add Rubisco here and I will add CO2 in this room. Okay. And in this room, students, right now, what is happening? Oxygen cannot enter right now. Oxygen is not able to enter in this room. Now, students, you tell me in the chat right now, is there any way, is there any way, students, food respiration can happen here? Is there any chance that food respiration can happen here, students? In this one room right now, I have Rubisco, I have only CO2. What will happen? Now here, there is no chance at all. No chance at all. There is no option available. This CO2 has to bind with Rubisco, or Rubisco has to bind with CO2 here. There is no way it can bind with oxygen here. So this is what happening, students. This is what happening inside. Where? This is what is happening inside your C4 cycle. This is what happening in C4 cycle, students. In the case of C4 cycle, students, in the case of C4 cycle, we have something called as Kranz anatomy, students. We have something called as Kranz anatomy. Now, because of this Kranz anatomy, students, because of this Kranz anatomy, what is happening here is that, can all of you see, students, see here? Here we have inside bundle sheet cells. Bundle sheet cells right now inside. And they are in a wreath-like structure. They are in a wreath-like structure here, students. Now, inside this bundle sheet cells here, oxygen cannot enter here. It cannot enter. It cannot enter, students. Now, here what does we have? Here we only have CO2 and Rubisco. Only CO2 and Rubisco present here, and that is how only your C4 cycle works. In C4 cycle, what happens is that there is a wreath-like structure. The separate compartment is there. There are two cells here. We have outer cell, inner cell, and inside the inner cell, oxygen cannot enter. Only CO2 is entering. Only CO2 is entering here, and always food respiration is not happening here. Food respiration is not happening at all, students. It is not happening. Done, students? Done, all of you? All of you understood the concept here? This is what happens. See, okay, all of you see here. Here outside we have mesophyll cells here, and inside we have bundle sheet cells here. All the bundle sheets are very tightly packed. They're like very tightly packed. They're very tightly packed. Nothing can enter inside. Nothing can enter inside. Nothing can enter inside. This is the wreath. This is called as a wreath here. Wreath. See here. Bundle sheet cells are arranged in a form of wreath here. Wreath. Around vascular bundles. Bundle sheet cells have characteristics by having large number of chloroplasts, thick walls, and no intercellular space. So it means they are very tightly packed here, and nothing can enter inside. Okay. C4 plants tolerance to high temperature, high light condition, lack of food respiration, and great productivity of biomass produced here. More biomass produced here. Okay. See, students. It is also called as Hatch and Slack pathway. C4 is also called as Hatch and Slack pathway, students. Hatch and Slack pathway. Okay. Now see, students, this is what happens. See, can all of us see here? Outside we have mesophyll cell, bottom we have bundle sheet cell. Outside we have mesophyll cell, on the outside we have, and inside we have bundle sheet cells here. And bundle sheet cells are very tightly packed, and nothing can enter inside. Nothing can enter inside, students, here. Nothing can enter inside. So what do we have here, students? Initially what is happening? Initially what is happening here, students? CO2 is entering inside. Here CO2 can enter inside. Because outside mesophyll cells, CO2 can enter inside. Okay. Now, students, this CO2 is how many carbon? One carbon CO2. Okay. Now here, students, here the primary CO2 acceptor molecule is PEP. What is PEP? Phosphoenol pyruvate. Students, who was the initial acceptor of CO2 in the case of C3 cycle? Can anyone tell me, remind me? That was nothing but who was there? RUBP was there, students. RUBP was there. Okay. So PEP is taking it up, and in the presence of enzyme, again, pepcase, where mesophyll cell. Pepcase is present here, not Rubisco. We get OAA. OAA is nothing but oxaloacetic acid. Oxaloacetic acid is 4 carbon here. 3 carbon plus 1 carbon, 4 carbon. This is 4 carbon. That is why it's called as C4 cycle. It is called as C4 cycle, students. Now, this OAs get converted to malic acid. Malic acid is now transferred to your bundle sheet. In bundle sheet, malic acid is converted back to OAA. Now, this OAA is now converted to pyruvic acid. And that is how CO2 is released, students. That is how CO2 is produced in this particular area. So, do you realize we are transporting CO2? We are transporting CO2 from here to here silently. By like, let's say we have We took the CO2, we transferred salinity there. Transferred the CO2 right here. Now what happens, student? Now what happens? Now this CO2 now is entering the C3 cycle. So can I tell students, can I tell a statement here? C3 cycle happens in C3 plants. C3 cycle also happens in C4 plants. Tell me yes or no, students. Tell me yes or no. Tell me yes or no, students. C3 cycle happens in your C3 plants and C3 cycle also happens in your C4 plants. But the location is different here. Here C3 cycle is happening in bundle sheath cells here. Bundle sheath cells. But in the case of your C3 plants, it is happening in mesophyll. It is happening in mesophyll, students. Mesophyll. Okay. Now what happens? This pyruvic acid is again sent back up, is getting regeneration to again back to PEP. Again back to PEP, and this is how the entire C4 cycle works. Entire C4 cycle works, students. Because here we are making sure that in bundle sheet we only have CO2. See here. In bundle sheet, students, in bundle sheet we have only, this is a bundle sheet. In bundle sheet we have Rubisco enzyme and CO2 here. The trapping is done nicely. Trapping is done here, students. Trapping is there. And in this bundle sheet we have Rubisco enzyme. We have the Rubisco enzyme in the bundle sheet. In mesophyll we have pepcase here. Pepcase. Students, all of you understood? Students, all of you understood, students? Tell me yes or no. Students, all of you understood right now? Yes or no? This is what is happening. This is what is happening right now, students. Okay. This is your entire C4 cycle. See, students. Cell type in which Calvin cycle takes place. Calvin cycle happens in mesophyll here. Calvin cycle happens in bundle sheet, C4 plants. In the bundle sheet happens. Okay. Cell type in which initial carboxylation happens. Mesophyll, mesophyll both. Okay. Initial carboxylation, mesophyll here. Mesophyll. How many cell types? One cell here, two cells here. Two cells are present here. Which is the primary CO2 acceptor? RUBP here, PEP here. How many carbon? 5 carbon. PEP is going to be 3 carbon. Okay. PEP is going to be 3 carbon. Primary CO2 fixation product here, PGA is first product here. OA is the first product here. 3 carbon, C3 cycle, 4 carbon, C4 cycle. Does the plant have Rubisco? Yes, both have Rubisco. Does the both have pepcase? No. Here we do not find pepcase, here we find pepcase, present pepcase. Which cell the plant have Rubisco? Here mesophyll, here bundle sheet has Rubisco here. You can see. Bundle sheet has Rubisco enzyme bundle sheet. Okay, bundle sheet. CO2 fixation rate under high light condition here medium. Here very high fixation will happen. Whether photorespiration occurs at low light? No. Low light doesn't matter. Doesn't matter. Now, does the photorespiration happen in a highlight? It happens here, but no photorespiration here. See, can you see, students? There is no photorespiration in any case here. Photorespiration does not happen at all in the case of C4 plant. Doesn't never happens. Okay. Temperature optimum is 20-25 degrees Celsius here. More temperature here. Example: wheat, rice, maize, sugarcane, sorghum is the example here. Maize, sugarcane, sorghum is the example here, students. Okay. Friends, answer the question in the chat, all of you. Answer the question. Quick answers I want. Students, I want quick answers. Students, I want quick answers here. Done, students? Yes. 2024 question. In C3 plants, some O2 binds to Rubisco and have CO2 fixation. Yes, is decreased. Food respiration happen here. In C4 plant, mesophyll show very little. No, they will never show. They will never show, students. They will never show food respiration. Never show food respiration, students. Never. Students, answer the question. Students, answer the question quickly, students. Done, all of you? Done? Done? Okay. The P is the primary sodium acceptor in the case of C4. In the case of C4 students. Now students, we come to the last topic. Now we come to the last topic of this chapter. After this, I'll go off. Okay. Students, after this you can also get a break also. Again students, here you will not get a lot of questions here. Very few questions will come from here. Factors affecting students. Look at the face. This looks like your face also right now. Your face also looks like this only right now. Your face, because all of you are tired. All of you are tired. I realize. Okay. All of you need a small break also. Okay. Now students, understand, all of you understand. What exactly are the factors affecting photosynthesis here? Factors affecting photosynthesis here. There are multiple internal factors. All of them play a major role in increasing or decreasing the photosynthesis. Now, these are genetic ones. Then there is something called as external factors. Okay. Depending on the predisposition. Okay. For example, number, size and age of the orientation of leaf, number of mesophyll cell, again genetics. All genetic factors. Number of chloroplast, amount of chlorophyll, everything, all of these is called as genetic factors, internal factors. All of them are called as internal factors right now. Internal factors. Now, students, there is something called as Blackman's law of limiting factor. Okay. Now, students, all of you tell me in the chat right now. Let's say last year, all of you finished reading the NCERT 2 times or 10 times. All of you did PYQs. You did well in physics, chemistry, bio, but your marks did not increase, right? You did not get good marks. Why? Because you did not do a thing called as mock paper. Okay, so there was one factor which was done in least amount. Let's say you did only 2 mock paper. You did not do 20-30, right? So there was one factor which was holding back your rank, holding back your good marks, holding back you from getting to a government medical college. That one factor which is present in least amount, which can change your future, was a limiting factor then. Similarly, in the same case we have in the case of your chemistry also, right? Now here also what happens in the case of plant is that when there are multiple factors, if a chemical process is affected by more than one factor, in your preparation multiple factors are there, right? One factor, then rate will determined by the factor which is nearest to its minimum level. That is, whichever factor is present in least amount determines the rate of photosynthesis. The rate of photosynthesis is determined by factor which is present in least amount. Let's say we have water is very less. In summer time, water is very less. Summertime, right? So water becomes limiting factor. Let's take in winter. Let's say a lot of rain is there. If a lot of rain is there, sunlight is not there. So sunlight is very less. Sunlight becomes a limiting factor. Sunlight becomes a limiting factor there. Okay, friends. So we have light, CO2 concentration, temperature, water. So all of them, if whichever is present here, students, whichever is present in less amount, that becomes a limiting factor. That becomes a limiting factor, students. Now, students, do you think? So it looks like you're looking at God, no, students? Looking at God right now. Okay, students, do you think? See, students, full. Do you think sunlight is a limiting factor, students? Do you ever think sunlight is a limiting factor? No. Sunlight is generally present a lot. Sunlight, we have a lot of sunlight, and sunlight is generally not a limiting factor. Not a limiting factor, students. But it may be limiting factor, students. It may be limiting factor in certain dense forest. Certain dense forest, it might. Certain dense forest, you might. Okay. Okay. Now, students. Now, duration in sunlight also, right? There are 3 things. There's something called duration of sunlight. It doesn't matter much. We have short day plants, long day plants. It doesn't matter much. Look at sunlight. Okay. Light quality also doesn't matter much. We got red light, blue light. It doesn't matter. Sorry, it does matter. Okay. Whichever light we have, quality does matter. Red light and blue light, more it happens. Okay. Then we have light intensity also, students. Light intensity matters a lot. For example, students, let's say we add the light intensity, okay, between O to A, right? As we increase the light intensity, as we increase the light intensity, the rate of photosynthesis also increase. So O to A, as we increasing the light intensity here, there is an increase in photosynthesis. Happening. Now B to C, there is no increase here. There is no increase still. There is no increase in photosynthesis. Why? Because, students, O to A, it is increasing because initially light is a limiting factor. Initially, light is a limiting factor, but after some time, when the light is enough, the plant is like, I am enough with the light, I don't want more light. What will I do with 1 kg more light? Okay, plant doesn't need more light also. So after some time, what happens is that light is no more limiting factor. Here initially light is a limiting factor, but after some time, light is not a limiting factor. That's all, student, that's all. Okay. Now, what about CO2, students? CO2 is very important. CO2 is a major limiting factor because in the atmosphere, CO2 is very less. CO2 is very less, students. CO2 is very, very less in atmosphere. And as usual, if you increase the CO2 concentration, photosynthesis will also increase. Can you see here? Increase in concentration of up to 0.05% cause the increase in CO2 fixation rate. Obviously, that's why we see in a greenhouse. More bigger capsicum you will get, big capsicum you will get. Okay, now students here understand. Here C3 plants and C4 plants have a different saturation rate. Like all of you can't eat 1 kg biryani. Some of you can eat 1 kg biryanis who are very special, but all of you cannot eat 1 kg biryani. All of you have a limit to how much you can eat. So here also similarly, C3 plants saturation is around 450, while C4 plant is at 360. So C3 plants will work where? C3 plants will work very well in more CO2. So C3 plants can take more CO2. C4 plant very less CO2 they can take. Saturation is very less. Saturation is comparatively less here. Okay. Now, in a greenhouse, should I grow? In a greenhouse, should I grow C3 or C4 plants? Which one should I grow? Students, you can eat 1 kg biryani here, next level then. In a greenhouse, we generally grow C3 plants here because C3 plants need more CO2, they can take more CO2. They're very hungry people. Okay. So tomatoes and bell pepper are generally grown where? They're generally grown in your greenhouse because their saturation is very high. Very high, students. Okay. C3 plants. Now, students. Temperature has a very important role again, students. Temperature has a very, very important role, right? Temperature controls dark reaction because all the enzymes are present there. We have peptidase enzyme, Rubisco enzyme. So all of you tell me, students, if temperature goes very high, can the enzymes work? No. Enzymes will denature. Enzymes cannot work if your temperature goes very high. Temperature if it goes high, enzymes cannot work because they get denatured because they're proteins. Temperature goes very low, they cannot work again. Okay. So tropical plants have higher temperature optimum. Now again, students, C3 plants are lower temperature optimum than C4 plants. C4 plants can take more temperature. They can take more temperature, students. More temperature they can take. Okay. Now, last one is called as water. Water has a much more indirect role here. Understand here. That is, let's say there is less water. If there is less water, what happens is that all the leaf will curl. Students, can anyone remind me which cell is present in the case of monocot, which will help in curling of leaf? Bulliform cells. Let's say there is a monocot leaf. Okay, monocot leaf, and in this monocot leaf, all the water is lost. Surface area decreases. If surface area is decreasing, very less light is falling. Very less light. If very less light, less photosynthesis. Second case, students. Very less water, what happens? All the stomata close. If stomata is closing, what happens? CO2 is not entering. If CO2 is not entering inside, decrease in photosynthesis. Students. And with that, my session is over. Done, students. Done. With that, my session is over right now. Okay. Bulliform cells, exactly. Bully there. They're bully people. Okay. Students, that is your entire chapter. I want all of you to focus on entire again after the class. Revise one more time, and one more time you have to revise the entire C3, C4 and non-cyclic, cyclic. Apart from the students, entire chapter is very easy. Students, I believe it took 2 hours, I guess. Right? We started around 12:30, 12:40 we started around 12:40 and we finished right now, students. Okay. So with that, remaining 2 chapters I will teach once Gopika ma'am teaches 2 more chapters. See, if I teach all botany at once, you will go mental. Like, oh, so much botany, you will cry. Okay, I don't want you to cry. Okay, basically. So what we'll do is we'll ask Gopika ma'am to come now. Gopika ma'am will take some classes and then again I'll come back for respiration. Okay. Students, this is the one-shot lecture of all the human physiology and all the plant physiology. I want all of you to share this video to all your friends. Right? Share this video to all your friends today itself, right now itself, so they can join and enjoy the entire learning curve. Okay. Now, students, at least 340 plus, not 320, 340 plus. Okay, let me just call ma'am, students. Students, break now. Okay, break till 2:45. Break till I forgot about the break. Okay. Then, students, so 2:45 you can come back, all of you. Okay, students. So, joke. So, life only is joke, students. What joke I'll give you? Tell you. Life only is joke. Okay. So, human physiology will start again back at 2:45. I'll see you that time, all of you. Ma'am will see you. Okay.
Hi students, starting now, just give me a minute. Took a nap. My God, so much luxury. Took a nap, it seems. Students, exam is when? 3 more days. Okay. It's okay to take a nap, no problem, but still, 3 more days. Why did Basavaraj tie the money? Welcome, NEET. This is complicated than our minds. Okay, chalo, let's start. Ready? Ready, steady, go. Got imported, huh? Where is it? Where is it? Okay, board is only giving up, students. It's like, why you people continuously torturing me? Okay, so I hope all of you had lunch. Okay, energy is there. Yes, I have also had lunch. There is full energy. Which chapter are we going to do now? We are going to do body fluids and circulation. So the pattern is one heavy chapter, one light chapter, one heavy chapter, one light chapter. So this way even you won't feel like, oh my god, ma'am is telling so much information at one time, and also you will feel a little more light. Okay, let's go. Okay, yes. So we will start with body fluids and circulation. Okay, look here, students. Again, this is our previous notes itself. You have the actual notes of all of this. Okay, so don't waste your time asking the PDF of this. You already have each chapter ka notes. Okay, wherever I have added an image, I will send it to you. Okay, now. So blood, students, blood has two parts, plasma and formed elements. In that, plasma is constitute the 55% and formed elements constitute 45%. Okay, now plasma has so many components. In that, the main component is going to be what? Water. Okay, so 55% of total. In that, 90 to 92% is going to be water. 90 to 92% is going to be water. 6 to 8% is going to be protein, and many years this question has been repeated. This question has been repeated. Okay, 6 to 8% is protein, students. Which are the three proteins that is there? Albumin, fibrinogen and globulin. Okay, albumin, it's osmotic pressure. They will ask you match the following. They will ask you what? Match the following. So repeat after me. Albumin is going to help with osmotic pressure. Okay, when I used to teach before, I used to teach you about pressure in excretion chapter, but later I realized that no questions are coming from the topic, so why to waste students time. So it used to, like when we used to teach that, we used to talk about osmotic pressure there, how albumin will stop the water molecule, so it helps in balancing the pressure in the your tubules. Okay, fibrinogen, students, helps in clotting. Globulin, defense mechanism. Immunoglobulin, what is the meaning? Defense mechanism, right? Defense mechanism. So other than that, they also have nutrients like iron, sodium, calcium, HCO3, chlorine, magnesium, all these are present. And very important question, students, what is serum? Okay, plasma without clotting factor is called as serum. Plasma without clotting factor is called as serum. Clear? Now, look here, formed elements. Formed elements, why to even take tension? Because it is RBC, WBC and platelets, okay? Or erythrocytes, leukocytes and thrombocytes, correct? Yes or no? Yes, ma'am. Now, let's try to understand the formed elements in a tabular column to understand that, Is it possible to increase your marks? Too late to increase your marks. Okay. So look here, students. Number, the amount, how much number of each formed elements are there? RBC students 5 to 5.5 million. 12 to 16 grams of hemoglobin will be present in 100 ml of blood. That means in a healthy person, 12 to 16 grams of hemoglobin will be present in 100 ml. WBC students, 6,000 to 8,000 mm. 6,000 to 8,000 mm, and platelets is going to be 1.5 to 3 lakhs mm cube. Clear to all of you? Yes or no? Now, structure, when you talk about RBC, RBC has a biconcave or a dumbbell shaped structure. Okay, biconcave or dumbbell shaped structure. They are enucleated. They are enucleated. That means they do not have nucleus. They do not have nucleus. WBC on the other side, it is amoeboid in shape, asymmetrical in shape, because of which they are able to do diapedesis, right? Amoeboid movement. That means these people are amoeboid shaped, okay? Asymmetrical, nucleated. These people are nucleated. When you talk about platelets, platelets are not true cells. That means what? Ma'am, they are actually formed of, okay? They are actually formed of a big structure, okay, a big cell which is formed in the bone marrow, which is called as a megakaryocyte, and that megakaryocyte fragments to form your platelets. So the only cell which has nucleus, that if I want to do DNA fingerprinting, if I want to do something, okay, where I've got a blood sample, I have to take the WBCs because these people are nucleated, okay. Yes, excellent, Subra. Frog have nucleated, okay, frog have nucleated RBC. So that can come as a question. What is the difference between human and frog RBC? And multiple options will be given. Every student will tell enucleated, but no, frog has nucleated, humans have enucleated, okay. Yes, megakaryocyte. So that is not an actual cell, no. Now if I break a bigger portion and make small portion of it, I am not going to call that true cells, right? So same thing happens here. Now students, lifespan is 120 days. This is short lived, okay. WBC is short lived, and these people are mainly going to help with clotting, okay. Formed in the bone marrow, graveyard is going to be spleen. All this you people know. Erythropoietin protein. This is the protein that helps for erythropoiesis. One of the years they have asked you a PYQ. Erythropoiesis. Which of the cells among this help in erythropoiesis? So they had given JG cells as an option. So if you see JG cells and erythropoiesis, please write that as the answer, because JG cells releases erythropoietin, which is helping in erythropoiesis. Clear? Yes or no? Excellent. Very good. Okay. Now, short-lived students, granulocyte and agranulocyte. That means WBC is divided into granulocyte and agranulocyte. Okay. Now, platelets, clotting, factor 3 and 13 secreted by the platelets. So, there are around factor 1 to 13 are there. In that, 3 and 13, it is secreted by the platelets. Rest everything is secreted by the liver. Listen to the small things that I am stressing on. Okay, because that might come as a question tomorrow and you shouldn't regret. Shouldn't be like, oh, I should have listened to ma'am. Who's talking in my class, yeah? Huh? Okay, moving forward. Now students, WBC is just diapedesis, right? All of you know this. Squeezing out of the WBC. Now look here students, WBC, based on the presence of granules, it is divided into agranulocyte and granulocyte. Agranulocyte, you have monocyte and lymphocyte. In granulocyte, you have Bentens, Ben is there, no? Benten scale we had. So like that only. Basophils, eosinophils and neutrophils. Basophils, eosinophils and neutrophils, okay? Other factors are secreted by the liver. So there are 1 to 13 factors which are responsible for the clotting. Okay, in that 3 and 13 is secreted by platelets and the rest are secreted by the liver. Understood? Understood? Yes. So such small thing can come as question. Now students, look here. Never let Mohan eat bread. It's a trick to remember the order depending on the number. Okay, number of granulocyte and agranulocyte. Look here. Never let Mohan eat bread. Neutrophils are highest in percentage, 60 to 65%. L for lymphocytes, 20 to 25%. M for monocytes, 6 to 8%. E for eosinophils, 2 to 3%. B for basophils, 0.5 to 1%. Okay, they can ask you the highest and lowest, or they can ask you write it in order or arrange it in order, and they'll give you that current sequence, numbers they will give you. Never let monkeys eat banana or never let Mohan eat bread. Anything you can make. It is already made trick. It is not made by me. So you can just remember whatever you're good at. Okay. Thank you, King. Okay. Clear to all of you? Yes. Never let monkey eat banana or never let Mohan eat bread. Okay, I like never eat Mohan eat, sorry, never eat Mohan eat. Okay, so never let Mohan eat bread. I like it more to remember faster. So yes, you can remember as you want. Okay, monkey also if you remember, if you can relate more then please remember. Okay, so here students, granulocyte and agranulocyte. So percentage is 2 to 3%. Percentage now you already know. According to never let Mohan, just remember the percentage. Okay, 60 to 65, 20 to 25, 6 to 8, 2 to 3 and 0.5 to 1%. Okay, so here eosinophils is 2 to 3% students. Basophils is what? Okay, basophils is what? 0.5 to 1% and neutrophils is 60 to 65%. Now, depending on the shape, they can ask you questions. Okay, they can ask you questions. That is appearances, students. First, you have eosinophils. So, E for ears, E for headphones, earphones. Okay, yes, possible. Yes, what is happening? Okay, now look here, students. E for eosinophils, E for ears. So remember the headphones that we use, no? Same shape the nucleus is, one here and one circle here, okay? Basophils, S you remember, S you remember. So it is somewhat like S-shaped, okay? S-shaped. And neutrophils, students, it is multi-lobed, okay? Multi-lobed. Even if you take the shape of N also, you can just remember like this, multi-lobes are present, okay? Multi-lobes are present. Okay, very good, very good. Now, function, very important questions come from the function. Allergic reaction. Eosinophils help with allergic reaction. Now, question has come from basophils, okay, PYQ question. That is, which is the cells that secrete heparin, serotonin and histamine? What will your answer be? What will your answer be? Come on. Quickly. Nice trick, okay. Anyone from Karnataka? Oh, hi Preetu. Yes, tell me students. So the cells, okay, the granulocyte or agranulocyte, whatever they can ask you, but here you know it is granulocyte. The granulocyte that secretes histamine, serotonin and heparin. What will your answer be? Basophil. Okay, basophil. Again, they are going to help with inflammation. That means when you get an allergy, vasoconstriction, vasodilation, all that job is done by your basophils. Clear? Yes or no? Now, neutrophil students, phagocytosis. Neutrophils and monocytes both are going to help you with phagocytosis. Phagocytosis means engulfing of the foreign particle or the bacteria. Clear to all of you? This one box is enough for you to study. Don't break your head. Don't have to learn anything else. Percentage students, 6 to 8%. Lymphocytes 20 to 25%. Okay. Now look at the structure of it. Monocyte is kidney shaped structure. Okay. The nucleus is kidney shaped. Okay. Lymphocytes, actually the nucleus will be bigger, but I have just drawn to show the shape. So it is round or circular in shape. Okay, circular in shape. Again, I told you monocytes help with phagocytosis and lymphocytes. You already know from human health and diseases. These people are going to help with immune system. B cells, T cells, lymphocytes. Okay, yes or no? Clear? Yes. Okay, very good. Now, moving forward, blood grouping. Okay, blood grouping. So you understood WBCs, you understood the formed elements. You do not have to break your head in anything else, only numbers you remember. This box you remember very well. Okay, this box, the images you remember, and this also, not this. Okay, this box also has everything that you need. Okay, this box also has everything that you need. Now we will learn blood grouping. Okay, blood grouping. So students, when we talk about blood grouping, what do we know? We know that there is ABO blood grouping that we normally talk about, okay, which was given by Lansky. Now, I cannot see you people answering. If you don't answer, you're not studying, okay. Please keep answering or please let me know that you're understanding. Got it? So, it is easier for me. Yes, okay, very good. So, ABO blood grouping, which is given by Landsteiner, RH blood grouping, similar to the rhesus monkey, we will come into that, students. So, what is this ABO blood grouping? blood grouping is just to make us understand the compatibility. That means if blood grouping did not exist, if rhesus grouping did not exist, we would never know if someone is compatible to someone or not. Like if I want to do a kidney transplantation, I cannot do that, right? So ABO blood grouping makes us understand that this blood group can receive blood from this person because this person has this antigen or this antibody. Okay, so depending on that, there is blood grouping done. Now look here, students, look here. Similar to the rhesus monkey one, we have A positive, which is accumulated like A positive, that is rhesus positive is there for 80% of the people and 20% of the people are negative. How many of you are negative students here? Not negative by like in the sense mentality wise, I'm telling blood group. Blood group, okay. Now, two antigen students chemically that induce response, two antibodies, that is anti-A and anti-B immune system, okay. Yes. Now, look here. Rh positive, that is if we are A positive, that means we have Rh grouping in us. If we are negative, then we do not have Rh grouping in us, okay. And Rh incompatibility leads to erythroblastosis fetalis, okay. Erythroblastosis fetalis, we will come into that. First we will learn this box, okay. First we will learn this box. Look here, blood group A is there, okay. If A blood group is there, students, the antigen is going to be what? A, because this is the one that is going to make the chemical response, okay. So A will be there. A antibody, what will be there? Anti-B will be there. The antibodies in the plasma is going to be B. Now please remember, antigens are on the RBC and antibody is in the plasma, and this was asked as a PYQ and majority of the students could not answer. Why? The option was given antibody in RBC, antibody in RBC, antibody in plasma, antibody in plasma, okay. So students did not notice the plasma word. So please, I am telling you again and again, antibodies in plasma, you have to notice if it is plasma, and here you have to notice if it is RBC. Will you all do that? Will you all do that? Okay, all these are small tips, okay, that you know students miss out on. Now, donors group students, that means A and O can give the blood to these people, okay. Now, B, what will happen students? Obviously, the antigen will be the same, B will be there, but antibody is going to be the opposite. That means anti-A will be present in the plasma, and B and O blood group can give. Now, AB students, AB can be given, A can also give, B can also give. That is again, sorry, they have antigen A and B, but they do not have any antibody, because of which these people will become universal recipient, okay. They can take blood from anyone and everyone, okay, because these people do not have antibodies, okay. So that is why they can become universal recipient. Okay. So that is AB, A and B. Okay, A, B, A, B, O. Now O blood group students, these people do not have any antigen, because of which these people become universal donors. Even though they give blood to everyone, they only can take it from O. Okay, they can only take it from O. Otherwise, the chemical response will happen. So in scenarios where we cannot do mixing of this, but there is also scenarios where we cannot do mixing of the RH complex, okay. That is, if A positive is there, that means this person is RH, okay. That means it has the RH group, okay. If the person is negative, that means this person doesn't have RH. So this and this will not match, correct. So that leads to erythroblastosis fetalis. That leads to erythroblastosis fetalis. So here you see students, RH antigen similar to one present in rhesus monkey will be present on the surface of the RBCs of majority. Such individuals are called as RH positive, and when this antigen is absent, they are called as RH negative, okay. Now, RH negative person, if exposed to RH positive blood, will form specific antibodies. Obviously, ma'am, if a positive person meets a negative person, automatically there will be some reactions that happen in the positive person's body or negative person's body, depending on it. But here, since we are talking about erythroblastosis fetalis, we know RH negative woman, if she conceives RH positive baby, okay, in the starting there will be no mixing up of blood, correct, okay. In the first pregnancy, the mixing up of blood will be not there, but during delivery of the first child, there is a possibility of exposure of the maternal blood to the small amount, okay, small amounts to the Rh positive blood in the fetus. So the moment the mother gets exposed to the, blood, the mother's body will start making antibodies. But will it affect the first child? Tell me yes or no in the chat box. Will it affect the first child? Come on, tell me. Will it affect the first child? I'm Rh positive. Okay, I'm not a negative person. Okay. Yes, Rh negative mother, Rh positive fetus, erythroblastosis fetalis. Come on, tell me. Yes, very good, very good. Ma'am, it will not affect the first child. Okay, until students, there is a mixing of blood before the delivery. Okay, if there is any complication and mixing up of blood has happened in during the pregnancy time, then the first child is in problem. Otherwise, generally the first child will not be in problem. Second child will be in trouble because mother's body will recognize that Rh positive as a foreign particle and body has already started making antibodies against it. Okay, antibodies against it. Okay, clear to all of you? Okay. Now, in such cases, the mother starts preparing antibodies against Rh antigen in her blood. In case of a subsequent pregnancy, the Rh antibodies from the mother can leak into the blood of the fetus and destroy the fetal RBC. That is why it is called as erythroblastosis fetalis. Okay, fetalis. And this could be fatal to the fetus or could cause severe anemia and jaundice to the baby. Okay, it's not like death will happen immediately, but it can cause severe anemia and jaundice, and this condition is called erythroblastosis fetalis. This can be avoided by administrating anti-Rh antibodies. Okay, Rh antibodies to the mother immediately after the delivery of the first child, or we tell about the Rogam vaccine. Okay, Rogam vaccine which can be given, which will eradicate the antibodies that is made against Rh that is positive. Okay, yes, very good, Rihanna. Yes. Anyone from Karnataka? I am from Karnataka. What do you want more than this? What do you want more than this? Huh? Okay, let's do coagulation of blood. All of you, some energy I want. Come on. All of you, some energy. When I feel the energy is going down, I'll also feel sleepy. So come on, give me some energy. Your energy is directly proportional to my energy. Okay, opposite of Boyle's law. Come on, faster, faster. Very good, I'm proud of all of you. Been staying for good amount of time here. Yeah, Rogam in even Malayalam is disease only. Rogam we tell. Okay. Yes. I am from Kerala, but I am here also. So I am a hybrid. Okay, yes. Okay, students, there's a trick to remember this. Okay, there's a trick to remember the coagulation. What is the trick? PK went to PT field. PK is there, no? PK movie. PK went to PT field. Okay, so P for thromboplastin, K for thrombokinase. I'm only focusing on the second word because thrombo is going to repeat. Okay, so P for thromboplastin, K for thrombokinase went to PT field. P for prothrombin, T for thrombin, F for fibrinogen. So in that exam tension, no? Okay, exam tension, you will forget. So remember, okay, PK, where did he go? He didn't go with Anushka. Okay, he went to PT field. Okay, so PK went to PT field, thromboplastin, thrombokinase, PT, prothrombin, thrombin and fibrinogen, and then ultimately fibrin. Okay, so you can also remember PK, PT field also. Now students, remember factor 4 is an ionized calcium. Vitamin A also is very needed for your clotting. Okay. Now what exactly is clotting, ma'am? Now let's take example. Who wants to fall? Who wants to fall in the class? Can I call anyone's name? Okay. So let's say Purple. Purple is the name. Purple fell down. Okay. While he was running to NEET examination, or while he was coming very happily after NEET exam, he fell. So immediately blood will start going. But after some time, he'll notice that even though he's in pain, okay, that he still okay to walk. Okay, he's still okay to walk, and he noticed that the blood has stopped. Okay, so he'll be thinking what exactly happened, and obviously whom will he call? He'll call me because I'm his zoology teacher. He'll call me and ask, ma'am, exactly what happened. First, I will scold him for not studying the concept and going for NEET. Second, I will tell him the answer. So, basically, students, whenever an injury happens, okay, whenever an injury or a trauma happens, first thing that will be released will be thromboplastin, okay, thromboplastin. After that, that will activate thrombokinase. So, please remember, this is a cascade of reaction. That means one after the other, one after the other, one after the other. Now, imagine you arrange the cards and you touch one card will hit. So, each step is important in the whole process, okay. So, thromboplastin, thrombokinase, thrombokinase to prothrombin, thrombin to thrombin. Thrombin is like an enzyme, okay. That's thrombin to fibrinogen, fibrinogen to fibrin. Now, remember, this fibrin will be active but it will be insoluble, which will make an entire mesh, okay, which will make an entire mesh, and that entire mesh is going to block the blood from going out anymore. Clear? Yes or no? Yes or no? Okay, so that is what happens, okay. PK went to PT field, injury or trauma, thromboplastin, thrombokinase, prothrombin, fibrin, thrombin, fibrinogen and fibrin. Clear? Yes or no? Yes or no? Okay. Next one is about heart. Is this clear to all of you? Any doubts so far? Any doubts so far? She. Okay. You people should put names. Now, how do I know what color is purple? What is the gender of purple color? It's very difficult for me to understand. No, so I'm telling with assumption. Assumption. Okay, so you people shouldn't be like, oh, take it to heart. At this, like in my class, just forget your gender. It does not matter. If I'm telling she, it's she. If I'm telling he, it's he. Okay? Clear ancient. Okay. Okay. Who is this fellow? I think he opened a gym, student, and he has come on YouTube for free ads. Okay. What and all variety creatures come to class, you see. Variety of variety creatures. Yes. Now, vertebrata. Okay, vertebrata, that is we are going to find about the heart. Okay, the heart. So, look here, all of you. This is your fish. This is not your fish. This is the heart of a fish. Okay, this is heart of fish. Got it? Yes? Okay, now look here. Students, single circulation happens. That is, the body has used up the blood, okay, the oxygenated blood, and it has made it deoxygenated. Now, this deoxygenated blood, you all know fish has two chambers, atria and ventricles. This deoxygenated blood will come like this all the way, and it will get purified in the gills, and the purified blood will again go back to the body. Now, to give extra support to the atria and ventricles, okay, extra support to auricles and ventricles, we have sinus venosus and conus arterios, okay, sinus venosus and conus arterios. Clear to all of you? So, this we have already studied in a frog as well, okay, sinus venosus and conus arterios, triangular structure and sac-like structure. So, they are going to give an additional, like they are like the accessory chambers, okay. So, this is the one-step circulation, nothing much to break your head for. Now, incomplete circulation, that is amphibians and reptiles, okay. This is three-chambered, right atrium, left atrium, and everything mixing up will happen in the ventricles, okay. Mixing up will happen in the ventricles. Now, complete double circulation happens in our body, okay. Complete double circulation happens in our body, okay. Now, let's learn the heart. Ready for heart? Ready for heart? They're like, yes, ma'am, we are ready to give you our heart. But please teach us this. Come on, quickly. Oh no, Meenakshi. Students, I know data will be consumed, but I hope all of you have Wi-Fi. Okay, and your parents will recharge today and give you people because they know that you're doing something good. Okay, you're doing something good. So ask them to recharge and give, they'll recharge and give. Okay, come on, let's go. Yes, crocodile is an exception, okay. Yes, now look here, students, this is your heart, okay. Don't worry about the complicated structure here, you only learn what is required for you. First of all, you know your heart has 4 chambers. Anyone has doubt in that? No. Right atrium, right ventricle and left ventricle, okay. Now, first of all, think of it, I need to give my deoxygenated blood to the heart. So I am going to give the deoxygenated blood to the heart with the help of superior vena cava and inferior vena cava. Superior vena cava will collect all the blood from the superior part of the body. Inferior vena cava will collect all the blood from the inferior part of the body and give it to the right atrium. Now right atrium, obviously atriums will not carry it for longer time, it will give it to the ventricles. So right atrium to right ventricle, the blood has gone. But between this, I need to make sure there is no mixing up of blood, there is no backflow of blood. For that, I have kept a valve which is called a tricuspid valve. Why do you call it tricuspid, ma'am? Because it has three cusps, okay, three cusps. So it is called the tricuspid valve, which is like the Benz symbol, okay, tricuspid valve. Now students, remember, the blood has gone here, okay. Now from here, from the right ventricle, we need to take the blood out to all the body parts. Yes or no? We have to take it to the hoot. We have to take it to the lungs because oxygenation has to happen. So now to take the blood from the left ventricle, there is going to be a pulmonary artery that is going to come. Who is going to come? Pulmonary artery is going to come. The only artery that is capable of carrying deoxygenated blood. Generally, our artery will carry oxygenated blood. The only artery that carries deoxygenated blood is going to be a pulmonary artery. Now, pulmonary artery will take the blood and give it to the lungs, and lungs will do oxygenation, add the oxygen which we studied in breathing and exchange, and give it to the left atrium. With the help of whom they will give? With the help of pulmonary vein. Please remember there is two pairs of pulmonary vein, one which is opening here and one which opens here. Okay, one which opens here. So the blood will come into the left atrium to the left ventricles it will come. Now again, from the left ventricles there should be someone to take it. For that we are going to take the help of aorta. Aorta will give with the small arteries, okay, hepatic artery, renal artery, it will give the blood to all the body parts. Clear to all of you? Clear to all of you? Yes or no? Yes? Done, done, done? Done, done, done? Is this clear? Now, let's learn the other structures which we did not learn. First of all, left atrium to left ventricle, there is a bicuspid valve because there is only two cusps. Okay, two cusps. That is called the bicuspid valve or the mitral valve. Tricuspid valve can also be called as the AV valve. Now, students, if you look at the heart, there are clear partitions. Those are called as septums. What are they called as? Septums. Okay, atrioventricular septum. Okay, atrioventricular septum. And these septums will make sure that the heart is proper in chambers. Now, yeah, look here. Can you see these foldings that I have done like a muscle foldings? These are called as papillary muscles. That is this portion which is like a wave. Students, like this. In physics, you have no compression and rarefaction. Like that, this portion is called your pulmonary muscles, from where the chordae tendineae arises. Chordae tendineae means, okay, chordae tendineae means what, students? A flap-like structure that is going to hold the valves in place. A flap-like structure that is holding the valves in place. Clear? So, here try to understand, this much only is there for you. Okay, this much only is there for you. Yes. Now, students, this pulmonary artery and aorta also, we should make sure that with the contraction, yes, they are taking the blood, but there is chance of the blood coming out. Backflow can happen. To avoid that, there is a semilunar valves in the pulmonary artery. So, we call it pulmonary aortic or aortic semilunar valves. And sorry, aortic semilunar valves is in aorta. Pulmonary artery also has semilunar valves, which is called as pulmonary semilunar valves. Aortic also has it, which is called as aortic semilunar valves. Is that clear? Yes? Clear? Done, done, done? So, look here, you will be able to understand. Okay, you will be able to understand. So, these are your chordae tendineae, like a flap-like structure. This is your chambers, and you can see how the pulmonary vein is opening inside. Okay, one circle here, and those two are opening inside. And you can see how the interventricular septum is very thick. Okay, very thick, and you can see how inferior and superior vena cava is opening into the right atrium. Can you see that in the diagram? Yes or no? Okay, valves of Eustacia is for your here vena cavas when we tell. Okay, walls of Eustacia. This one, walls of Eustacia is actually valves which are present in the inferior vena cava. Some additional information there. Okay, additional information there. Clear to all of you? Yes sir? Yes or no? Okay, done. Now students, this is the whole story what I told. The whole story what I told, I have written it here. Look here. Right atrium. Okay, blood is coming to the right atrium, yes or no? How is the blood coming to the right atrium? It is coming with the help of vena cavas. It is coming with the help of vena cavas to the right atrium. Right atrium to the right ventricle, students, the blood is flowing with the help of what? Tricuspid valves. With the help of tricuspid valves. Right atrium to the lungs, it is opening with the help of pulmonary artery. I mean, the blood is getting carried with the to the lungs with the help of pulmonary artery. Now, in the lungs, oxygenation will happen, and the blood from the lungs, it is coming to the left atrium with the help of pulmonary vein. Yes or no? Yes or no? I just want yes or no in the chat box. Okay. Pulmonary artery done. Now pulmonary vein. Lungs to the left atrium, pulmonary veins. Left atrium to the left ventricle is bicuspid valve. Yes, ma'am. Opening. Understood. Now, left atrium to the body I want to give. For that, I am going to take the help of aorta. Correct? Now, tissues, that is tissues or bodies. Body will be waiting to give the deoxygenated blood. To whom? To vena cavas. Vena cavas are going to bring it to the heart. Heart is giving it to the lungs for oxygenation. Yes, this is the whole story that I taught you right now. That's it. This much only you have to study. This much only you have to study. Clear? Questions will come here and there, but nothing to worry. This much, this is the only thing that you have to worry about. Okay, worry about. Okay, now cardiac cycle. Ready for cardiac cycle? Okay, let's go. So, cardiac cycle, students, look here. Basically, cardiac cycle happens in 0.8 seconds. Okay, 0.8 seconds. This I think all of you know now. In that 0.8 second, 1 second, 0.1 second is AS. Atrial systole. Systole means what? Contraction. Diastole means what? Relaxation. Okay. And then students, what do we have? We have 0.3 seconds. We have ventricular systole. Okay, for longer. And 0.4 seconds we have joint diastole. JD. Okay, JD we have. Now look here. During atrial systole, what is going to happen? Ma'am, obvious, ma'am, atrias are going to contract. Now, students, remember, when you hear the sound lub and dub, always remember that closing is the how the sound will be made. Now, if I am opening a door, sound will not be made. But if I shut a door, sound will be made. So, here, atria will contract. Okay, atria will contract. AV valves are going to open. Okay, 30% ventricles are going to get filled. This is also a PYQ question. This is also a PYQ question. How many what is the filling? How much does the ventricles fill? Okay. Then ventricular systole, students, ventricles contract. Okay. Isovolumetric contraction. That means the entire volume will be maintained. Iso means same. Okay. Semilunar valves will open. Semilunar valves of pulmonary artery and aorta will open. Why? Simple, ma'am. Let's, students, consider the heart. Okay, consider the heart. My hand is the heart. Okay, right atrium. Right atrium contracted. Now the blood has come to the left atrium. Now left atrium contracts. What will happen? They will be forced. The blood will be forced from the right ventricle and the left ventricle to go outside. Correct. So simple, no? Semilunar valves are opening. Yes, ma'am. Very simple. Right. Very simple. Yes or no? So opening will happen. Now, joint diastole, students, 70% ventricle filling will happen. Joint diastole means what? The blood has gone and complete relaxation is happening, leading to the ventricles getting filled again and complete relaxation. Okay, complete relaxation. 19 conjues are there in the class. Oh my God. That's a lot of them, yeah? How do we tackle 19 conjues? Quickly smash the like button, students. 19 of them have not liked. See, we got data also. Okay, we got data. Look here. Complete cardiac diastole, atrial systole and ventricular systole. Can you understand this? So, atrias are going to contract. Okay, you think of the heart. Whenever I am telling something, you think of students, can you believe after this, we will not have Gopika ma'am's class. You will not have schedules. Okay, for any, why my heart is not coming today. Okay, you will have, I'll not send message on Telegram asking you to join, right? So, this is like our last proper lecture. After this, only question practice. So, after this, you will not get my classes where there is entertainment, there is, roasting. So, today you should be more energetic that, okay, let this, I also pray, let this be your last class which you're watching of mine also, so that next year you're in your MBBS college. I do not want to be, you know, what is that? That possession shouldn't be there. Oh, my students shouldn't leave me and go. No, I want my students to go with flying colors. I want you people to come back and tell, right now I am in this government college, ma'am. Right now I'm in AIMS, ma'am. I'm living a nice life. Thank you. You have contributed something to it. Okay? Right? 2026, I'm there. I'm telling my 2025 batch should move out. Okay? Move out. Right? Exactly. My students should move out. NEET 2025 students should have that relief. Okay? Definitely, I'll pray for all of you. Okay? So, students, here what is going to happen, students? See, right atrium, okay? Right ventricle, left atrium, left ventricle. So first they are telling that atrial systole will happen. So here first contraction will happen. Yes, ma'am. Here if contraction happens, students, the blood will move down. Yes, ma'am, the blood will move down. Okay, blood will move down. Now when here contraction happens, okay, when here contraction happens, what will happen, students? The blood will be forced. The blood will be forced to move out to this, through this people, right? Blood will be forced to move out from this pulmonary artery and aorta. Okay, it will be forced to move out. So that is what they are trying to say here. Okay, that is what they are trying to say here. Correct? Yes or no? Yes, you people are also always in my heart. Okay, every batch of students, I still remember their names and I still think of them. Some of them text me now also on Instagram, on Telegram, they text me. You know, whenever they're going through some problem, they text me. Okay, now look here, students. This is the entire phase. Okay, first we will start with atrial systole. So the SAN now generates an action potential which stimulates both the atria to undergo simultaneous contraction. Both the atria are going to undergo simultaneous contraction. Who is going to initiate it? SAN, sinoatrial node. Okay. Now, the atrial systole. This increases the flow of blood into ventricles. Okay, this increases the flow of blood into ventricles by 30%. Clear? This is from your tattva. This is from tattva. Okay. Now, ventricular systole. The action potential generated by the SA node, okay, SA node is conducted to the ventricle side by the AVN and the AVN bundle. Understood? So, you start with the SAN node, okay, SAN will bring it to AV, AV will bring it to who? Bundle of His. Okay, bundle of His. So, like that it has moved slowly to the ventricles. Okay, slowly to the ventricles. Okay, now look here where the bundle of His transmits it through the entire ventricular, okay, musculature. Okay, why do you get reply after 1 week? Because I'll be 24/7 taking class here. Okay, no time to even open Instagram, students. The atria undergoes relaxation. Now what will happen? The atria will relax. Okay, he's like, okay, my job is over. You people will relax, no, on May 5th, like that. Okay, they'll be relaxing because coinciding with the ventricular systole. Okay, ventricle systole increases the ventricular pressure, allowing the closure of tricuspid and bicuspid valve. So the first sound will be made here. The first sound will be made here. That is going to be what, students? Lub sound. Right. First sound that is going to be made is going to be lub sound once the AV valve closes. Okay, lub sound. Now, attempted to the backflow of blood into the atria. This produces the first heart sound. Got it? As the ventricle pressure increases further, the semilunar valves guarding the pulmonary artery and aorta will open. They are forced to open. Why? Because of the pressure. Because of the pressure. Clear to all of you? Is this super clear? Any doubts in this? Clear to all of you? Yes or no? Please tell me. So, students, what has happened? First sound. Okay, first sound is going to be lub sound. Okay, lub. No song like that exists. Okay, I'm only making it up. When I started the song in my head, I was like, okay, this is going to be a song now. But then I realized there's no song like that. Okay, I'm just making it up. So, students, I made 2 tricks for it. Okay, because I had a lot of time in my life. No, so lazy and dance on Sunday is one trick. That is L for lub, A for AV valve, D for dub, S for semilunar valves. Okay. Second trick is Ammu wants laser bulb. This is more easier to remember because you call me Ammu ma'am. It's easier to remember. Ammu wants laser bulb. In that want spelling is V. Okay. A V valve closes to cause lub sound. AV valve closes to form lub sound. Okay. Clear? AV valve closing leading to lub sound. So students, lub, L is there. No. So these are low pitched sound but long duration sound. Low pitched sound but long duration sound. Okay. Now semilunar valves are closing, students, that will lead to dub sound. That is high pitched and short duration. So remember this much only. L for lub, L for low pitch, L for lub, L for long duration. Uska opposite you write here. Okay. Oh, Hindi and all is coming, students. I don't know after 7 hours what language and all will come. Okay. Hindi and all is flowing in my mouth. Yes. Singing so good. That's not even a song. Okay. Some song I only made. Okay. Yes. So I'm very creative when I lack sleep and when I'm stressed. Okay. So God is like, that's why I give you more stress. Oh. Okay, come on. Now students, listen here. Again, we have to draw the heart. Okay. Done. Heart is done. Okay. Yes. Now, how does this cardiac cycle that you talk about now, how does it happen? So, we have atrial systole, ventricular systole and joint diastole. No, it initiates from this point. Everything initiates from this point. Okay. That point is what point? Okay. SAN node. Okay. That is called the pacemaker. SAN is also called the pacemaker. Pacemaker. So he is going to regulate the heart. Okay, he or she. Okay, he or she. That I'll sing for you people this time also. Okay, so SAN node. Okay, SAN node, students, it will generate the action potential. The moment the action potential arises, you know that atrial systole will happen. That is around 0.1 second. So you please connect this and cardiac cycle together. Now from here, it is going to go where, students? To the AV node. Okay, sorry, to the AV node, which is present near to the AV, that is the tricuspid valve. Okay, now from there, it is going to this point, which is the bundle of His. Okay, bundle of His. From bundle of His arises your Purkinje fibers, which are completely present in the ventricles. Okay, completely present towards the ventricles and not towards the atria. Okay, Purkinje fibers. So remember that the moment the SAN node, brings in an action potential to the AV node, by then atrial systole would have happened. AV node will give it to bundle of His. By that ventricular systole will be ready. Okay, for 0.3 seconds that will happen. By the time both the valves are going to open and the blood is going to go out with aorta and pulmonary artery, and then joint diastole will happen. Clear to all of you? Yes or no? Yes or no? Will you forget cardiac cycle again? Cardiac cycle again? They're threatening, students. Thank you, Vaishnavi. Correct? Done? Yes, lower left. Big boss. Tanish, चल क्या रहा है big boss? Okay, big boss चल रहा है. Yeah. Now students, ECG. Okay, we are going to be done with the chapter. Just imagine in 30 minutes, no, 45 minutes I finish the chapter. ECG is electrocardiogram. Okay, electrocardiogram. Now, the problem is people could have made the life little more simpler by labeling the graph as gram and the instrument as gram. Instrument as gram and the graph as graph. But no, they wanted to twist it. Okay, they wanted to make your life more easier and feel so lucky. So they made the instrument name as graph. Okay, electrocardiograph, and the graph name as gram. Okay, the graph name as gram. So electrocardiogram and electrocardiograph. Okay, clear to all of you? Yes. So exactly, they'll know. What is that? 38 minutes to 3. Now all of you wake up. They'll play a music and you'll start dancing, or the task for the day. Okay, Task for the day is to smash the like button. Come on. Instrument, electrocardiograph. Graph is electrocardiogram, students. Basically, they will connect 3 leads to a person. Okay, 3 leads to a person. 2 on the wrist and 1 in the ankle. Up to 12 leads you can also, nowadays up to 12 leads also the electrocardiograph can be fit into. This is to just make you, this is to understand the heart, again the cardiac cycle, how the heart contracts, relaxes. So look here, this is your entire graph. Okay, now if you see here students, first you will start with P. Now many students in the examination, what happens is when they are trying to learn something, they have a tendency of forgetting, is it D or it is repolarization. So I'll give you a trick for this. The trick is doctor. See, I know whoever forgets it, you people will not forget, because this is what your name, front of your name, it is going to be there. So D means students depolarization. Okay, even for your neuron control also, remember this. First you have depolarization, then you have repolarization. Okay, now when you are doing depolarization, just like that brackets that you study in maths, students, inside the bracket, let's say 3x plus 6 is there. Okay, and here 2 is there. You will multiply both, no? You will multiply this, you will also multiply the same way. Depolarization you will do for atria and you will do for ventricle, then only you will go for repolarization. At this point, I don't know what I am teaching, students. Like, I don't know if I'm teaching maths or biology or chemistry or what. Okay, so is that clear to all of you? Is that clear to all of you? Doctor, you remember, start with depolarization, then go with repolarization. Okay, so the first amplitude, the wave that you are seeing, P wave, is going to be atrial systole, that is atrial repolarization, exact copy of what you studied in cardiac cycle. You start with atrial systole, then you go for ventricular systole. Okay, same thing is going to follow. Clear? Now, second, the biggest wave that you are seeing is QRS. In that, S is going to be the lowest amplitude, R is going to be the highest amplitude, and that is going to lead to ventricular depolarization. What is going to happen? Depolarization, ventricular depolarization is going to happen. Clear? Now, after depolarization, what should happen? Repolarization should happen, right? Ultimately, we have to go to a resting. So, next is going to be ventricular repolarization, that is of the T wave. Okay, T wave. Clear? Successful. Wow. After 5 hours. Okay, yes. Now, students, remember that atrial repolarization also happens, but the amplitude is too small to be captured on an ECG. Okay. Now, have you seen in this serials and all? The serial, okay, serial, what will happen? The heroine will be coming. That time this fellow's heart will be like this, okay. When the heroine enters, suddenly his, you will see his ECG going like this, okay. And doctor will be like, oh my god, miracle, medical miracle has happened. But is this medical miracle? Of course not. Because if you look at the ECG, the doctor can understand if the patient has any problem with the heart or not. Okay, we also say heart murmuring. That means if the waves are in a different pattern, then we will immediately understand that there is an issue with the person's heart. To a limit, looking at the ECG, the doctor will understand if there is any problem in the heart. Otherwise, definitely you have to go further to do the scans and other things. Okay, yes, okay. So, will you do a question now? Students, all the disorders you revised from my video, okay? Now, no time to do disorders. Come on, answer the first question. Ho gaya chapter. Done. I am also proud of myself. A medical miracle. Yes, every serial mein the doctor will come and tell. I saw one serial, students, my god, that lady, he died. Okay, he died. Doctor comes out and doctor is like, I can't help it. That lady goes inside. She's like, kya doctor hai? She pushes him. She goes inside. She does the operation and she'll be like, andar jaake dekho. She'll tell, go inside and see. My husband is alive. Who are you to tell that my husband is, like she's asking a doctor, who are you to tell that my husband is dead? Like, who else should tell? Okay, she expects that person who is selling mask in the ground floor to tell that your husband died. No, she's like, who are you to tell my husband died? She goes inside, students. That serial shocked me. That serial, though, it took me like 5 minutes to process. What am I doing? And that too, I'm a teacher who teaches for NEET. I'm thinking what to do. Okay. What happened, Lakshmi Priya? Are you okay? It's like, Lakshmi Priya was in between the sleep. Suddenly she woke up and she's like, my God, I didn't study malaria's life cycle. So I will type it in the chat box. Okay. Male is not there. It is only in female Anopheles mosquito. It is only in female Anopheles mosquito. Chikungunya and dengue notes, I think last times I have. I will share that, okay? Okay, I'll share that. Admit card got released, huh? Okay, let me check. Wait. Are you sure? Chumma, don't tell. Okay, done? Okay? Real. Yeah. Okay, look here. Answer this. Answered. Which one of the following is a matching pair? Yo, what is the answer for this? Lub sharp closure of AV valves at the beginning of ventricular systole. Dub sudden opening of the semilunar valves. Not at all opening, students. Opening is wrong. Okay. Pulsation of radial artery. No. Initiation of heartbeat. No. So obviously it is option number A. Okay, it's option number A. Correct? Confirm. I can't check. I'm not able to check. Okay, in standard ECG, students, the P wave represents what? Quickly answer. P wave represents what? Okay, wait. 1 second. Why it's... Oh, yeah, released. Released, no. 34. Released. Yes, students, it is released. Okay. Yeah. Okay, just now a notification came. Okay. Done. Okay. You got your admit card. Oh, nice. It's not April Fool. It got released. Huh? Yeah, students' answer is going to be what? P wave is going to be depolarization of atria. Correct? Yes. Okay. Now, this question. Answer this question. Lot of questions like this come. Okay, lot of questions like this come. Answer this last question and then we will go to the next chapter. I know all of you ran to download your admit card. Yes, it is released. Please explain now. Now I can't explain, dear. Let's complete this now. Answer this. P wave. What is P wave? Depolarization, right, of atria. Q wave is going to be depolarization of ventricle. Okay, QRS. QRS also is there. No, students, wait. QRS is there. 1 second. Depolarization, okay, of complex atria. Beginning of systole. Depolarization, repolarization. Okay, what's the answer? D. Yes, perfect. Okay. So A3, okay, P this, okay. B, Q wave is beginning of systole. QRS is this. They could have given little more clearly the systole thing. Okay. Yes. Yes, exactly. No one will steal it. Why will anyone want the admit card? Okay, students, we will do nervous system. Then we'll do excretion and chemical coordination. Okay, one light chapter, one heavy chapter. Neural control and coordination is actually light chapter. Excretion, if I do now, no, that's all. You people will faint. Okay, ready? Come on. Let's put some fire emojis again. Fast, 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 fast. Put some fire emojis. Yes. Okay, I'll also drink some water. We will start neural control and coordination. Last time you people wanted me to teach the impulse, no? So today I'll teach you the impulse. Okay, I'll teach you the impulse. Hey, what is there to check the? Relax. Admit card will be there only. Okay. 5 minutes break. Take 5 minutes break. I'll sit here only. Take 5 minutes break. Okay. Take 5 minutes break. I'll sit here only. You tell me when you're ready. I'll come. Okay. Yes. 2 minutes break you take. Just drink water. Use restroom and come. I'm here only. Anyone has anything? Okay. Enough, no? Yes. That's my phone. Such an automated phone I have, students. When it got charged, automatically it fell down. Do you people have such phones? No, no. Huh? Yes. Come on. Let's do it. Let's do it. Let's do it. 10 minutes away, yeah? Okay. Yeah, okay. Yeah. Students, if any of you downloaded the admit card, just share it to Basavaraj sir.
Just share it to me or share it to Gopika ma'am?
Yes.
I don't know. Then share it to me only. Otherwise, Gopika has to share it to me then.
Okay, just share.
Yeah, sharing is caring.
Ma'am, are you covering all topics enough for NEET? No, I'm not.
Ma'am is covering only students. It says thumbnail only says 90 marks, right?
Students, every line of NCRT I'm doing. More than this, what to cover?
Exactly.
Huh? I'll have to take encyclopedia and teach you. Chumma, don't ask stupid question
students,
last
just
minute.
Go live, ma'am.
Yeah.
4 p.m.
Okay, come on. Neural control and coordination. 2 minutes break over. Okay, I give you all breaks, lot of breaks. Now come here. Neuron. Okay, all of you know a neuron. No one is there who haven't seen a neuron. Okay, so look here, students. This is the cell body. Okay, this is the cell body. So we can call this whole portion as cell body or cyton. This long portion which you're seeing is the axon, and down it is the axon terminal. Okay, down it is the axon terminal. Got it? So if any of you have downloaded the admit card, just share it with me or Basavaraj sir on Instagram or Telegram. We just want to see it. Okay. So look here, students. Inside this is cytoplasm, nucleus is there, and this is the dendrites. Okay, this is the dendrites. Those like finger-like projections that you're seeing is the dendrites. Okay. Now when you come down, you see a long structure which is called the axon. Now, most important things I'll tell you. This cells you're seeing here is the Schwann cells. Now, many students ask me, ma'am, 2 Schwann cells are there or what? Here and here. Not like that, students. Nothing is there. I don't have anything to show you people. Okay, let's try on this bottle only. Okay. So, students, you study. Admit card, what you will do with? Now, see, like this it is. No, like this. Can you see the rope? Not the rope, the charger. So, when I'm taking like this, I'm showing this side and this side. You understood? It is not like two cells. It is basically this side and this side. Clear? So this is the Schwann cells that I'm showing you. See, if you look like this, you will notice this side also is there, this side also is there. So it's not two Schwann cells, it is one Schwann cell. How we draw it, no? It is seen like that. Okay? Done, done. Thank you, Preeti. Yes. Okay, come on. Now look here. Schwann cells. What is the use of Schwann cells? They are the ones who are going to produce the myelin. Okay, the whitish color structure which is formed is produced by your Schwann cells. Okay. Now students, the places where the axons are going to be exposed. This is a PYQ question. The places where axon is exposed is called as nodes of Runway. What is it? Nodes of Runway. Okay, where the axon is exposed. Where the axon is exposed. Clear? Now, remember, in the cytoplasm, there is something called as Nissel's granule, that is rough endoplasmic reticulum, which has the ability to do protein synthesis, but again, not of all the neurotransmitters, only one type of neurotransmitter. Can you remember all of this? Okay? Oh my god, there are so many news channels in one comment box. Yes, everyone got to know. Relax, students. Okay? Yes, 1 second. Okay. Yes. So look here, all of you. Nissel's granules is capable of doing protein synthesis, but not of all the neurotransmitters. Only one type of neurotransmitters. Now, students, this is the axolemma. That is the cytoplasm of the axon, and the plasma membrane is going to be axolemma, axoplasma, and then you have synaptic bulb. All this you know, no? A normal neuron structure. Okay, normal neuron structure. Now, the synaptic bulb, if I take a magnified image, like if I want to go and see the image, it will have a synaptic vesicle, and inside the synaptic vesicle, there is going to be a synaptic bulb. Inside the bulb, what is going to be there? The neurotransmitters are going to be there, like your acetylcholine, dopamine, etc., etc. Okay, GABA, all these people are going to be present inside that, the neurotransmitters. Clear to all of you? Yes or no? Now, students, neurons are of two types. Depending on if the Schwann cells are making the myelin sheath or not, they are divided into two categories, myelinated and non-myelinated. Myelinated means the ones which have myelin sheath. Non-myelinated means the completely exposed axon. They do not have any myelin sheath. So look here, non-myelinated neuron and myelinated neuron. Okay, myelin sheath act as an insulator. Insulators. Got it? Yes. Okay, now nervous system. Now this I will just like simultaneously I will just tell you. In nervous system, you know CNS and PNS is there. Central nervous system and peripheral nervous system. Now the neurons, okay, the myelinated cells are going to be oligodendrocytes, and in PNS it is going to be Schwann cells. What did I tell? The ones who are going to give myelin sheath, form the myelin sheath, in CNS is going to be called as oligodendrocytes. And the same cells who are going to make this myelin sheath in PNS is the Schwann cells. Clear to all of you? Yes. Now, this I'll come into later. Okay, this I'll come into later. Yeah. Okay, now look here students, the different types of neurons and where they are present. They will ask you the location, they will ask you the match the following of that. Look here, unipolar neuron. That means one axon is only thing that is there. You will not see a dendrite or anything, just one axon is there. This is seen in embryonic stage. Okay, unipolar is seen in embryonic stage. Okay, that is single. Bipolar means two, that is one dendrite, one axon. This is seen in the retina of the eye. Okay, pseudo unipolar. First it acted like it is going to be unipolar. Have you seen some people, first they'll act like they're good students, suddenly they become spammers, like that only. Okay, so they acted like they're going to be unipolar, suddenly they became one axon and one dendrite. So they are called as pseudo unipolar, which is present in the root ganglia of the spinal cord. Okay, ganglion means a group of cell body. Okay, cyton. So root ganglia of the spinal cord. Clear to all of you? So embryonic stage unipolar, retina of the eye bipolar, pseudo unipolar present in the root ganglia of the spinal cord. Easy to remember students, easy to remember. You can easily remember. Yes, here comes the most complicated thing that you have complicated in your head. Okay, action potential. I will teach you this in 10 minutes time. 10 minutes, just listen to me carefully. I'm going to finish teaching you this. Okay? Oh, no Vicks chocolate, students. Can I go and take the Vicks and come? Just give me 1 minute, otherwise my voice will just stop. Okay? 1 minute I want. Okay, I'll just get my bag. Hello, 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 hello. I'm back. Okay, come on. Let's do this. Okay. Students, I don't want any of you to complicate it. Okay, let's keep it simple. Let's just understand the charges. Let's just understand what is there in the extracellular fluid, what is there in the intracellular fluid. Okay, yes. Now, look here. Students, first of all, the neuron is going to be at the resting potential. Now, before I go into that, first let me show you the diagram. Okay. Yeah, I drew this diagram for you people in the break time. Okay, so I can show you a little more clearly. So, in the resting potential, that is, if I take my neuron, okay, if I take a neuron, okay, if I take this portion, okay, if I take this portion, God, yeah, if I take this portion in a magnified way, this is how it is going to look. Okay, this is how it is going to look. Okay, so that means your neuron, okay, your axolemma, it has extracellular fluid and intracellular fluid. Okay, extracellular fluid, students, it is positively charged. Why? Because sodium ions are there. Okay, and in the intracellular fluid there is negative charge. Why? Because of the potassium ion as well as the negatively charged protein ions. Now, what happens whenever a stimulus is there, the signal has to jump. For that, the neurons will go through a depolarization, resting potential. So, this jump is going to happen. For that to happen, they also have specialized, sodium and potassium pump. 1 ATP. If I use 1 ATP, students, that will take 3 sodium ions outside and 2 potassium ions inside. How do you remember? Nokia. N for sodium, O for out, K for potassium, I for inside. Nokia? Remember like that. Okay. So, the potassium pump, with the help of 1 ATP, it will take 3 sodium outside, 2 potassium inside. Clear to all of you? Yes or no? Clear to all of you? So, sodium is outside more sodium is outside the membrane and it is nearly impermeable to sodium. That means the membranes are not permeable to sodium but they are permeable to potassium. Okay, potassium is high and membrane is permeable. Clear to all of you? This is the lines of NCERT. So, just understand that potassium and the negatively charged proteins are making the ICF, intracellular fluid, negative. Okay, mainly it is the negatively charged protein. And on the other side, you have sodium ions, which is making the ECF positive. Clear? This is necessary to keep your neurons polarized. Okay, ionization is the reason for your neurons to be polarized and also react when an action potential is coming. Done? Okay, look here. Now comes the complicated part in your head, but it is not that complicated. Okay, listen to me carefully for next 5 minutes. I have drawn 3 sets. The yellow lines are the sets. Okay, 1 set, 2 set and 3 set. Can you all see that? 1 set, 2 set and 3 set. Okay, 3 set. So, sorry, here 2 set and 3 set. Now look here students, this is how it happens during a resting. Minus 70 MV, A is also polarized, B is also polarized, C is also polarized. That means I have divided it into A, B and C. All the 3 are polarized. Correct? All the 3 are polarized. Correct? Now look here, when a stimulus comes, any stimulus for that matter, you touched a hot object, dog is coming to bite you, any stimulus, admit card, release, any stimulus has come, how does it jump? How does the charges move? Look here students, when the stimulus comes, there is a high chance, okay, there's a high chance that this is the polarized state. Suddenly there will be an influx of sodium. The moment the stimulus comes, there is an influx of sodium, leading to the inner membrane being positive. Can you see? ICF has become positive. And obviously, the outside is going to be negative because all the positive has come in, the outside is going to be negative. Yes or no? Correct? So, understand, when we are in the most positive things in our life, we always get attracted to negative things. When we are very happy, we want to do something like drink alcohol or go out with friends and have a wrong party. So that is because whenever positive is there, positive is always attracted to negative. Correct? So same thing will happen here. This positive charge will get attracted to this negative charge. This positive charge will get attracted to this negative charge. This positive charge will get attracted to this negative, and this positive charge will get attracted to this negative charge. Got it? Now what will happen? So, very simple thing happened. One stimulus came leading to sodium influx. Sodium influx means all the positive charge inside, all the negative charge outside. Suddenly, the next person, that means from A to B, okay, look here students, A to B and B to A, both are getting attracted, okay. Again, A to B, B to A is happening here. After this, current flow will start. That means charges are going to shift. Charges are going to shift because they got attracted. Okay, they got attracted. Now the third step, what is going to happen is, because these people got attracted, you will notice that again, positive inside, positive inside. So, when an action potential happens, 30 to 40 MV, A will go for depolarization. I hope no one has a confusion. You start with doctor. D के बाद R. Okay, D के बाद R. Clear? Huh? Huh? Here, see? Listen to me carefully. Okay? So, here students, what happened? Action potential came, sodium influx happened. So, sodium got inside, positive charge, outside negative charge, positive getting attracted to negative. That is leading to A stage. This stage becoming depolarized. Again, B and C remains polarized. B and C remains polarized. Now, since these people were attracted and there is a current flow, definitely B is going to change the charges, right? If you see in B, again sodium influx have happened, positive inside, negative outside. So now if you look here, A has gone for repolarization, B has gone for depolarization, C is only polarized. Are you able to understand this? Are you able to understand this? Try to understand it aram se, okay? Don't break your head. Don't break your head. Got it? So, this fellow was in this situation. He's trying to come back to polarization. So, that will be, here you will see depolarization and repolarization is happening. Okay? Done, done, done. So, resting potential, all three are polarized. Action potential, A is getting depolarized because there is an influx of sodium. So, positive charges have come in, negative charges are going out. Correct? B is polarized, C is polarized. Again, what is going to happen? A is trying to come back to polarization, to the resting potential. Then this will go for repolarization. This will move to depolarization. This will be polarized. Again, here to here, same thing will happen. So, polarized, repolarized and depolarized. Like this, it is going to continue. Okay? Yes. Okay. Got it? Okay. Got it? Done or not? Okay. 1 second. Done. Okay. So this is what is happening. Okay. Understood? Done. So 1 ATP can throw 3. So students, here remember, if I want to go to this state, I have to make sure that I bring back the potassium ions and I take back the sodium ions, right? So that time energy will be utilized to take 3 sodium and throw it out and put 2 potassium back in. Okay. So look here. This is your axolemma, this is your axoplasma. So there are sodium channels, there are potassium channels. Then with energy, you have to use sodium potassium pump. Okay, sodium potassium pump. Remember, less sodium influx will be there, more potassium ions will go out because the membrane is permeable to potassium ions. 1 ATP, 3 sodium, 2 potassium. Clear to all of you? Everything we have told is what I believe. Okay, that's it. Okay. Now let's see this image. Okay, let's see this image to make you understand better. Depolarization in neuron. Okay, let's try to understand. What is that sound, yeah? Okay, let's try to understand this, students. What is going to happen? Depolarization. No, what you will have noticed. Can you see that the sodium has started coming in? Sodium and potassium. See, can you see sodium has started coming in? Okay, sodium has started coming in. So here you will notice sodium potassium transporters. Sodium has started coming in. Okay. Now potassium also after some time will start going out. That day I told you about that sibling fight. No, first sodium is going to come, fight nicely, and then he will stop. Then potassium will get angry. Potassium will come inside, start fighting. Then we will use energy to throw these 2 people outside. That is how the entire situation is going to come in rest. Okay, entire situation come to rest. Okay, done. Done. Okay, charges also I have mentioned here, same thing which I drew only. Okay, same thing which I do only. Repolarization, you can see how the ions are going out. Okay, how we are bringing back the ions. Okay, now students, chemical synapses. Chemical synapses. Okay, chemical synapses, understand there is electrical and chemical. Electrical means the neurons are going to be very close to each other. Axon and dendrite. Axon and dendrite. They are going to be close to each other, right? So electrical impulse, it will just get transmitted. But when there is a gap between the neurons, there is going to be chemical synapses. Okay, chemical synapses. And look here, so electrical synapses, students, no neurotransmitters. Chemical synapses, neurotransmitters are present. Synaptic cleft gaps are small in electrical, more in chemical. Very rare, most common, fast, slow. Okay, here look here, how do the neurons get, how do the exchange happen? This is post-synaptic, this is pre-synaptic, post-synaptic and synaptic cleft. Okay, this is pre, post and this my face is the space, synaptic cleft. Here, he will have at the axon ending, he will have neurotransmitters, he will have the receptors to accept the neurotransmitters, he will open the synaptic vesicle, neurotransmitters will get transferred, he will accept it with the help of receptors and the actions will be continued. Clear? So, impulse, okay, impulse, you will notice that there is an action potential created. So, impulse has come, immediately when action potential, the gates are started to open, calcium will get released and the synaptics will open, the synaptic vesicles will open, neurotransmitters will get transferred, they will be taken up by the receptors, binding will happen. I have written all the steps. Binding will happen and this is how your chemical synapse is going to get carried away. Chemical synapse is going to get carried away. Now let's look here also, students. See, first a nerve impulse have to come. Okay, first a nerve impulse have to come. Second, what will happen? Your calcium gated channels are going to open. Okay, calcium gated channels have to open. That is your second step. As they open, you will notice that the neurotransmitters will start getting released. Okay, neurotransmitters will start getting released. That is the third step. Okay, that is the third step. Neurotransmitters will get released. Fourth step will be receptors accepting the neurotransmitters, and the postsynaptic potential and the nerve impulse will continue. Nerve impulse will continue. Okay, got it? Okay, done. Done. Now, students, brain. Last day also I taught you brain. So, brain basically is divided into forebrain, midbrain and hindbrain. Okay, forebrain, midbrain and hindbrain. Okay. Now, forebrain is divided into olfactory lobes, cerebrum, diencephalon. Okay, diencephalon. Now, in this diencephalon, please remember epithalamus, thalamus and hypothalamus. In that, thalamus is the region from where all the relay happens. That means, now let's say I am taking this thing here. Okay, this, and I am giving to this person. Vishal. Okay, Vishal is asking for support and all. Vishal is standing here. I am running from here and giving this to Vishal. Now, Vishal has to take that and run to someone else. So, this is a relay. Basically, bringing the information from here to here, taking the information from here to here is relay. So, all the relay activities happen in the thalamus. Okay, in the thalamus. Clear? Clear? Okay, very good, very good. Now, students, midbrain you have corpora quadrigemina and you have cerebral aqueduct. Hindbrain you have cerebellum, pons and medulla oblongata. All this you have studied in EVS. So, you know, we are not going into the depth of that. Okay. Now, cranium bones already done. Here, meninges. Meninges are important for you people. Meninges, clear? Okay, meninges, students. Meninges, you remember DAP and PAD. PAD and DAP. DAP, DAP, PAD. PAD, PAD, DAP. Okay, so I'll tell you why is that. See, if you look at the entire brain, to protect your brain, there are three meninges that are given. Three meninges that are given. Okay, three meninges are going to be. Yes. So, first one is going to be called the dura mater. Okay. First one is going to be called the dura mater. Second one is going to be called the arachnoid. Third one is going to be called the pie mater. That is what, students? Dura mater. Okay. Arachnoid, pie mater. DAP. DAP. Outside to inside, DAP. Okay. Outside to inside, DAP. Okay. Inside to outside, PAD. Inside to outside, PAD. Clear to all of you? So, here what is going to happen, students? Dura mater is the first one. Between the dura mater and the arachnoid, you have subdural space. Subdural space. Okay? Arachnoid and pie mater, subarachnoid space. Subarachnoid space. Okay? So, in to out PAD, out to in DAP. In to out PAD, out to in DAP. Clear? Yes or no? Yes or no? Okay. This is the protective covering. Okay. So, very simple. Between dura mater and arachnoid, subdural space. You take the name of the first fellow. Arachnoid and pie mater, you take the first name. Subarachnoid space. These are meninges. One layer after the one layer after one layer. In that, they are to just to protect our cranium. Okay. Okay. Next one. Next one. Okay, yes. Epithalamus, thalamus and hypothalamus. So, students, this you have to remember, it is a PYQ question. Okay. Who helps with all the relay. Okay, that is very, very important. Very, very important. People are, you know, making me like those. Yes. Come on. Come on. Huh. So, Epithalamus is present in the roof of, this also will come as a question, okay, location. Where is it present? In the roof of the diencephalon, and hypothalamus in the floor of the diencephalon, okay, floor of the diencephalon. It forms a stalk, all that is not, this is very, 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 very important. Major coordinating center for sensory and motor signaling. That is why we tell for all the relay, okay, for all the relay activities, this is what is going to happen. Got it? Clear? Good. Good. How many ever accounts you come, we'll remove you. Okay. Yes. Okay. So epithalamus encloses the pineal gland. Thalamus as sensory. See, last time also I told you PYQ. Very, very important. Okay. Very important. Okay. Neuron passes through the thalamus. Hypothalamus glands hypothalamic hormone. Students, this is very, very important for all of you. They will ask you the question, who controls the thermoregulation? Okay, thermoregulation, hunger and thirst center. This has come as a PYQ, so please remember this. Okay, please remember this. And amygdala and hippocampus are the ones who are responsible for all the sexual behaviors and emotions, and they are present in the lymphic system, or we call that the lymphic lobe. Lymphic lobe. Clear? People are not listening. Come on. Now the midbrain. Midbrain has two parts. Corpora quadrigemina, okay, corpora quadrigemina and cerebral aqueduct. Corpora quadrigemina is made up of colliculi, that is superior colliculi and inferior colliculi. Superior colliculi is going to help with the visual reflex. Inferior colliculi is going to help with the auditory reflexes. Okay, clear to all of you? Yes or no? Yes. Done, done, done. Done, done, done. Okay, good, good. So, brain is clear to all of you. I've told you which are the most important things, okay? Yes, so here also students, all this you please remember, this image is very important because they will ask you hypothalamus controls the body, temperature, hunger, fatigue and sleep. Amygdala, which is helping with the memory, decision making and emotional response. Thalamus, regulation of sleep, consciousness and alertness. With that, you also remember the relay thing. Okay, relay thing. And here students, basal ganglia, no need. Okay, this no need. Hippocampus, memory and navigation. Memory and navigation. Done? Okay. Students, I am done with nervous system. Okay, do you want me to do chemical coordination also? Or shall I send sir? Shall I send sir? I'll just check with sir. I don't know if sir is live. Is sir live? I will teach you the entire chemical coordination with this one tabular column. Okay? With one tabular column. One second. Did sir go live, students? Sir went live already. Okay, shall I send sir then? If people want a break from human physiology. Or we will do of chemical coordination also, no? But don't tell sir, okay? 5 minutes break, huh? Okay, chalo. 5 minutes break ke baad I'll do. Okay, 5 minutes break will be give. I've added the notes here, okay? Like detailed notes I've added, but I will teach you only from that box because chemical coordination, only questions that are going to come is going to be about the neurons. So this box is more than enough because see, this box, students, is from Tattva. This box has hormones and the function. So I'll teach you all this. I'll teach you all this, okay? This many box we have. This will take us hardly 20 minutes. 20 minutes we will finish it. Okay, 20 minutes we will finish it. So I'll give you a break now and I will come back. I'll only come back, and after that last chapter we have to do is excretion. Can you believe? One more chapter we will be done with human physiology. What is the time now, students? 4:23. Okay. 4:35. Enough? Break? Okay, break. Let's make 1.2 likes. Okay, 1.2k likes. Okay, yes. Break is here. 5 minutes if it's enough, then I'll come back. I'll wait here only. Okay, 5 minutes if it's enough means then by 32 you can come back. Okay, 32. Almost 10 minutes break. Excretion, yeah, only excretion left after chemical coordination. Okay, so I told you I will not waste your time. I told you that if I am taking a 5 or 6 hours, I'll complete all the chapters. Okay, I will not waste your time taking 3 hours, 4 hours for each chapter, because this is not the right time for that. How many other people might tell that yes, it is the time to do each chapter in detail? No. Detail means this much detail is enough, where we are going through the surface level, at the same time stressing on the topics which are PYQ necessary. Okay, got it? Students, your life, students, we cannot play with your life, right? Your time is more valuable for me than my time. This is the second time my phone is falling. Did you people put nazar on my phone? Huh? It's falling everywhere. In my watch now it is 4:32. Oh, रात से ज़ेनो London clock is. Come on. Ready? Voice is audible. Come on, let's start with this. Okay. I think first I will teach you once about the infundibulum because questions come from there. And after that, we will go into this. Okay. Okay. Students, look here. So hypothalamus is like the principal. I told you always, no? Hypothalamus is like the principal and pituitary is like the, who is that? What do you call for them, students? Principal? Headmasters. Okay, headmaster or HOD. Okay. So hormones can be of two types, releasing or inhibitory. Okay. They can be releasing or they can be inhibitory. Now, hypothalamus has to give its secretions to pituitary. For that, they are going to take the help of neurosecretory cells. I hope all of you got to know this. Neurosecretory cells are going to give their secretions. But understand, pituitary mainly has three lobes. In that, two lobes are most highlighted, anterior lobe and posterior lobe. So, anterior lobe will be like, okay brother, no problem, you give me the secretion. So, he'll be like, okay, I do not want to come there and give the secretions. Can I give it with the help of an hypophyseal portal system? Like a portal system. Porter means what? A person who carries our luggage from the railway station till the train. Okay, portal system. So, hypophyseal portal system. Anterior lobe will be like, okay, fine, no problem. You give me the secretions like that. But posterior lobe is like the boss. He is like, excuse me, porter, no, you directly come and give me the secretion. So, hypothalamus will be like, okay, neurosecretory cells, you please go and give the secretions to the posterior. Okay. So that is why you are seeing these two neurosecretory cells directly into the posterior and you are seeing these two neurosecretory cells giving it to the hypophyseal portal vein. Now, these anterior lobe, intermediate lobe and the posterior lobe have different names. Okay, different names. What are the different names, students? Look here. Anterior pituitary can be called as adenohypophysis. Also, it can be called as pars distalis. Pars distalis. Okay. So, you remember like this, anterior pituitary called as distalis, AD. Okay, AD you remember. And posterior pituitary is called neurohypophysis. With that only some connection you have with pars nervosa. Pars nervosa. So, PN you can remember. Okay, posterior pituitary N. So, ADPN. ADPN. And students, there is an intermediate lobe which is not that prominent, but we still call it the intermediate lobe. Okay. So, posterior pituitary directly regulation of the hypothalamus. That means hypothalamus have to directly come and tell her, please take my secretions and store it. Okay. So, it has to directly give the neurosecretory cells job to go and secrete it in the posterior lobe. On the other side, you have an hypophyseal portal system. And between the hypothalamus and the pituitary, there should be a stalk, no, to connect. That stalk is called the infundibulum or a stalk-like structure. Clear to all of you? Clear to all of you? Spelling you see. Heart attack. Okay. It's okay. Come on. Okay, look here. The structure, little more clear. Hypothalamus. Okay. The infundibulum, the stalk. You have your pituitary. You have pars distalis, pars intermedia, which we together call adenohypophysis, or even for anterior lobe, mainly we tell adenohypophysis. And students, infundibulum stalk, pars nervosa or neurohypophysis, and this is the entire infundibulum. Clear? Clear? Okay. So, these are the neurosecretory cells which are like the nuclei. Okay. Neurosecretory cells, you can see how they are adding the secretions into the hypophyseal portal vein and how they are bringing it to the anterior pituitary. Clear to all of you? Okay. Got it. Got it. Now, can we go further? Can we go further? Students, this box also is a beautiful box. You can see which are the things that are released by hypothalamus, which is going to the anterior lobe, which is going to the posterior lobe. Where is the target organ? Target organ, because endocrine and exocrine gland, you know, exocrine have ducts. It will directly add the secretions into the ducts. Endocrine glands on the other side, it will give it to the blood. Blood will give it to the target organs. Okay? Yes? So, you can see how GnRH releasing, R for releasing, okay? CRH, TRH, PRH, GHRH is all going here to the adenohypophysis. From there, that hormones are going to the target organs. Clear? Got it? Done. Okay, now I will teach you the boxes, okay? So, the hormones, okay? First one we have adrenocorticotropic releasing hormone, that is ARCH, okay? Okay, sorry, CH, okay? ADCH stimulates anterior lobe to secrete ACTH, okay? So, this one, that is adrenocorticotropic hormone releasing hormone, helps in releasing ACTH, okay? Then we have growth hormone releasing hormone, okay? GHRH, okay, or GHRH you can write, stimulates anterior lobe to release the growth hormone. Clear? Now, prolactin releasing hormone, PRH, prolactin releasing hormone will stimulate anterior pituitary to secrete prolactin. Prolactin is going to do what, students? Prolactin is going to do what? Please tell me. Now, MSH releasing hormone stimulates the intermediate lobe. This is the only one that is secreted in the intermediate lobe. Okay, the only one that is secreted in the intermediate lobe to secrete melanocyte. Okay, melanocyte, yes, milk secretion, very good, melanocyte stimulating hormone. Now, thyrotropico releasing hormone, TRH, stimulates the anterior pituitary to release thyroid stimulating hormone. Okay, gonadotropin releasing hormone stimulates the anterior lobe to secrete LH and FSH. Okay, prolactin inhibitory, students, inhibitory also is there, that will inhibit the secretion of prolactin from the anterior pituitary. Okay, again MSH inhibitory also is there, that is which has to inhibit the thing. Okay, inhibit the melan. Now, if we go here, okay, acts on melanocytes. So, melanocyte stimulating hormone acts on melanocytes and regulates skin pigmentation. So, we have inhibitory as well as releasing or stimulating, because otherwise the skin shade will change. Okay. Now, oxytocin, students, remember these two are present in the posterior lobe. So, the question comes, okay, these two are present in the posterior lobe or neurohypophysis, and these two questions come for you. Somatostatin is your growth hormone inhibiting one, okay. So, oxytocin and vasopressin is the one that is present in the posterior lobe, stored and secreted in the posterior lobe, stimulates the contraction of the uterus and of the at the childbirth, and also stimulates the milk ejection, not production, okay. Vasopressin, students, otherwise called your ADH, antidiuretic hormone, stimulates reabsorption of water and electrolytes from distal tubules of the kidney. Okay, possible to remember? So, these two you can remember easily, oxytocin and vasopressin. The rest we will revise once again, okay. Now, pineal gland, students, melatonin. Pineal gland is the gland that does not work for any of us because it is completely confused. That is the one who regulates our sleep and wake up schedule, our circadian rhythm and biological rhythm, okay, biological clock. Maintains the sleep and wake up cycle, also the body temperature. You have to make sure that your pineal gland is working properly up to May 4th. Okay, especially on May 3rd, this has to work well. Otherwise, your brain will also not work according to this. Okay, clear? Now, thymus gland, all of you know thymosin. This is the one who is going to help us with immunity. That is, helps in differentiation of the T lymphocytes and promotes the production of antibodies. That is why we tell cell mediated immunity does not form antibodies but helps in the promotion, okay, production of antibodies. Yes. Now, thyroid gland, students, you have thyrocalcitonin and thyroxine, decreases the calcium level in the blood, and this fellow regulates the BMR, basal metabolic rate, and metabolism of carbohydrate, fats, proteins, support RBC formation. PYQ question. Okay, they have asked which gland supports RBC formation. Your answer should be thyroxine. Okay, thyroxine. Shall we revise once again? Shall we revise once again? Okay, let's go. First, we will revise this page because this page is easy. Then we will come to the last page. So, students, we start from the posterior lobe. So, in the posterior lobe, which are the two hormones that will be secreted? Come on, all of you, quickly tell me in the charts. Let's have a quick rapid fire. Okay, students, I missed off pituitary only. I skipped off to this one. It's okay. We will come back to this. Yes, tell me the two hormones that are secreted. Quick, we will have a rapid fire. Excellent. Oxy and vaso. That is oxytocin and vasopressin, which are secreted in the posterior lobe, okay, from the hypothalamus. Oxytocin will do what? These people are going to help with milk ejection, and they're also going to help Yes, very good. Very good. ADH. ADH, where did I teach you, da? See what I've taught you? You're not this. What I've taught you? ADH. Okay. Sorry. Vasopressin. Okay, in my head, I read it as ACTH. Okay. Oxytocin stimulates contraction of uterus at the time of childbirth and also stimulates milk ejection. Vasopressins will do reabsorption of water and, electrolytes. Pineal gland, students, melatonin. Okay. Melanocyte is melanin. Melatonin, who controls the sleep and wake up schedule or the biological clock. Okay. Then we have thymosin. Thymosin, that is, thymus will release thymosin, which helps the T lymphocytes differentiation and also helps in the promoting the production of antibodies. Okay. Then you have thyroid gland. Two things are there: thyrocalcitonin and thyroxine. Thyrocalcitonin reduces the calcium level. Thyroxine, BMR, body temperature. Then it also helps with the fat, carbohydrate, protein breakdown, and also helps with RBC production. Okay, RBC production. Clear? Clear? Yes or no? Now we will go to pituitary. So, students, pituitary, again, target oriented, it has to reach. Growth hormone stimulates the growth of the body. Growth hormone. Prolactin. So, whatever you studied here, whatever the hypothalamus is, stimulating the functions, you're going to study here in detail. Okay. So, prolactin, students, growth of the mammary glands and formation of the milk. Thyroid stimulating hormone, TSH, synthesizes and secretion of the thyroid hormones. Adrenocortico, that is ACTH, synthesizes and secretion of the steroid hormone. Last minute you revise once, okay, you will be able to remember. Again, gonadotropin hormone, luteinizing hormone in males, LH stimulates synthesis and secretion of androgen, whereas FSH regulates spermatogenesis. Follicular stimulating hormone in females, LH induces ovulation, whereas FSH is responsible for the growth and development of ovarian follicles. This you write as such from reproduction chapter. Okay, as such from reproduction chapter you can write it. So in this chapter, there's nothing to teach. Like it's like just given there, just like just like brutal truth. Okay, no news channels can add on anything. It is like, okay, this is the truth. So there's nothing to teach here. All you have to do is, it's like a chapter that you have to mug up. Okay. Now thyroid, this we already understood. Parathyroid, students, parathyroid is present right behind the thyroid. Okay. This is your thyroid gland. Okay, this is your thyroid gland. Right behind the thyroid gland, you will see like this parathyroid hormones. Okay, increases the calcium levels in the blood. And adrenal gland, that is adrenal cortex and adrenal medulla. Cortex, you have mineralocorticoid, glucocorticoid and sex corticoid. Maintains the sodium potassium levels, balances. That's why we do the RAS mechanism, aldosterone. Glucocorticoid, that is your cortisol, which actually works when we are stressed. Okay, when we are stressed. Haircare routine, definitely after NEET. Okay, glucocorticoid, students, cortisol stimulates glyconeogenesis, lipolysis and proteolysis, and this is the fellow who gets released when we are stressed. Okay, we get cortisol face, cortisol belly, all that we get. Okay, yes, very good. Hypercalcemic hormone is PTH. Okay, that is your parathyroid hormone, who is going to increase the calcium level. and to reduce it, we have our thyrocalcitonin. Okay, they both are going to work anthropogenic, not anthropogenic students, antagonistic in nature. Antagonistic in nature. Okay, these two are going to work antagonistic in nature. Stimulate the growth of pubic hair, axial hair and facial hairs during puberty. Sex corticoid. Adrenal medulla, adrenaline and non-adrenaline. Okay, that is the fight flight hormone. Fight hormone. Okay, yes. Rata's hair, that's why students, Rata's hair is like this. Have you seen Rata's hair? Like this, like cockroach antenna. Because he's drinking cockroach juice every day. This is how his hair is. Got it? Yeah. Done. Okay. Like antenna it is. Okay. Moving forward. Pancreas students, insulin and, okay, insulin and glucagon. Insulin, what they are going to do? Ma'am, obviously, ma'am, they are going to reduce the glucose level. What is glucagon going to do? Ma'am, they are going to do increasing the glucose level. How, ma'am? By glyconeolysis. Okay, glycogenolysis, that is forming the, okay, breaking down the, stimulating the glycogenolysis, resulting in increased blood glucose level. That fellow is going to reduce the glucose. Okay? Got it? Got it, all of you? Epinephrine, non-epinephrine, here only. Okay, here, adrenal. Okay, adrenal. Catecholamines. Now, students here, wait, I'll tell you insulin and I've made a, I took a flow chart. Yeah. Mechanism of insulin action. Okay, mechanism of insulin action. Clear? Look here, students, what is going to happen? So, when there is an increased glucose level, the pancreas will understand, okay, hypoglycemia. The pancreas will stimulate insulin. Okay, insulin stimulates the glucose uptake by cells. It will tell the cells that you please utilize the glucose. This fellow is continuously eating gulab jamun and everything and he is not stopping. So the cells will be like, okay, fine, I will take it. Then some of the pancreatic beta cells will release insulin. This you understood. Insulin will stimulate glycogenesis. Okay, glycogenesis means to break down the glucose, okay, and convert it. So in the liver, so the liver will convert all the glucose to glycogen and store it. Okay, store it. Now, automatically the glucose will come down. Automatically the glucose will come down. Now, when there is a glucagon will do what? Increase the glucose level. So that is when, students, when we have decreased glucose level, hypoglycemia will happen. That time the pancreas again will try to do, it will tell the alpha cells. This is beta, this is alpha, delta cells also are there. Pancreas has 3 cells, okay, remember. Alpha is glucagon, beta is insulin, delta is somatostatin. So, sorry students, something and all is problematic, okay. Statins, okay, somatostatins, that is again inhibitory, growth inhibitory ones, okay. So, it will tell the alpha cells release glucagon. What glucagon will do? It will start producing neogenesis, new glucose, okay. It will stimulate gluconeogenesis and glucogenolysis, that means to break down the glycogen to glucose and also produce more glucose and release the glucose into plasma, and automatically plasma will come to normal levels. Is this clear to all of you? Is this clear? It is very easy, no? One will break down the glucose and convert it to glycogen. One will break down the glycogen and release it to glucose. That's all. That's all. Okay, that is this 2 job. Now here gonad, students, this is somatostatins which are by the delta cell, okay. Delta cell, somatostatins inhibits the secretion of insulin and glucagon. You people didn't leave admit card, huh? People didn't leave about admit card. 1 second. Yes, clear? Students, this fellow, okay, this somatostatins, okay, they do not only inhibit that, they also inhibit, okay, they are generally inhibiting, they also inhibit hypothalamic one, that is the growth hormone as well. Okay, they also inhibit the growth hormone. Now androgens, what will they do? They stimulate spermatogenesis, regulate growth of the secondary sexual character. Estrogen will do the same thing, stimulate the development of secondary sexual character. Progesterone maintains the pregnancy. Will you be able to revise this? Will you be able to revise this? Yes or no? Yes or no? Got it? Students, that's all here, okay? And here also I have put about the parathyroid hormone, what they are going to do. PTH is going to increase the calcium levels by removing the calcium from the bones, okay, and giving it to the blood vessels. At the same time, you have your thyrocalcitonin who will decrease the calcium levels, okay. So this PTH is going to reabsorb everything from the kidney, okay, and they are going to take as much as calcium possible and increase the calcium levels. On the other side, that fellow will decrease the calcium levels, okay, decrease the calcium levels. Do not panic, your admit card will take some more time, do not panic, okay, do not panic. 2025. Okay, I'll just call Baswa sir, stay here only. Okay, that is your break. By the time I go and call, no, that's your break. So I'll come back again for excretion, and by then we will be done with human physiology. Starting of the day in the morning, we started with breathing and we finished locomotion. Then we did body fluids and circulation and neurons, and now we also did chemical coordination. Now one more chapter, excretion, which will take me 1 hour or 45 minutes. We will be done with that as well. Okay. Good job for all of you who stayed back. Really proud of all of you. Proud of all of you. Okay. Stay till the end. Don't give up.
Students, is downloading. It will take some time. Students, can you hear me, all of you? Students, quickly tell me in the chat right now if I'm audible to all of you students. Quickly tell me in the chat right now if I'm audible to all of you students. Quickly in the chat, all of you. If I'm audible, and if I'm not audible, also tell me in the chat. Quickly, all of you. Quickly tell me in the chat right now if I'm audible to all of you. Yes. Ouch. Hello, 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 students. Quickly in the chat, all of you. Yes, yes, yes. Downloading is done. Perfect, students. Perfect. Students, how were the human physiology classes, students? Hello, 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 hello. How was the human physiology class, students? Yes, yes, yes. All fine? How was the human physiology classes? All good? All of you? Yes. All of you students, good evening, good evening, good evening, all of you, good evening. Yes, good evening. From good morning, it became good evening already, students. Do you realize? By the time this session gets over, by the time the entire session gets over, it's going to be good night. So by the end of today's session, we'll say good night and we'll go, all of us. Okay. So let's just import in the file that might happen and we'll start, students. We'll start. Now, students, can all of you tell me in the chat right now what is your preparation level for respiration plans chapter? What is your confidence level for respiration plans, students? What is your confidence level for respiration plans? Yes, yes, yes. SGS category, how much score? 450 is not enough, students. 450 is not enough. Vedant, 450 is not enough. You have to go above 500. All of you students, how is your preparation? How is your confidence level for respiration in plants, students? Okay. All of you. What is your confidence level for? Oh, finally, students. Finally, students. Okay. 0 to 5. What? Students, this is very easy chapter. All of you. Okay. So, students, do not worry at all. I will teach you right now glycolysis. I will teach you entire Krebs cycle. I will teach you the electron transport chain and also the remaining part of the chapter. So, it becomes 5 out of 5 all of you students. It becomes 5 out of 5 for all of you. Minimum marks to get into private college, students. For private college, all you need to do is qualify NEET. 50% of marks. 50% is enough for that. 50% is more than enough. Okay, 0.5 students. I am dumb and students. Plant physiology is the most beautiful thing. Okay, students, let's get warm up now. Let's warm up. All of you ready? Ready. Now, students, I want all of you to tell me in the chat right now. What is the difference between breathing and respiration, all of you? What is the difference between breathing and respiration? Let's say, okay, all of you. Now, is this breathing or respiration? Inhaling and exhaling, is that breathing or respiration? Students, inhaling and exhaling, is that breathing or respiration? You tell me, sir, what a joke. It is nothing but breathing. It is breathing because breathing is a physical process. Breathing is a physical process which we can control to some extent. We can hold the breath. If you do hold the breath for a very long time, RIP in that case. But we can maintain, we can control this to some extent. Okay. And it happens because of the nostrils. It happens because of the lungs and everything. Now, what about respiration, students? Respiration. Can we control respiration? Absolutely not. You cannot. Can we see respiration? No, you cannot see respiration, students. You cannot. Can we? What do you say, students? Where do you think the respiration takes place? Do you think respiration takes place in the case of your brain, lungs, kidney? Where do you think it is, students? Where do you think it takes place? Where do you think the entire process of respiration takes place. The entire process of respiration takes place inside a cell. It takes place inside a cell. That is where it happens. So it is a much more chemical process. It is much more of a chemical process. Respiration is more of a chemical process rather than a physical process. Rather than a physical process. That is going to be respiration. Okay. That is going to be, students, respiration. Okay. Students, give me a second. If this starts to work, I can switch my slides to respiration. Okay, students. Okay. Now, I told you, students, breathing is a physical process. Respiration is a chemical process right here. Respiration is going to be a chemical process right here. Purpose is to intake of CO2, intake of O2, release of CO2. Energy production, control, no control. It can be controlled, it cannot be controlled. Energy is not produced here, energy is produced here. So that is the basic difference between your breathing and respiration. Breathing as well as respiration. Now, students, a simple question to all of you. Do you think respiration takes place in the case of unicellular organisms? Tell me yes or no in the chat, all of you. Do you think the entire process of respiration, right, happens in the case of unicellular organisms? It does happen. It does happen because they also need energy. Without energy, your amoeba, bacteria, all they cannot survive. So in those organisms also, the process of respiration takes place. Respiration has to happen. Okay. Respiration, it has to happen. Okay. Now, students, understand what exactly happens. Understand, students, what exactly is happening in the case of respiration, all of you. Now, let's say this is one cell here. This is a cell right now. Now, inside this cell, oxygen is able to enter. If it is in the case of plants, it is produced inside itself. Okay. Oxygen is entering here. This oxygen is acting on the food here. That is, it is making the food undergo oxidation here. Yes, it is making the food undergo oxidation by breaking the carbon bonds here. And once the food undergoes oxidation, it will release energy, and this energy is so massive. This energy cannot be released as such. This entire energy has to be stored in the form of ATP, adenosine triphosphate. I'll repeat again, students. First, initially we have O2. The oxygen acts on the food here. The food undergoes oxidation here. Students, again, can anyone tell me what is oxidation, students? Oxidation is loss of electrons or gain of students, what? Gain of oxygen. Okay. It can be loss of protons, loss of electrons. Loss of protons or loss of electrons can happen here. It undergoes oxidation and energy is produced. Energy is stored in the form of ATP here. Now, students, for now, all of you need to remember that it is a catabolism, because as we go towards the end of the chapter, I will teach you a word called as amphiboic pathway. But right now you remember, students, it is a catabolic reaction. It is exothermic in reaction because energy is released here, and it is a physico-chemical reaction. It is a physico-chemical reaction, students. It is going to be a physical-chemical reaction. So it is going to, food is undergoing oxidation here. That is biological oxidation of organic molecules. So organic molecules are undergoing oxidation. That's all. That's all, students. Now, what exactly is this food? The food which it say here, right? The food here, students, I told you, the oxygen is acting on the food. So what exactly is this food, students? Is it chapati? Students, is it chapati, students? Students, tell me, students, is it a chapati? It can be chapati. It is going to be your biryani. It is going to be curd rice. Yes, it can be curd rice. It can be paneer. It can be proteins, or it can be any type of biomolecule. It can be any type of biomolecule here. This type of food here. So it can be carbohydrates, it can be fat, it can be proteins, or it can be organic acids. So all of these food items can provide energy. They can provide energy. As respect to water, hormones and minerals, they do not provide energy. No, they do not provide any type of energy here. So energy is mainly provided by the following respiratory substrate. The respiratory substrate is nothing but the food item here. The food item is called as respiratory substance. Till here, any doubt? Yes, sambar is very nice. Even I like sambar. I like my spicy sambar. Okay, students. Till here, any doubt, all of you? Students, till here, any doubt, all of you? Favorite food item? Okay. Till here, there is no doubt. Okay. So here we have all of these are called as respiratory substrates which can undergo oxidation. All of them can undergo oxidation right now. Oxidation. Okay, now students. Here we have an ATP molecule now. How does it work? Students, understand how does it work? Let's say we have an ADP molecule here. Let's say we have an ADP molecule. Okay, phosphate here. Let's say we have an ADP molecule right now. ADP. Now what happens, students? This is ADP molecule. ADP. Okay, ADP molecule. So let's say I took a protein bar and I ate the protein bar. So when the protein bar is going inside my body, the food is undergoing oxidation. Once the food undergoes oxidation, it will release all the energy. Now, using that energy, using the energy, we can make something called as ATP. Now, that is here, students. All of you can notice here very well. All of you can notice here is that initially I had 2 phosphate. Now I am taking one more phosphate and I am adding it here, students. So all of you tell me in the chat right now, can we add 2 things without energy? For example, let's say I have one paper here and I have one paper here. How do I stick to papers? I need to add gum or glue. Now gum or glue here is the energy. So using the energy here, we can bind this phosphate to this one and ADP becomes ATP. ADP becomes ATP. So if I ask you a question right now, where is all the energy stored? All the energy is actually stored in the bond here. All the energy, students, is stored in the bond here. Now you tell me in the chat right now. Let's say I ate the protein bar. After eating the protein bar, students, I am going for running now. I am going for running or I am going for some hiking or I am doing any type of your activity. Okay. That time I need more energy. My body requires more energy. Or plant requires more energy suddenly. Let's say the plant wants to go for jogging. Not possible, but I'm telling you. Let's say a plant wants to go for jogging. Okay. The plant is going for jogging. It needs more energy now. It needs more energy. From where do you think the energy will come? What will happen now, students, is that this ATP molecule, right? This ATP molecule right here, this bond will be broken right now. This complete bond will be broken. So all of us know the energy is present in the bond. So all we need to do is break the bond. So we break this bond and release the phosphate as such, and we're left with what? ADP. So what is actually happening here, students, is that first we have a ADP molecule, adenosine diphosphate. We convert this to ATP. How, students? By using the energy. Okay. Now, where is this energy coming from, students? This energy is coming from where, students? Energy is coming from food which we eat. Okay. That's how we get ATP molecule. And in this ATP molecule, we have 3 phosphate like this. Now, let's say after jogging, I need energy. What do I do, students? Here is that we take this ATP molecule. Students, which has 3 phosphate here. And what do we do is we break this bond. Katak, dogon. Okay. And what do we get, students? We get something called as ADP plus energy. This is a simple concept how energy transfer happens inside our body. This is what happens in our body, how energy is derived. Because when we eat the food, right, all the energy cannot be released as such. It will become waste. Imagine if you make one hole, all the energy will go away. Not very efficient. So what do we do in our bodies? We take the energy, store in the form of ATP. Students. Now what is the difference between ATP as well as NADPH? Understand. Now let's say students, we will take Madhu. Okay. Now Madhu is going to, let's say one shop. Madhu goes to a shop to buy one dozen egg. Now to buy 1 dozen egg, students, how do you think he'll do the payment? He will use Google Pay, he will use Phone Pay, he will use Paytm, UPI basically, or he will give cash. To buy 1 dozen egg, he'll give 100 rupees. But would you tell me in the chat right now, is it practical? Is it practical to give, let's say, I will write a check for 100 rupees. 100 rupees check. And I will give this 100 rupees check to the person there sitting from them. What will he do? He will take the check, throw it at your face. He will take the check and throw it at your face again. Why? Because he doesn't understand the check, because he doesn't want check here. Because if he has to give this 100 rupees, if he wants this 100 rupees back, he has to take the check, go to the bank, give the check to the bank, and then they'll give you the money. So basically, students, this is the small concept of ATP and NADPH. ATP is like money. Direct transaction can happen with the help of ATP. You can give the money, you can get the eggs. But this check you cannot use directly. The NADPH and FADH cannot be used directly. We cannot use them directly. So what we need to do here is that we have to take the check, go to the bank and then get the money. Similarly, students, this NADH and FADH, they cannot be used. They cannot be used as the energy source. They cannot be used. So what do we need to do? We need to take this NADP, take the FADP, give it somewhere. That is ETC. You give it to ETC and they will give you the ATP. So you take this NADH and FADH, give it to ATC. ATC, students, ATC is your like bank here. ATC is like your bank here. And with the help of NADH and FADH, you can get a ATP here. Students, is the concept clear? Here, students, one NADH will give you 3 ATP and one FADH will give you 2 ATP. 2 ATP and 3 ATP. Like similarly, from a check you can get cash, from NADH you can get ATP. So ATP is like energy currency. ATP is like your energy currency in your body. Every single time there's a requirement, ATP is given, there ATP breaks down. Students, all of you understood? Students, all of you understood? Tell me yes or no. All of you understood, right? The concept of ATP and NADPH. All of you understood? Clear, all of you? No. This is the PYQ, students. One FADH gives how many? 2 ATP. One NADH gives 3 ATP. Okay. Now, students, based on utilization of oxygen, we have two types of respiration. Understand? One is called as your aerobic, one is called as anaerobic. In aerobic, presence of oxygen, cytoplasm and mitochondria takes place, CO2 and H2 are released here, more energy is released here in general. While at the same time, anaerobic respiration is absence of O2, no O2 at all here. It happens only in the cytoplasm. Ethanol and lactic acid are the byproducts here and less energy is produced, very less energy, students. Now, friends, which of the two happens in your body? Friends, all of you know aerobic respiration happens in your body. Do you think anaerobic respiration also happens in your body? Tell me yes or no, students. Tell me yes or no. Do you think anaerobic respiration takes place in the case of your body? It does. For example, let's take all of you. Let's take Purple, Madhu, Lakshay, Purna, Purni, right? All of you, I will take all of you to run a marathon. After running for 2 kilometers, you'll be like, I can't run. Students, I can't. You start panting a lot. You start getting very tired. So whenever your body also doesn't get required amount of oxygen, your body also does lactic acid fermentation. That is production of lactic acid in your muscle cells. In the case of muscle cells. Okay. So students, remember, this much you should remember. But this will generate very less energy. So be very efficient. Okay, be very, very efficient. Okay, now students. Now, what exactly happens in the case of aerobic respiration? Okay, students, how many of you ran marathon? I can run 10 kilometers. I usually go for participating marathons, 10 kilometer, 5 kilometer marathons. How many of you can run marathons? Tell me in the chat. How many of you can run marathons? Okay. Now, this is what happens in the case of aerobic respiration. We have glucose plus oxygen and we get carbon dioxide plus water. This is what happens in the case of aerobic respiration, students. Aerobic respiration. Now, let me give you an entire overview of the chapter right now. The overview of the chapter is very simple. It is starting off with glucose here. Glucose is your respiratory substrate. All of you agree? Yes. Now, starting it undergoes glycolysis right now. 10-step process. After that, we get pyruvate. This pyruvate undergoes link reaction to produce acetyl-CoA. Link reaction, acetyl-CoA. Now, this link reaction now enters something called as Krebs cycle here. In Krebs cycle, we get something called as NADH and FADH, which are used in the ETC here. ETC. ETC has 5 protein complexes here. And after ETC, students, we finally get the ATP here. We finally get the ATP. So, students, this right here is the entire chapter. All of you ready? Students, what is that sound? What is that sound? There is some weird sound coming from somewhere. Can all of you hear that? Students, can all of you hear that sound? Weird sound. I don't know what is that. Okay. Now, this is your entire chapter here. Glucose, glycolysis, pyruvate, link reaction, acetyl-CoA we get, Krebs cycle, NADH, FADH, enter the ETC, and then ATP formation happens, students. ATP formation. So, it is going to be, let's say, students, I will take probably 1 hour. In 1 hour, I'll finish the entire chapter, power packed session. Just let me know in the chat right now. All of you ready? Is every single one of you ready now, students? Ready, all of you? Next 1 hour is going to be power packed right now. If you give me the next 1 hour, I will teach you the entire chapter in just 1 hour. All of you ready, students? Ready, all of you? Power packed, no distraction. Forget anything else aside you. Just focus on what I'm saying. Focus on my words, and you're good to go, students. You're good to go. Next 1 hour is going to be very crucial. You have a very high chance that the concept I'll teach you right now will be asked in examination directly. Ready, all of you? Ready, all of you? Can I get a fire emoji in the chat? Can I get a quick fire emoji in the chat? Yes, sir. We are ready for the next 1 hour. Yes, we are good to go. Okay. Now, students, the first concept for today is called as, where do plants get the respiratory substrate from? Where do you think a plant is getting from? Do you think behind your back? Do you think plants are going to a shopping market? Behind your back, plants are going and taking 1 kg sugar, 1 kg rice, 1 kg wheat? No. Plants do not need the respiratory substrate from outside. Like how, and you and me, we eat respiratory substrate. We take carbohydrates. We take something called as your protein. We take fat. But for the plants, not required. Plants are like, you are inferior, I am superior. I do not need to take from anywhere. I make my own. Respiratory substrate, students. So plants can make their own respiratory substrate. They are self-sufficient. They don't have to care for anyone. They don't have to, students, care for anyone. They make their own 1 kg glucose or 2 kg glucose they can make. Now, do plants breathe? Where do plants get oxygen from? Do you think, students, like all of us, we have heard the stories. We have heard the stories that plants take CO2. But do you think plants also require oxygen? Behind your back, are they like, when you're not looking around, they're taking all the oxygen inside? Not really, students, not really. Plants do require oxygen, but they make their own oxygen also. Students, truly magnificent. See here, all of you should know, students, there are tiny pores like stomata here. There are something called lenticels here. But in reality, students, in reality, plants do not require a special organ for breathing here, because each plant part can take care of its own needs. Demand for O2 is very less here. The demand for O2 is very less, students, and as a matter of fact, photosynthesis byproduct, that is going to be oxygen. So plant already has the oxygen inside a cell. Plants are anyway doing photosynthesis, and all of you know, whenever there is something called a splitting of water. In non-cyclic, there is something called splitting of water. When the water undergoes photolysis, it is producing oxygen again. So plants has the oxygen in the cell. Plant is making its own food also. So tell me now, students, who is superior? Tell me now, students, isn't plant superior so much? Yes, they can't run away if someone is coming to eat them, unfortunately. But plants are so independent. They make their own oxygen. They make their own food also, which can undergo oxidation now. That's what answer. Now there is a small comparison of your respiration and combustion, which has much more similarities. They're showing you similarities. Okay. Now, students, now we come to the first topic, main topic on the hand, that is called as first step in all types of respiration. It could be aerobic, anaerobic, doesn't matter. This is the starting step in every single type of respiration. It could be your aerobic, anaerobic, unicellular, multicellular, prokaryotes, eukaryotes, plant, animals, in every single organism. This is the first step in respiration. Students, what is it, students? The first step in respiration is nothing but glycolysis. It is nothing but, students, glycolysis. Here, glyco means glucose or sugar. Glucose or sugar, and lysis means to break. So basically, we are breaking the sugar or breaking the glucose, chop, 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 and getting the ATP. Getting the ATP, students. Now, location is cytoplasm here. The process of glycolysis. It happens in the case of aerobes, anaerobes, doesn't require oxygen. That is why it happens in both. It doesn't require oxygen. Number of steps, 10 steps here. 10 steps here. See, students, in the case of plants, what happens? In the case of plants, the sugar is stored in the form of sucrose in the case of plants. So if a plant has to undergo glycolysis, it has to convert sucrose into glucose first. then it can undergo glycolysis. We cannot let sucrose directly into glycolysis. Not possible. Not possible. Only monosaccharides has to be there. Monosaccharides. So with the help of invertase enzyme, sucrose is converted to glucose and glucose enters the glycolysis. Glucose enters the glycolysis, students. Enters the glycolysis, students. Now, what is the other name of glycolysis? The other name of glycolysis, it is also called as EMP pathway. Not EMP pathway, students. That is Embden-Maroff-Parnas pathway, students. EMP pathway, also called as. Now, a small revision for all of you before we go further. All of you should know, students, what is oxidation? Loss of electrons or loss of protons. Oxidation, protons. What is reduction here? Gain of electrons or gain of protons is called as reduction. Is these 2 points clear to all of you, students? Is this clear to all of you? What is oxidation and what is reduction? This everyone should know. This everyone should know. Reduction is going to be gain. Oxidation is loss. Clear all of you? I want everyone to type this in the chat. I want every single one of you to type this in the chat. Oxidation, loss, reduction is gain of electrons. This much you type in the chat. This much all of you type in the chat. Okay. Now, the entire glycolysis can be broadly classified into 2 categories. The first category is called as preparatory stage. The second stage is called as payoff stage. So basically what we do here is, in preparatory stage we invest. Investment is happening. And in payoff, returns is coming. So when you invest money, you get the money back. You invest your time in studying right now, you will become a doctor later on. That is the returns. Okay. So here also, students, we have something called as preparatory stage and payoff stage. Preparatory and payoff. Now, understand what is happening in the case of preparatory, right? Preparatory. Understand. Okay. In preparatory, the first step is called as what? Here, first step is called as phosphorylation. Here, the first enzyme is called as hexokinase. Students, you don't have to remember all the enzymes. Remember invertase, remember the first enzyme, and remember the last enzyme. Anything in the middle, they'll not ask you. They'll not ask you, students. Okay. Now, what exactly is happening, students? This right here is a glucose molecule. Right here is, see, forget all this, focus on this. Here, right here, we have a glucose molecule here, 6-carbon molecule. Let's say I have an ATP here. Now, what am I doing is, students, I am breaking the bond here, investment, right? Breaking the bond here, and I'm taking this phosphate, and I'm adding it to the 6th position here. This phosphate is added to the 6th position here. So what do we get? What do we get? What do we get here, students? We get from glucose, we get glucose 6th position and phosphate, 6th position phosphate. So glucose will become what? Glucose will become glucose 6-phosphate. Understand? Very easy now, very easy. First we have glucose. We take a ATP molecule, break the bond, take the phosphate, 6th position. So we get glucose 6-phosphate. That is the first step. That is going to be the first step right here. First step right here. And in this first step, ATP is consumed. ATP is consumed, students. ATP is consumed. Now, students, now understand what happened. This is what we get. Glucose is phosphate and we get here. Phosphate is added here and it becomes ADP now. It becomes ADP, obviously. Now, students, what happens then? Now, something called as isomerization. Students, is the voice low? Students, is the voice low for all of you? Students, is the voice low? Tell me yes or no, sir. Tell me yes or no. One second, give me a mic. One second, students, 1 second. Give me 1 minute, 1 minute. One second. Let me just fix it. Don't worry. Let me just fix it. Till now, I'll just explaining. Listen to me. If I'm not audible, then I'll fix it one more time. I've asked it. The cell is coming. A new cell is coming, student. New cell is coming. Okay. One second. Give me 1 minute. Give me 1 minute. Okay. I'll get it fixed. I'll give you a recap also before I go further. Okay, because I don't want the flow to break. I don't want the flow to break, students. Give me 1 minute. Students, have you smashed the like button first of all?
Muted है ना?
Wait. Camera is. Students, quickly tell me right now if I am clear. Is it clear now? All of you? Clear? Back to normal? Students, all of you, is it fine now? Students, is it fine?
Is it fine, guys?
Yes? Yes, all of you? Okay. Now, students. Admit card, I think it's fine, students. I already finished admit card part. That's fine. It's fine now. So, I'll give you a small recap what we learned right now. Here, what we do is the first step is that we take glucose. Hexokinase is the enzyme. Phosphorylation is happening here. That is, this ATP, this bond is broken. This phosphate is added here, and glucose becomes what? Glucose 6-phosphate. It becomes glucose 6-phosphate. It becomes what, students? Glucose 6-phosphate right now. Now, next step is called as isomerization, students. Step 2 is called as isomerization. Now, what exactly is isomerization, students? Same but different. Very simple, students. In isomerization, what happens is that isomers are molecules with same chemical formula but different structure. So, what we do here is we convert glucose 6-phosphate into fructose 6-phosphate. See, chemical formula will be same, just the structure will differ. That's all here. Okay, so glucose 6-phosphate becomes fructose 6-phosphate right now. It becomes what? Fructose 6-phosphate. That is your second step here. And students, remember always, whenever there is isomerization, there is no energy transfer. ATP conversion is not happening. Okay, so second step is called as what? Isomerization here done. So glucose to glucose 6-phosphate, first step. Second step is isomerization. Now, what exactly happens in the third step again? In the third step, we take one more ATP. See, all of you tell me in the chat right now. We take one ATP, I break the bond here, and I take this phosphate here. Now, 6 position, 1 phosphate is there already, fructose 6-phosphate. I take this phosphate and I add it to the first position now. Tell me, students, what do you call this now? Initially, it was called as fructose 6-phosphate. Can I call right now fructose 1,6 bisphosphate now? Tell me yes or no. Students, tell me yes or no. Students, you don't have to remember this. Don't worry. So, what do we get? Fructose 1,6 bisphosphate. So, first we had glucose to glucose 6-phosphate, isomerization, fructose 6-phosphate. Now, fructose 1,6 bisphosphate. Done, students. So, till here, I believe there is no doubt. Glucose to glucose 6-phosphate, students. Then we have isomerization, and then fructose 6-phosphate becomes fructose 1,6 bisphosphate. First step, second step, third step. In first step and third step, ATP is being consumed here. Students. I want everyone to type this in the chat. All of you. Fructose 1,6 bisphosphate. Now what happens, students? Again, we'll just follow back here, come back here. Here, fructose bisphosphate. Till here, we are done. Till here. First step and third step, ATP is consumed. Okay. Energy consuming process. It's called as energy consuming process. Now, students, now we come to something called as next step here. This is very important, students. Now, what happens, sir? Fructose 6-phosphate, which was initially a 6-carbon molecule. Here. 1, 2, 3, 4, 5, 6. Now what happens is that this 6-carbon molecule will completely property we are dividing, students. Imagine it happens in the house, no? One brother is there, 2 brothers. After both of them get married, we divide the property. Now here, both the brothers are become old now. We are dividing the property now. So this will become a separate 3-carbon compound. This will become a separate 3-carbon compound right now. See here. And the first one is called as glyceraldehyde 3-phosphate. GAP, you can call it as GAP also, and other one is called as dihydroxyacetone phosphate. So what do we have, students? Here we have something called as your GAP, glyceraldehyde 3-phosphate. It can also be called as PGAL, that is phosphoglyceric acid, right? Glyceraldehyde, sorry, 3-phosphoglyceraldehyde. Both are same here. The other one is called as DHAP, that is dihydroxyacetone phosphate. So one fructose 1 bisphosphate is dividing into two molecules. It can be called as GAP or PGAL. Same thing. Okay, that is the one 3-carbon compound here. This is the other 3-carbon compound here. That is DHAP, dihydroxyacetone phosphate. Now, students, DHAP and PGAL are like your twin brothers. They can interchange. What do you mean? What do we mean by that? They are also isomers now. They are also isomers. So we can interchange. Yes, we can interchange, obviously. So what do we do here right now is we will interchange them. And for convenience, from now onwards, from this point onwards, what do we do here is that, students, instead of writing two separate, what will I do is, right, I will tell two molecules of GAP or two molecules of PGAL, okay, into two, because DHAP right now, DHAP is now converted back to PGAL because they are isomers. Since they are isomers, I am converting it back. I am converting this to this for convenience right now. I am converting DHAP to PGAL or GAP. Okay, here. So from now onwards, you can see in your NCERT also, everywhere we write into 2, into 2, into 2, into 2, into 2. Because this DHAP is now converted to PGAL or GAP. Is that point clear to all of you, students? Is that point clear to all of you? It is clear to all of you. So what happens next step? This is again 1, 2, 3, 4, 5. Fifth step. Now what is exactly happening in the sixth step, students? What exactly is happening in sixth step? In sixth step, the entire molecule is undergoing oxidation now. This entire molecule, friends. Now this is what payoff stage starts. From here, payoff stage starts right now. Now what is happening is that, students, this molecule, two molecules of GAP, glyceraldehyde 3-phosphate, okay, glyceraldehyde 3-phosphate, right, it is now undergoing oxidation. Now can anyone remind me what is oxidation again? Friends, what is oxidation? Oxidation is nothing but loss of electrons or loss of protons. So this GAP molecule, glyceraldehyde 3-phosphate, right, it undergoes oxidation. It will release electrons, release protons, release energy. Everything is released. Everything is released here. 2 electrons, 2 protons, 2 energy is released now. Now what is happening, students? Who is coming and picking these two? Let's say someone is coming, right? Someone has to come and pick them. These electrons and protons released by the oxidation of GAP, is now picked up by NAD+. Where NAD+ is coming and picking it up. All of these electrons, the protons and electrons are going to NADP. Now, students, all of you tell me this right now. If this NAD+ is gaining, it is gain of electrons happening here. So, can I tell this NAD+ is undergoing reduction now? Yes, NAD+ is undergoing reduction right now because it is gaining this electrons and protons here. It is doing. And students, that's how after gaining the electrons, we end up getting NADH here. So NADH. So again, students, if you go back to this entire flow chart here, you will notice in the sixth step here, there is a formation of NADH here. That is, NAD+ is getting reduced to NADH, while this molecule, that is GAP, is undergoing oxidation. GAP is undergoing oxidation, while your NAD+ is undergoing reduction to produce NADH. Students, how many of you understood? How many of you understood, students? How many of you understood? Raise your hands. Here, NAD+ is getting reduced to NADH. It is gaining the electrons, gaining the protons released by GAP after oxidation. After oxidation, students. After oxidation. Now, students, what is the use of the energy now? This energy has to be used somewhere, no? It has to be used. So, what are we doing with this energy, students? What do we do with this energy right now? Now, we will take this energy. This energy is used now. This energy is used to bring inorganic phosphate phosphate from the cytoplasm. Now what we do is we use the energy and bring a phosphate here. Now what we do here is that, students, one second students, one second. What do we do right now is we take this phosphate and attach it here. Attach it here, students. Now it was called as glyceraldehyde 3-phosphate, right? Then what do we call it now? What do we call this, students? What do we call it? Glyceraldehyde 3-phosphate will now become what? 1,3 bisphosphoglyceric acid. Students, concept all of you understanding now. It became what now? 1,3 bisphosphoglyceric acid. And you can see here, students, and you can see here in the entire flow chart here, it became 1,3 bisphosphoglyceric acid. Now all of you know exactly why it is becoming exactly 1,3 from 3 glyceraldehyde 3-phosphate. From glyceraldehyde 3-phosphate, how it actually became 1,3 bisphosphoglyceric acid. 1,3 bisphosphoglyceric acid because this extra phosphate is added to here from the energy released by the oxidation. And we can see, students, we can see a NAD plus here. Now, students, it is always into 2 right now. Into 2 because 1 from here and 1 from here into 2. Now, students, is that point clear to all of you, students? Is that point clear to all of you? 1,3 bisphosphoglyceric acid. Now, students, what we do here is that I have told you all the energy is stored in the bond. All the energy is stored in the bond here. So, what do we do here? We break the bond here. We break the bond. If this bond is broken right now, this phosphate is free. This phosphate will now go and attach to this phosphate, and that's how we get a ATP here. That's how we get a ATP molecule. That's how we get a ATP molecule. And from 1,3 bisphosphoglyceric acid becomes 3-phosphoglyceric acid, because from the first position the phosphate has been removed right now. It has been removed from here. So it became what now? 3-phosphoglyceric acid, and we get 2 molecules of ATP here, because into 2 everywhere, into 2. Now, students, what do we do? Next step is again isomerization. So what we are doing here is from 3-phosphoglyceric acid, this P is transferred up. It is transferred up now. It becomes 2-phosphoglyceric acid, 2-phosphoglycerate here. So 3-phosphoglyceric acid to 2-phosphoglycerate. Again, isomerization, there is no change, no energy change. Okay. Now again, students, what we do is we remove the water molecule from here. We remove water molecule, it becomes PEP, phosphoenol pyruvate. PEP. Remember in C4 cycle, same PEP. Phosphoenol pyruvate. Now this phosphoenol pyruvate, what we do here is, again, this bond is broken here. This phosphate is released. It goes and attaches here. Bond is broken. And we get pyruvate in the end, and we get ATP, students. And we get a ATP molecule here one more time. We get a ATP molecule here one more time, students. So first step, second step, third, 4, 5, 6, 7, 8, 9, 10. 10 steps in total, students. 10 steps in total for your entire glycolysis. Now, students, you might be wondering, sir, do I have to remember this entire thing? Not really. You have to really focus on what actually is happening at every step. Or which step? I'll tell you. Let me tell you. How will the question be asked in examination? This is the question will be asked in examination. Which step ATP is consumed? First and 3. Which step ATP is produced? 7th and 10th step here. You can see here, students, in 7th and 10th step. In the 7th step and 10th step, ATP is produced. So you should remember what exactly happens 7th step. 1,3 bisphosphoglyceric acid to 3-phosphoglyceric acid. So 1,3, 1 is removed, so we get 3. So ATP is produced. Here we get last step. All of you should know pyruvate formation. That is PEP to pyruvate. 1 ATP is produced. So this 10th step and 7th step you should know. You should know. Now, rate determining step here means the slowest step here. Slowest step. 1, 3 and 10. Oxidation, 6th step, all of you know. PgAl to 1,3 bisphosphoglyceric acid. Here oxidation happens. EMP pathway, Emden Mayer of Parnas. Emden Mayer of Parnas right now, students. Okay. Which step what is released? 9th step. It happens in aerobes and anaerobes. Both it happens. First enzyme. The first enzyme is called as hexokinase. Last enzyme is called as pyruvate kinase. Pyruvate kinase. If by chance a question does not come from anywhere, the question will come from here. here all of you know, in preparatory stage, minus 2 ATP consumed, 1 and 3 steps. Payoff stage, 2 NADH. All of you should know 1 NADH will give you 3 ATP, 3 into 2, 6 ATP, 6 ATP, right? 2 ATPs, right? 2 ATP in 7 step, 2 ATP in the 10 step. Always into 2 right now. So in total, how many ATPs do we have? Let's say 6 plus 4, 10 ATP in total. Now if you do minus 2, this one minus 2 if you do, it becomes 8 ATP. So 2 plus 2, 4, 4 plus 6 is 10 gross, minus 2 ATP in preparatory, you get 8 ATP is the net gain here. 8 ATP is the net gain here, students. So is that point clear to all of you, students? Is that point clear to all of you? This is called as a net profit. This is called as net profit, students. And that is your entire glycolysis. Okay. Students, is that point clear to all of you? And students, do you realize in any of these places, right? In examination, let's say in examination, if they ask you how much of ATP is produced directly, that is substrate level. If they ask you in examination how much of ATP is produced directly, when they ask you question directly, in that case, you don't have to include this. In examination, if they ask you how much of ATP is produced directly, that time don't include this. Then how much would tell me? Here 4, here 2. 4 minus 2 is 2 ATP. Students, 2 ATP in that case. In that case, 2 ATP directly, without NADH. Without NADH, that is. Students, is that clear to all of you? Is that clear to all of you, students? 2 ATP in that case, directly, without NADPH. Students, this is how the question will be asked. In plants, sucrose is converted to 2 molecules of monosaccharides due to the action of invertase. Then both enters the glycolysis pathway. Identify the monosaccharides. That is, what is the sucrose are made up of? What is sucrose made up of? It is made up of glucose and fructose. It is made up of glucose and fructose. Glucose and fructose. See here. Or question can be asked like this. In glycolysis, 1,3, all of you remember, 1,3 bispecific acid to 3-phospho. See, this one molecule is removed here and that goes into ATP formation. So it is going to be a energy yielding process. It is going to be a energy yielding process. So all of you understood? It is going to be a energy yielding, it will give energy. See here. ATP is used in 2 steps in glycolysis. All of you know it is in 2 steps. Use 1 and 3. The first ATP is used to converting glucose to glucose 6-phosphate. Correct. Second ATP is used to convert fructose 6-phosphate to fructose 1,6 bisphosphate. Diphosphate here. Tell me, students. Correct? Yes. Makes sense? Makes sense. So, first step and third step ATP is consumed. That is what they're telling you. So, do you realize? Question will be asked from which step ATP is consumed, which step ATP is produced, which step water is removed, which step oxidation happens. That is how question will be asked for examination. That is how question will be asked in examination. See this question, see this question. 2019 question. Conversion of glucose to glucose 6-phosphate. The first irreversible reaction of glycolysis is catalyzed by. I told you, the first enzyme is hexokinase. Now, sucrose is converted to glucose and fructose by the enzyme invertase. Friends, all of you understanding? See this. Glycolysis is found in cytoplasm of virtually all type of aerobic and anaerobic cells. In this process, glucose is converted to the end product here. If that is going to be pyruvic acid. Students. Pyruvic acid. Look at this question, students. In which of the following reactions of glycolysis, a molecule of water is removed from the substrate? That is the ninth step. That is PEP formation. 2 phosphoglycerate to phosphoenol pyruvate, PEP formation. Water is being removed here. Water is being removed, students. So that's how question will be asked. That's how question will be asked in an examination. Don't expect any direct question. You will have to think. See, if you know the questions I gave you, what step what happens, which step what is removed, which step you have ATP is consumed, which step ATP is produced, you're good to go, students. You're good to go, all of you. So, students, can all of you say you truly understood glycolysis? Instead of sitting and by-hearting the entire thing, smartly you understand first and learn the questions, you're good to go. No need to sit and by heart the entire thing. No need to sit and by heart the entire thing, students. Okay? Okay. Okay, students? All of you? Okay? Done? Now, students, what is the fate of pyruvate now? That is, once a pyruvate is made now, after glycolysis, what are the life possibilities it has? For example, after 10th standard, you can take commerce, you can take science, you can take arts, you can do anything. Similarly, students, let's say this is a class 10 student now, right? After pyruvate formation, it has multiple options right now, multiple options. Okay, let's see. Let's say glucose glycolysis partial oxidation. Friends, why do I call it partial oxidation? Why do I call glucose as partial oxidation? The answer is very simple. Because in oxidation, you should always get CO2 as the end product here. But students, in the entire glycolysis, did you find CO2 release anywhere? No. In the entire glycolysis, there was no CO2 release anywhere. That is, carbon was not converted to glucose. Carbon was never converted to CO2 and released. Never converted to CO2. So it is called as partial oxidation because there was no CO2 released here. Okay. Now, pyruvate is 3 carbon, 3 carbon. Here, students, again, into 2. All of you should know, into 2 pyruvate. From 1 glucose, we get 2 pyruvates. We get 2 pyruvates. Now, this pyruvate here, in the absence of oxygen, in each cell, it undergoes alcoholic fermentation, which all of you want to learn. Okay. Now, what do we get here? We get ethanol here, we get CO2 here, energy. So, students, here, pyruvate is 3 carbon, 1 CO2 is released as such here. So, can anyone tell me ethanol is how many carbon then? Ethanol is how many carbon? See, 3 carbon here, 1 carbon released here, so 2 carbon. So, all of you should know ethanol is going to be 2 carbon in nature. Chemistry makes sense. Okay. Now, again, in the absence of oxygen, muscle cells, lactic acid fermentation, lactic acid plus energy is produced here. And you can see there is no energy being released here. Okay. Presence of oxygen in mitochondria, we get oxygen, water, energy. This is what we are going to be interested in. This is what we are interested in mainly. Okay. Now, students. Now we come to anaerobic respiration, lactic acid and alcoholic fermentation. Understand, students, understand. This is very, very important. If you understand this concept right now, instead of by hearting it, you will understand it very well. Listen to me. Now, what happens actually here? All of you know, students, this NADH right now. This NADH right now, students, where is it produced from? This NADH is coming from where, students? It is coming from glycolysis. Okay. And all of you should know that NADH is produced in glycolysis. The NADH produced in glycolysis is a result of oxidation. All of you agree with me? 6th step of glycolysis. In 6th step of glycolysis, the NADH was produced as a result of glycolysis, as a result of oxidation. Right? Now, if you open your NCERT right now and you check a line there, it says that NADH undergoes reoxidation. Many of you will not understand it. Many students will just by heart this. Many students just by heart it. Okay. It says NADH undergoes reoxidation. So it means that it has undergone oxidation before. So this right here is a product of oxidation before. Now it is undergoing reoxidation. That means oxidation one more time. That is a concept which I want you to learn. This is not mentioned in your NCERT clearly. If you are in my class, you will learn this. That is the advantage of my class. We go in detail. Even though exam is in very few days, exam is this Sunday, right? I still teach you in detail. Okay. So it undergoes reoxidation. Now, can all of you tell me, students, if this NADH is undergoing reoxidation, will it release electrons or gain electrons? This NADH will release electrons and release protons right now. Electrons and protons. Now, these electrons and protons, once it releases, can you see here? It becomes NAD plus. Oxidized after oxidation. After oxidation, students. Okay. After oxidation right now. Okay. Now students, understand what happens here. These electrons and these protons right here, these electrons and protons are now taken up by pyruvate. They are taken up by pyruvate right now. Now, can I tell students, can I tell pyruvate right now is the final, first and final electron acceptor in the case of lactic acid fermentation. Now you tell me students, if pyruvate is accepting these electrons, can I tell pyruvate is undergoing reduction right now? Reduction, yes. Pyruvate is undergoing reduction right now. Once it undergoes reduction, we will get lactate here, lactate. And here in examination, very high chance they will ask you this, the enzyme present here. The enzyme present here, students. Okay, the enzyme present here is none other than the lactate dehydrogenase. It is going to be lactate dehydrogenase, the enzyme here. So NAD is undergoing reoxidation. Electrons and protons are taken up by pyruvate, undergoes reduction to produce lactate here. Okay. Now the same concept applies here also. But here what happens that students, pyruvate in the starting, it doesn't undergo directly. It is not picking up electrons directly, not undergoing reduction directly. This pyruvate right now, students, undergoes something called as decarboxylation. It will lose the electrons and protons here. Okay, so it will lose CO2 here. So it undergoes decarboxylation to release CO2. CO2 is released. So here we have 3 carbon, we have 2 carbon here. 2 carbon here, students. And enzyme involved is pyruvate dehydrogenase. Decarboxylase is the enzyme here. Carboxylase is the enzyme here. Now, students, now this acetyl group is here. Now again what happens is that this NADH here undergoes reoxidation. Reoxidation, students. Undergoes reoxidation. And we end up getting the NAD plus here. NAD plus. Okay, we get the NAD plus here, students. Okay, so it will release the electrons and protons here. Now, so if I ask you a question right now, who is the final electron acceptor? Who is the final electron acceptor in the case of alcoholic fermentation? What is your answer, students? What is your answer? All of you. The final electron acceptor here is going to be acetaldehyde. It will take up the electrons and protons. And it will undergo now reduction. Undergoes reduction to produce the ethanol here. Ethanol. So, and the enzyme here is alcohol dehydrogenase. Alcohol here, alcohol no, dehydrogenase here, dehydrogenase. Pyruvate decarboxylase, from pyruvate decarboxylation is happening. If you go back here, students, lactate dehydrogenase, dehydrogenase here, dehydrogenase. Alcohol dehydrogenase. Pyruvate decarboxylase, pyruvate decarboxylation is happening. So that is your entire alcoholic fermentation, students. Entire alcoholic fermentation. Now, this is your NCERT diagram here. You can see clearly here it says that here PP, pyruvic acid, lactic acid fermentation, alcoholic fermentation. In both of these cases, NADH is again reoxidized to NAD plus. Can you see? Converted to NAD plus, students. Now, can anyone tell me what is the net gain of ATP in the case of alcoholic fermentation and lactic acid fermentation? See, all of you know, in preparatory stage it's -2. Preparatory stage it is -2. Payoff stage we get 4 ATP, we get plus 4 ATP we get. And we also get NADH. But now, students, we cannot take this NADH to consideration right now. We cannot take this NADH into consideration because it is being used here. It is being used here. So the net gain of ATP is going to be 4 minus 2, 2 ATP we gain, that's all. Only 2 ATP is the gain for 84 alcoholic and lactic acid fermentation. 2 ATP gain is happening here. How much, students? 2 ATP gain is happening here. Students, remember I told you, in the case of your entire anaerobic respiration, very less energy, students. Very less energy is produced here. Very less energy is produced here, students. Now, students, here's a question to all of you. Choose the wrong statement, students. Choose the wrong statement here. Students, what is the answer? I'm telling you, no, I'm telling you how many ATPs are produced. CAT is harmful molecule, correct here. Breathing is part of respiration. Respiration always requires O2, no. Respiration doesn't require, students, doesn't require always O2. Doesn't require always O2. Respiration can happen without oxygen also. It can happen without oxygen also. Now, this is also one more important question here. What amount of energy is released from glucose during lactic acid? Very less energy, students, very less energy, 7%. Just 7% energy is released. Just 7% energy is released during lactic acid fermentation. Very less energy. Very less energy is produced. Now, students, now we come to aerobic respiration. Now we come to the entire aerobic respiration right now. Okay, that is glucose plus oxygen, right? Energy is released, CO2 plus H2O. This is the entire thing is called as aerobic respiration right now. Aerobic respiration. Now, what did we learn till now, students? What did we learn till now? Till now we learned that, students, in the cytoplasm what is happening? Glucose is converted to pyruvate. Now what happens, students? This pyruvate is now entering inside the mitochondria, students. This pyruvate right now is entering inside the mitochondria, where other next step will happen. Other three next steps will happen, students. Now this pyruvate is entering inside. It will now undergo link reaction, Krebs cycle, and etc, students. One by one it undergo etc, undergo link reaction, Krebs cycle, and electron transport chain. One by one it will go now, students, one by one. Now the first step is what, students? The first step is called as link reaction. Link reaction or gateway reaction. Link reaction or gateway reaction. Gateway reaction. Link reaction or gateway reaction, also called as, students, also called as what? Also called as oxidative decarboxylation. All ready, students? All ready? Again, by hearting is very easy. I'll make you understand, students. By hearting is very easy. I'll make you understand. Listen to me. Okay. Now, what exactly is happening here? All of you ready? Students, ready, all of you? Everyone in the chat. Ready, 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 ready? Now, students, understand. Now, what happens initially? First step. In first step, what is happening? Here we have pyruvate from glycolysis. Now, the first step here is that this pyruvate undergoes decarboxylation now. Decarboxylation. So, what do we have, students? If carbon dioxide is released from here, it is completely removed. What are we left with? We left with COCH3. COCH3. Acetyl group here. Yes. We are left with COCH3. We are left with. We are left with COCH3. Okay, COCH3 we are left with. Now, students, understand here this molecule right now. This molecule right now, after decarboxylation, students, this molecule will now undergo a process called as what? Undergo a process called as oxidation. It will now undergo a process called as oxidation, students. Now, all of you tell me in chat right now, if this molecule right now, it is undergoing oxidation right here, will it release electrons and protons? Yes, it will. It will release electrons and it will release protons. After releasing electron protons, this molecule here, we will add something called as coenzyme A, and we end up getting acetyl CoA. See here. First we have pyruvate here, decarboxylation, we get acetyl group. This is acetyl group, acetyl. We get the acetyl group here. This acetyl group undergoes now oxidation. This acetyl group is now undergoing oxidation, and to this we add a coenzyme part. Coenzyme is added to this. Now it becomes acetyl CoA. Now the question is, students, who is taking up the electrons and protons? Who is being receiving it, students? Who do you think will receive, students? Who do you think will receive these electrons and protons? Can anyone tell me in the chat? Who is the gracious person who will like give me? Who is the person who will say give me everything? Give me. Friends, quick. Tell me, students, who is the person who will say give me the entire electrons and protons? Anyone who can tell me? Students, anyone who can tell me who is receiving them? Anyone? NAD plus, very good. So these electrons, protons are given to NAD plus, and NAD plus undergoes reduction. And that's how we get NADH. And that's how we get NADH. All of this is happening inside mitochondria, inside mitochondrial matrix. Mitochondrial matrix. Okay. And the end, students, in the end, what do we get? From one pyruvate, after decarboxylation, after oxidation, after getting one NADH, we end up getting something called as acetyl CoA. And students, this acetyl CoA right here, it can now enter which cycle, students? This acetyl CoA can enter where? This acetyl CoA now can enter something called as Krebs cycle. Students, did all of you understand how do we get? Students, how do we get this NADH in the oxidative decarboxylation? Yes, it is also called as oxidative decarboxylation. Students, do all of you remember? Do all of you understood how do we get this NADH in a link reaction? If you open your NCERT, it tells you that in the entire link reaction, it shows that we get the NADH. That's how we get the NADH, students. That's how we get the entire NADH in this reaction. Okay. Now, students, if you understood this entire oxidative decarboxylation, which also happens in a Krebs cycle, it will be easy now. It will be easy right now. Okay, see here, students. Now, oxidative decarboxylation is a link between glycolysis and Krebs cycle. Glycolysis and Krebs cycle, students. Okay, it's a 2-carbon compound. Okay, 2-carbon compound here. Okay, so we are done with this one matrix. Now we come to Krebs cycle, now, students. Now we come to Krebs cycle. All of you ready? Students, all of you ready now for Krebs cycle? Students, I will teach you Krebs cycle in 1 minute and 5 minutes. 1 minute and 5 minutes, understand. In 1 minute, how do we learn Krebs cycle? Officer, can I keep selling sandwiches for money? Officer, can I keep selling sandwiches for money? Oxaloacetate, citrate, isotrate, alpha keto, succinate, succinyl coenzyme A, fumarate, malate. Done. Done, students. This entire Krebs cycle was discovered by Hans Krebs. That's what's called as Krebs cycle. Also called as TCA cycle, tricarboxylic acid cycle. Okay. See. Officer, O. Citrate, can I keep selling sandwiches for money? Oxaloacetate, citrate, isocitrate, alpha keto, succinyl coenzyme A, succinate, fumarate, malate. Oxaloacetate, citrate, isocitrate, alpha ketoglutarate, succinyl coenzyme A, succinate, fumarate, malate. And malate here. So all of you, type this in the chat. Done? Done, students. So this is your entire Krebs cycle. This is your entire Krebs cycle. That's all. Here, if you learn what is the carbon number, in what stage, what is being released here, Krebs cycle is done. That's all. Krebs cycle is done in that case. For example, students, acetyl CoA enters where? Acetyl CoA is 2-carbon. It is entering along with oxaloacetic acid, oxaloacetate, which is 4-carbon here. 4-carbon. 4-carbon plus 1 carbon is how many carbon, students? 4-carbon plus 1 carbon is how much? 6 carbon. So citrate, we get 6 carbon. Citrate to isocitrate. Again, students, isomerization, no energy released. Isomerization, no energy is being released here. So we get isocitrate now. Now what happens, students, here? The first OD happens. The first OD happens. What do I mean by first OD? Right? Understand, students, understand. This molecule right now, isocitrate to alpha ketoglutarate, it undergoes oxidation and decarboxylation. That is called as OD. So this molecule isocitrate is undergoing oxidation and decarboxylation. Okay, so can all of you see one CO2 is released out? It has been released out. One CO2 is released out here. And all of you remember, students, here what happens is that in oxidation, when it is undergoing oxidation right here, right, it is releasing electrons and protons. It is releasing. Here we have something called as NAD plus also. the electrons and protons released by the isocitrate is picked up by NAD plus, and that's how we get NADH here. That's how we get NADH, students. So, so how many NADH produced in oxide decarboxylation means we have to consider from Krebs cycle also. Now we have to consider only Krebs cycle that time. If it is not mentioned in your NCRT, if in the question paper they will mention how many NADH produced from link reaction or from pyruvate. In that case, you have to include link reaction. I'll teach you that right now. Wait for it. Now students, now again what happens? After alpha ketoglutarate, to again next step, one more step. Again, what do we have? We have the second OD here. Again, what happens? This undergoes decarboxylation and also oxidation. Again, here we have NAD plus, which is undergoing reduction to produce NADH. So that's how we get, from second OD we get 1 CO2 and 1 NADH, and alpha keto becomes a 5 carbon. 6 carbon to 5 carbon, 5 carbon to 4 carbon. So in examination, what they can ask you? In examination, they can ask you, students, which of the following is a 5 carbon compound? Tell me the answer, students. Tell me the answer. Which of the following is a 5 carbon compound? What is your answer, students? Alpha ketoglutarate. Alpha ketoglutarate is a 5 carbon compound. Simple. That's how the question will be asked in examination. Okay. Now, this succinate coenzyme gets converted to succinate by production of GTP. Now, here is one more question for all of you. Remember, students, this GTP produced here, this GTP produced right here, it is result of substrate level phosphorylation. Similar to how we make ATP. In the case of glycolysis, same way it is done. It is substrate level. This GTP is substrate level. Only substrate level here. That is succinate coenzyme, succinate coenzyme to succinate here. Then we get FAD, one between succinate to fumarate, and fumarate to malate when water has been released, and malate to oxaloacetate, we get an FADH. Then this FADH as well as this NADH or this FADH, both of them are result of normal oxidation. Not because of oxide decarboxylation. Decarboxylation will happen only twice here. Students. It will be because of decarboxylation. Now, students, let me ask you a question. The question is very simple. The question is, in one TCA cycle, how many times CO2 is released? Simple question. This is how the question will be asked in examination. In one Krebs cycle or one TCA cycle, how many it is released? Simple question. Students, what is the answer? How many CO2? 2 CO2. Answer is 2. See, that is isocitrate to alpha keto, 1 CO2, alpha keto to succinyl coenzyme, 1 more CO2. Now, next question I can ask you. How many NADH produced? In total, in this OD we have 2 and 1 independent. So 3 NADH. How many FADH? 1 FADH. How many substrate level? Substrate level is only 1. Substrate level is only 1, students. Okay. Now, I will change the question to, let's say, for pyruvate. For pyruvate in Krebs cycle, how many NADH? For one pyruvate only in Krebs cycle, I'm asking only one Krebs cycle. For a pyruvate in Krebs cycle, how many NADH produced? See, for one acetyl CoA, we get 3. And all of you know, students, pyruvate also same thing. Tell me yes or no. Pyruvate also same thing. So what do you mean substrate level? Substrate level means it is not using, see, here when I say this NADH is giving 3 ATP, this NADH is giving 3 ATP because of ETC. But this FADH also because of ETC. But this substrate level, Directly it is giving. Students, tell me in the chat. Same for pyruvate is the same thing. Tell me students, for one pyruvate in Krebs cycle, how many NADH? In one Krebs cycle, for one pyruvate, how many NADH, students? NADH will be same to same, right? NADH will remain same to same. 3 NADH only. One here, and one here. Tell me yes or no. See, this acetyl CoA is coming from one pyruvate itself. It is coming from one pyruvate itself, right? It is coming from one pyruvate itself, right? So, how many NADH do we see here, students? How many NADH do we see here? 2 NADH or 3 NADH? Tell me. All of you quickly in the chat. Student, this is how they will catch hold of you. I'm going to tell you how they can catch hold of you right now. I'm going to tell you how can they catch hold of you right now. Because I told the question is what? In Krebs cycle. That's why I did not use link reaction here. Okay. How many students? How many NADH? 3 NADH in that case. 3 NADH in that case. Now I'll change the question. I'll change the question to glucose. I will change the question to glucose. So how many NADH for glucose? Into 2. So we'll get into 2. Because one glucose will give you 2 pyruvate. So 6 NADH. 6 NADH for 1 glucose. 6 NADH for 1 glucose. Okay. 6 NADH for 1 glucose that time. It will become 6. It will become 6 that time. Okay. Now, per glucose, if I don't add Krebs cycle, then how many NADH? Per glucose, how many NADH now tell me? Per glucose, how many NADH do we get? If I just take 1 glucose and I give it to you, how many NADH do I get in total? From 1 glucose, students, from 1 glucose, how many NADH do I get? Do we get 2 NADH in glycolysis? 2 NADH in link reaction? 6 NADH in your Krebs cycle. How much, students? How much? All of you understood? Did all of you understand, students? Did all of you understood how they can catch hold of you? Yes? Okay. Now, if I change the question from link reaction, if I say from link reaction, this is removed. This is removed. This is removed that time. Done, students? Done? All of you? From link reaction to Krebs cycle. So, now, done, students? All of you done? One second. This has to be per glucose, no? Done, students? Okay. Last question before I go to next topic. How many CO2 released from glucose after glycolysis? Purni, it depends on the question. Depends on the question. Last question. Students, see here. All of you. It says after glycolysis, right? After glycolysis. how many CO2 released from one glucose after glycolysis? So after glycolysis, we get link reaction and we get Krebs cycle. Link reaction, Krebs cycle. In general, from one link reaction, how many CO2 do we get? In link reaction, how many do we get? Students, why? How come zero? Tell me first of all. After glycolysis, how many? From one link reaction, how many CO2 do we get release? In link reaction, how many CO2 do we release? In from one link reaction. From one link reaction, we get one CO2 for one pyruvate. One is released, right, for one pyruvate. Now, since I am writing glucose here, so we'll get 2 CO2 here. 2 CO2 here. Okay, 2 CO2 here. Okay, one glucose, 2 CO2. Now, when I say Krebs cycle, in general, Krebs cycle, how many CO2? 2 CO2 in Krebs cycle. In one Krebs cycle, we get 2 CO2 for one pyruvate or one acetyl-CoA. Since I am writing glucose now into 2 again, so we get 4 CO2 here. So in total, students, how many of you are understanding the concept, not by hearting any of this? So if you by heart, you will forget in exam. If you get a question like this, you will forget in exam. Okay, you will forget in examination. Okay, this is how you do it. This is how we do it, students. Done. 4 in the Krebs cycle, because of it says one glucose, not one pyruvate or not one acetyl-CoA. One acetyl-CoA is 2 CO2. 2 is 4. Link reaction 2. So 6 in total. Students, in one Krebs cycle, how many CO2? Tell me. In one Krebs cycle, how many CO2 do we get? Tell me in the chat. Krebs cycle, how many CO2? Krebs cycle, we have only 2 CO2 here. 2 CO2. 1 acetyl-CoA. 2 times 4. 2 times 4. Done. Students, how many of you understood the concepts? Here is one more plane. 2 CO2 released in total here. For 1 acetyl-CoA. 2 acetyl-CoA, 4. That's all. Friends, look at this question. Identify the step in tricarboxylic acid cycle which does not involve oxidation. Does not hold oxidation is going to be. See, don't focus on values. Focus on understanding concept. If you by heart, you will not understand it. Just understand the concept. No need to by heart any of it. Here we get GTP. Here we get GTP, students. Here we get GTP. See, simple question. Solve this. Simple question, solve this. How many times decarboxylation occurs during TCA? In one TCA, how many times decarboxylation happens? That is, how many times CO2 is released in one TCA? How many is released? Okay, that is going to be twice. 2 times it is happening. 2 times it is happening. For in one. Again, if I change the question, change the question to glucose, then it is 4 times. Then it is 4 times. 4 times for glucose. Okay. 5 carbon compound, alpha keto. Alpha keto. Now, students, I will teach you the last concept today. Remaining part, students, this you can study on your own. This you can study on your own. It is nothing much. This is a respiratory balance sheet. I'll teach you later on. Okay. This you can study on your own. This is very easy. Okay. That is, fatty acids undergo beta oxidation to give acetyl-CoA enzyme. That is called for backup here. That is, fats can be broken down into this. This is a PYQ. This is a PYQ also. Okay. This all of you know, glycerol undergoes oxidation to produce DHAP. So they can enter from anywhere. Proteins can also be broken down. So here what happens here is that, students, fats can enter through acetyl-CoA. Amino acids can enter through pyruvic acid. This you have to remember. That's all. This you have to remember. Very easy. And all of you know this also. The respiratory quotient also. That is, it is going to be 1 for your carbohydrates, 0.7 for your lipids, and it is going to be 0.9 for proteins. 0.9 for proteins. This much all of you know. Now, students, okay. Now, students, I will teach you the main khichdi of this chapter called as ETC. Ready, all of you? Students, last topic for today. Last topic. Shall we start? Students, I want everyone to call your friends right now, because now I'll be teaching you the entire ETC one shot. Last concept in this chapter. The last khichdi chapter in this chapter. Students, how many of you ready? In the chat. See, students, if I am ready to teach, you should be ready to listen. Last concept, the most khichdi, the most confusing concept, the most confusing, let's say confusing part of this chapter, that is going to be your ETC. All of you ready? Ready, all of you, students? Students, growth is very easy. Growth I will not teach you much. In growth, I will teach you only the what's that? Entire phytohormones. That's all is important there. Students, ready, all of you? Last part of this chapter. Ready? Students, ready, all of you? Nice. Let's go. Let's go, students. Let's go. Now, students, where does it happen? The NADH and FADH are transferred to electron transport chain here. ATP is formed as a result of transfer of electrons here. So what they do is, students, they play jumping game here. Jump. They keep on jumping here and there, and they use electrons, and they give ATP in the end. Okay, they give ATP in the end, students. It happens. The entire thing happens on the inner mitochondrial membrane. Students. Now here, I want all of you to write this down in your book right now. The protein complexes as well as the mobile carriers. Students, excuse me. Students, can we start? Okay. So these are called as your protein complexes. We have complex 1, complex 2, complex 3, complex 4 and complex 5. So here, students, we have in total 5 complexes here. We have in total how many? 5 different complexes here. Like a 5 building. Imagine there are 5 buildings here. Okay. Building 1 is called as NADH dehydrogenase, succinate dehydrogenase, also called as your FADH dehydrogenase. FADH dehydrogenase. Okay. Cytochrome BC1 complex, cytochrome oxidase, ATP synthase. Then we have ubiquinone here and cytochrome C. These two are called as what, students? Mobile carriers. Students, suddenly something went inside my nose, I guess. Okay. This is called as your mobile carriers right now. They are called, students, mobile carriers right now. Okay. This chapter will not evaporate, students, don't worry. All you need to do is revise the chapter one more time. Okay. Now, students, understand. This is the khichdi here. Students, at first glance, you might not understand anything here. At first glance, you might not understand. Now, I will teach you the entire khichdi. I'll teach you right now, point by point, and then you'll understand, students. All of you ready, students? All of you ready? Point by point. It looks very massive. It looks very, scary, but it is not. It is very, very easy. It is very, very easy right now, students. Ready, all of you? Ready? Students, all of you ready? Now, one by one, let's completely decode this. Okay, completely, let's decode this. Now, here we have, students, all complexes are not arranged in order here. First of all, we have complex 3, complex 1, complex 3, complex 2, complex 4 and complex 5. Okay? Okay, students. So, complex 1, complex 3, repeat after me. Complex 1, complex 3, complex 2, complex 4, complex 5. So, all 5 different complexes here. The only thing is there that complex 3 is coming first and complex 2 is coming later. They are interchanged here. They are interchanged, students. Okay. Now, this entire thing is happening, students. This entire thing is happening where? Entire thing is happening on the inner mitochondrial membrane here. Inner mitochondrial membrane. This part is called as a matrix where the Krebs cycle takes place, and this part is called as intermembrane space. Between the membrane, the space is between the membrane. So we have 1, 3, 2, 4, 5. Okay. Now, students, understand what happens here. In examination, they can ask you the filling inside also. Filling. Now, the first one is called as what? NADH dehydrogenase. Here we have FMN and FES. Easy to remember. What is complex 3 called as? Complex 3 is called as what, students? Complex 3 BC1 complex. Tell me yes or no. Cytochrome BC1 complex. So we have to fill BC1 here. Cytochrome BC1 and FES. BC1. Then we have FADH dehydrogenase, FAD, FES. Complex 4, here we have cytochrome C oxidase. Here you should remember, students, complex 4 has a lot of copper centers. Copper center 3, copper center Cu A, Cu B, and A1 should come here, students, and A3 should come here. And A1 and A3. A1 and A3. Then we have F0, which is present inside, students. This is supposed to be like this. This is supposed to be F0. Entire thing is F0. F1 is this top part. Top part is F1. Okay. The F0 is completely inside. A small mistake here, students. Write down. F0 should be completely inside this, and F1 should be on top. Okay. I'll repeat again. FM and FES. Cytochrome BC1 complex. FAD dehydrogenase. Copper centers, that is cytochrome BC oxidase. Copper center A, copper center B. Cytochrome A1, A3, F0, F1. Now students, understand what happens here. In the starting, whatever NADH and FAD is produced here, NADH is sitting on complex 1. NADH dehydrogenase, complex 1 is sitting. Now this NADH1 undergoes oxidation. It is undergoing oxidation. It will lose all the electrons, lose all the protons. So losing the electrons, losing the protons. Now, these two H plus is transferred directly here. Then students, this particular complex is doing some work and it is pulling 2 more H plus from the matrix into the intermembrane, and that's how we get this 2 H plus more. So in total, 2 H plus by the NADH, 2 H plus by the find this complex here, protein complex, pulling from here. So in total, we get 4H plus in the starting here. We get 4H plus in the starting here, students. Now what about the electrons? The electrons are now sent to a mobile carrier called as UQ. Electrons are transferred to UQ here. Now this UQ also receives a proton from the matrix here, 2 protons. Students, this UQ is gaining the electrons and gaining the protons here. So can I tell UQ is undergoing, ubiquinone is undergoing reduction here. It undergoes reduction to form UQH2. UQH2. Ubiquinol. It becomes ubiquinone, becomes ubiquinol here. Now again, it again goes back to UQ. It is a cycle, complete cycle here. When this cycle is happening, students, from this, again we go back to this, right? When you going from UQ to UQH2 to UQ, this 2 protons is transferred here, and out of the 2 electrons, 1 electron is transferred here, because 1 more electron is required for maintaining the cycle here. So at the end of it, 2 protons are transferred to your complex 3, 1 proton is transferred to complex 3. Electron is transferred to complex 3. Now what happens this side? Here what happens, students? FADH2 commences here and it undergoes oxidation to produce FAD. Similarly, students, 2 electrons and protons are released here. Now you might ask me, students, why don't we transfer these protons directly into this, like how we did here? We cannot do that. Why? Because, students, this complex here, your FADH dehydrogenase, right? FADH dehydrogenase or succinate dehydrogenase. This particular complex is also involved in Krebs cycle. And this complex is not present. It is not a intermembrane protein. Okay, no complex. All of these students, it is actually supposed to be bottom here. Okay, it's supposed to be here till here. It is supposed to come down here. Okay, all of them are supposed to move down. This complex is specifically inner membrane protein. It is going to be, students, inner membrane protein here. It is going to be completely inner membrane. It is not be all the electrons and protons from here, they cannot transfer here. Okay, not possible to transfer here because it is a complete inner membrane protein. It is supposed to be completely in this membrane only. It cannot transfer here. So what happens, students? Again, we have a mobile carrier here called as UQ here. Now these protons are directly transferred to UQ this time, and obviously from here electrons are transferred to this UQ again. Now understand what happens in the UQ. What happens in the case of UQ? Okay. Now this UQ is gaining the electrons, gaining the protons here, and this UQ undergoes reduction to form UQH2 right now. This H2 from here is directly going here. Here it went directly here. Okay. Now this UQH2 now again gets converted back to UQ. It is a cycle. Understand, Purni? Understand. UQ is now getting converted back to UQH2 by gaining the electrons and protons. UQH2 again goes back to becoming UQ. When it again goes back to becoming UQ, it will release the electrons and protons. Now similarly, these two H plus is released here, and you see, and out of the two electrons that are entering inside, one electron is sent here, one more electron is used to maintain the cycle. So in a similar fashion, both the sides, 2H plus here, 2H plus here, one electron here, one electron here, both are sent to this complex 3 here, complex 3. Now students, can anyone tell me the chart right now, in total, how many electrons and how many protons are entering complex 3 right now, students? Entering complex 3? In the end right now, total 2 from here, 4H plus transferred here, and 2 electrons are sent out. 2 electrons are sent out. Now students, we need to transfer these 2 electrons to complex 4. So we in the middle, you cannot transfer this way, not possible. So again we take a mobile carrier right now. The mobile carrier is called as what? Cytochrome C. It will transfer the electrons from complex 3 to complex 4. Transferred here, done. Now what happens? Electrons are going up. Now students, all of you know, this right now is a aerobic reaction. But students, did you see anywhere involvement of oxygen anywhere till now? Glycolysis, no involvement. Krebs cycle, no involvement. Link reaction, no involvement. Then why do I call it aerobic reaction? We call this aerobic reaction because students, the involvement of oxygen is happening at the end, because hero is entering at the end here. The hero is entering at the end here. Now, the final electron acceptor is going to be oxygen here. Oxygen is receiving the electrons here, and it gets converted to 2HO, half O2 plus H2 will become water molecules, and water is released. So all of you remember the reaction of your respiration. In respiration reaction, CO2 is oxygen is consumed, and the byproduct of your respiration is nothing but your CO2 as well as the water, and this is the water which is released. All of you remember, right? What is released as a result of your respiration? The water is released here. Concept is connecting here, students. Now, when this happens, right, 2H plus is again sent from the matrix into intermembrane. 2H plus is again sent back. 2H plus is sent back again. So in total, students, at the intermembrane space, we have 4H plus here, 4, 4, 8 plus 2. In total, right now, in the intermembrane space, 10H plus are present here. In total, 10H plus are present here. Now, students, understand, this is the main concept here. We have 10H plus right now. Now, here, out of the 10H plus, every single time, 4H plus is going up. Every single time 4H plus is going up, we get 1 ATP formation is happening. 3H plus is used to rotate this F0 ring. 1H plus is used to bring the phosphate here, and that's how we get ATP molecule. That's how we get the ATP molecule, students. You can see here also complex 1, complex 3, complex 2, complex 4, complex 5. You can see 4H plus, 2H plus here. We can see here also all the FM and FES, cytochrome B, C1 complex, FAD, FES, copper center A, copper center B. Here you can see F0, F1, F0 and F1. And we get here every single time 4H plus is sent from the F0 to F1 side, 1 ATP is produced here. 1 ATP is produced towards the matrix side. Matrix. ATP is being produced. Students. Students, that is the concept here. That is the concept of your entire ETC. Intermembrane space here, inner membrane. Students, can all of you see here also? Students, is it visible to all of you? You can see here all of these proteins, complex 1, complex 3, complex 4. All of them are intermembrane. Proteins. Intermembrane complex. They are present in intermembrane. But if you look at complex 2, it is stopping right here. Can all of you see, students? It is stopping right here. Right? It is stopping right here. So electrons cannot be, protons cannot be transferred this way. It cannot be transferred, not possible. Okay, it is stopping. We can see this one, this one, this one. All of them are coming this side to the intermembrane space, to the intermembrane space. But this one is not coming. It's stopping here itself. It is stopping here itself, students. Okay. Half O2 plus H plus we get 2H plus sent here, and water is released as a byproduct here. Water is released as a byproduct here, students. And that is the end of the chapter, students. That is the end of the chapter from my side. Students, this you can study on your own. It's very easy. Okay. That is the end of, from my side, the entire chapter, students. End of the entire chapter. Done, all of you? Done? One more thing I want to tell you. Respiratory, this thing. The respiratory balance sheet. Let's understand here. From glycolysis, -2 in the preparatory, 2 NADH, but into 2. Why? Because everything is into 2. 3, 1 NADH will give you 3 ATP, 2 NADH will give you 6 ATP here, 4 ATP here. 6 plus 4 is 10 ATP, -2 is 8 ATP, -2 is 8 ATP here. Link reaction 2 NADH, 6 ATP, because 1 NADH gives you how many ATP? 3 ATP, 3 into 2, 6 ATP. From Krebs cycle, we get in total we get 6 NADH from 1 glucose. All of you know that, students. All of you know that. All of you should know that, students. We get 3 NADH in one cycle for one acetyl-CoA. For 1 glucose, we get 6 NADH. 2 GTP. Why 2 GTP, students? In one cycle, we get 1 GTP, but it is happening 2 times, because for 1 glucose, right? Glucose will happen 2 times, 2 GTP. And we know we get 1 FADH. We get 1 FADH for 1 cycle. For glucose, 2 times 2 NADH, 2 FADH. So 3 into 6 is 18. 2 GTP will give 2 ATP. 1 will give you 2 ATP. 2 is 4. So 24 ATP here. 24 plus 6 plus 8 is 38 ATP in total. 38 ATP in total we get here. 38 ATP we get in total, students. 38 ATP we get. Done, all of you? Done, students? Let me just call Gopika ma'am. See, students. We learn that this glucose is produced by the plants. We learn that oxygen is involved here. That is the final electron acceptor. We learn that energy is released here in the form of ATP. We know that CO2 is released outside, and we also saw that water is being released at the end. We also saw that water is being released at the very end of ATC, of the ATC, students. ATC. Then all of you, students, done. All of you know where CO2 is released. All of us are CO2 being released in your link reaction, as well as in the case of Krebs cycle. In Krebs cycle 2, here we have 1. But since it's glucose now, for glucose into 2. Okay, into 2, students. Krebs cycle 3, sorry, Krebs cycle 2 only. Krebs cycle 2 itself. So CO2 is released in your Krebs cycle link reaction. What is released at the ETC? Oxygen is used in ETC. Glucose is used in the first step itself. Glucose is used in the first step for glycolysis, and that is your entire chapter called as respiration implants. Entire chapter called as, students, respiration implants, students. Respiration implants. Okay. So I hope it was easy for all of you, students. Yes. Does it make sense to all of you, students? Does this reaction make sense to all of you now? Every single step I've told you where the reaction is released. Again, this also happens in ETC. Released in ETC. Released in ATC, students. Again, this water can be used for, non-cyclic again. Non-cyclic. To split the water. Now, in non-cyclic, when we split the water, what happens? Oxygen is released. This oxygen again can be used here again. It can be used here again. So that's how plant is so sufficient, self-sufficient, very independent plant is. Very, very independent, students. Very, very independent. Again, this oxygen is used here. And how do we get this glucose again? We get this glucose from where? Glucose from the entire dark reaction. This CO2 is released where? This CO2 is used in C3 cycle. C3 and C4 cycle is used again. Used in C3 and C4 cycle. Students, do you realize how self-sufficient a plant is? It can use everything so efficiently everywhere. Friends, again, now I'll give you a 5 minutes break and then ma'am will join. Okay. We'll just take a 5 minutes break. We'll start exactly at 6:50. Okay. Class resume at 6:50, students. Class resume at 6:50. Take a 5 minutes break. Just refresh your brain and then ma'am will come. Okay. Sure then, students. Take a 5 minutes break. Chill for 5 minutes and then ma'am will come.
Hi students. My god. From morning you're seeing me, no? 10 o'clock first thing you saw me, and before you go to sleep also you're going to see my face only, right? Yes. I'm so happy for all of you. First of all, let me take a moment to appreciate all of you, right? Because most of the time what happens, no one appreciates you people. So I'm going to appreciate all of you for staying back, okay? Happy to see you again. So I told you, no? Yeah. Okay. So I'm very, very happy for all of you for staying back, okay, for having that trust that yes, this session is going to change my entire revision game on plant and human physiology. And students, as a student who is aiming for AIMS, Government Medical College, 90 plus marks is like gold, right? It is not just for, it's not just a clickbait, like you click on it, right? It's not like that. It is actual that many marks, because 6 chapters, 3 marks, if you calculate also, it is more than that, right? Yes, exactly. Okay, yes, Ishi covers everything. Yes, Ishi covers everything. Okay, yes, I am also alive. You're all alive. I'm just happy for that. Okay. Head is paining, body is aching, throat is paining, but still we are here. That is all that matters, students. At the end of the day, I'll give you a big life lesson. Just keep moving, okay? Because I've learned this the hard way, be it in your work, be it in your family, be it with your career. Every time in life, there will be a pause that comes, like something will try to pause you, but just keep moving. Every single day, wake up, try to do something. So if the momentum is there, no, nothing can stop you. The moment we decide that no, I am giving up, I am going to be static, I'm going to stop, then over, students. I'm telling you, everything in the world can put you down. So this is a life lesson. Keep it, and remember this life lesson when you're facing the most hardest time in your life. Just remember that, okay, I just did this much today, but I am still doing something towards my goal. That's all that matters. Okay, it might be the smallest of writing a to-do list. Enough. You're still moving, instead of just sitting like this and be like, oh, leave it, yaar. Who is going to do all this? Okay, yeah. Yes, you have to rock. Okay, come on. Okay, let's start. Which chapter? Last chapter of mine. Okay, I am completing it. Okay, students, am I doing it faster? Yes or no? Tell me. Am I not doing it faster? Okay, faster does not mean we are excluding anything, we are including everything. Okay, yes, just keep moving. Okay, this is one thing I want all of you to follow in your life whenever you are facing the hardest challenge. Okay, be it NEET 2025, be it your counseling, be it your college, be it anything in your life. Just keep moving, just do, even if it's the bare minimum, do it, because that gives you the power to stand up. And I'm telling from a point where it's not like I had always, that golden opportunities for me. Never. Okay, it was always for me a hard call with many things. So I'm telling you in that way. Okay, listen it from people who are experienced. Yes. Excretion is simple but little long chapter. Okay, yes. Come on. Yes. Very nice. That's the energy. Yes. Okay. Come on. Students, I would request all of you, please wear glasses. Okay, please keep the screen at a distance, because you're sitting here for 8 hours. I do not want you to strain your eyes, and that is the main reason that we did the marathon today. Because if we make it on 2nd or 3rd or even 4th, like 4th early morning also, it is too much strain for you. Okay, now at least you are relaxed. You're like, okay, I have 3 more days, I can do some questions, I can like come back to this and revise once again. But it 3rd, even tomorrow also, I wouldn't request any of you to watch any marathons. Please stay away from marathon. Let this be your last marathon, because 3 days you need to really not strain your eyes. Okay, yes, I'm always there. Okay, students, don't worry, you people are the smartest, you people are the best people, best battle, warriors. And you, just one thing, whenever you're sitting wherever, okay, in the exam hall, okay, sometimes we will not be able to follow, but when you're sitting in the exam hall, think that you're the best, think that you're prepared at the best. the moment you have this positivity, you know, every answer will automatically come to you. Okay, the moment you start looking at another person and be like, oh my god, he's from this institute, oh my god, he's from this institute. Okay, institute has nothing to do with that student. Yes, every institute try their really best to make the student achieve, but it is up to the student. There are people who pay 2 lakhs, sit in an institute and use phone. So what's the point, right? Maybe you are doing better than that. So just think you are the best and write the exam best. Okay, come on, let's go. Motivation over. Okay, motivation ka stamina is over. Now we will start excretory product and elimination. Okay, let's excrete out all the negativity and let's take only positivity inside. So students, here excretory products: aminotelic, ureotelic and uricotelic. So ammonotelism means what? Ammonia is the most toxic. I've told you among these three, the red flag is ammonia, the green flag is that is uric acid, and the white flag. No, some people are not red, not green. Okay, they just change their colors. They are ureotelic. Okay, so the red flag, why I'm telling, because ammonia is the most toxic. There is no way that you can keep it in your body. You have to readily remove it from body, and you cannot wait for the ultrafiltration, reabsorption, secretion to happen. So what you will do, you will be like, okay, let's remove it from the body surface. Okay, and these are in organisms who have a lot of water around them, like bony fishes, aquatic amphibians and aquatic insects. Okay, yes. Okay, now uricotelic organism means, students, urea, it is less toxic, but it is not as less, like it's not like, see, all are nitrogenous waste, they are all toxic. Okay, among the toxic people, who is the red flag and who is the green flag, we are trying to find. Okay, we are those people who are doing that, starch test on paneer now. That's same way. Urea, less toxic, kidney will remove it. And students, if at all ammonia is there in our body, it will go to the liver, and the liver has ornithine cycle or urea cycle, through which the ammonia will be converted to urea. Okay, it is soluble in water. Some urea will remain in our body. Mammals, amphibians, we give out urea. Okay, uricotelic is the least toxic. Now you can ask me, ma'am, why didn't you choose a green flag? Why did you choose a white flag? Okay, because they can change color anytime. Secondly, not that, students, because even though this fellow is less toxic, problem is he's very expensive. He's very, very expensive. That means what? He's not like, not like have to take him to 5-star hotels or anything. But the thing is, this person will need a lot of energy to be expelled out. So we humans don't have energy to even stand and study properly. How will we expel out? Okay, how will we waste our energy on secreting uric acid? So this is the whole thing. Okay, yes, very good, very good. Okay, so that is the thing. And students, birds give out pellets. Pellets means what? Semi-solid paste. That is your uric acid. Okay, then again here they will ask you, match the following: excretory structure and the organisms. Okay, first excretory structure is protonephridia. Okay, protonephridia or flame cells. Organism is going to be platyhelminthes or flatworms. Nephridia, students, earthworms and annelids. Okay, Malpighian tubules, all of you know most of the insects, but we have studied it in our very own. Okay, cockroach, who is Rathis, very favorite person. Okay, Rathis and cockroach has a very, what is that? Celestial connections. Antennal glands or green gland, students, crustaceans like prawns. Okay, crustaceans like prawns. So this is the important thing. Protonephridia, you already know, platyhelminthes. Nephridia, annelida. Malpighian tubules, most of the insects including cockroach. Antennal glands, crustaceans like prawns. Okay, done. Yes or no? Now human kidney. Okay, kidney, you know how? Bean shaped structure, there is ureter coming out of it, then you have urinary bladder and urethral opening. Okay, on the anterior, that is on the superior part. Before in the NCRT, something else was given, no? Dorsal, something else was given. Now they made it into superior part. Okay, we knew it was in the superior part. I think they had given anterior part, if I'm not wrong. So now they have made it into superior part. Okay, so superior part, there is adrenal gland. Okay, adrenal gland is present, urethra is present. Now students, question can come about the numbers. Okay, some of the numbers, that is, a pair of kidney is there, a pair of ureter is there. Yes, correct. Now the pair of kidney has 10 to 12 centimeter in length. 10 to 12 centimeter in length, 2 to 3 centimeter in thickness and 5 to 7 centimeter in width, 120 to 170 gram in weight. Clear? Yes or no? Yes, clear. And towards the, this is a PYQ question. This is a PYQ PYQ question. What is the PYQ question here? Towards the inner concave, there is a notch. What is that notch called? Hilum. Okay, there's a notch called hilum. Okay, some of you were asking me, ma'am, how do we connect with you? Students, you can, like, if you want to see my classes and you want to get some motivation and all of that, you can join my Telegram channel, Gopika Biology Official. Not very difficult. My name is Gopika. I'm teaching biology and it is official. Okay. So Gopika Biology Official. And if you want to connect with me on Instagram, then it is Gopika Biology, where you can message me. But again, please do not expect my reply very soon. It will take 1 week of time because there will be lot of requests. Okay, and some of the requests will be just hi. Okay, they'll be hi ma'am, hi ma'am. So I will think that okay, something emergency I open, only hi will be there. So sometimes it will take few days for me to reply. Okay, clear to all of you. So towards inner concave, there is a notch which is called hilum, and to the hilum, students, remember the ureter, the blood vessels, at the same time the nerves also are coming in. Okay, the nerves also are coming in. Clear? Done. Okay. Oh my God, Ratis ran away, students, because I took the example. Okay. Yes. Now let's go forward and understand the cross section of the kidney. If I take a cross section of this kidney, how will the kidney look like? Look here, students. The kidney has cortex and medulla. First of all, understand, adrenal had cortex and medulla. Now kidney also have cortex and medulla. Ovary also has cortex and medulla. So there are many structures which have cortex and medulla. Cortex is the outer region, medulla is the inner region. Okay. So this cortex, students, is the outermost region, and inside that there is medullary pyramids. Okay, proper pyramid like structure, medullary pyramids, and this medullary pyramids open into calyces. Calyces open into a broader funnel shaped structure which is called the renal pelvis, and from the renal pelvis it will go to the ureter and go to the urinary bladder. Correct? Yes or no? Yes. Okay, correct. So clear to all of you, cortex, medulla, medullary pyramids, calyces, renal pelvis and ureter. Done. Okay. Now there is a renal envelope to protect the kidney that is made up of your adipose tissue. Okay, if you can remember, you remember, otherwise no problem. Now one more important thing is that there is a renal column. That means the cortex. Okay, imagine, students, this is the cortex. Cortex is moving towards the medulla. No, cortex is moving towards the medulla, and that portion is called as column of Bertini or renal columns. Okay, renal columns. Okay, I should slow down a bit. Okay, sure. So this is the cross section of the kidney. Okay, the kidney has a cortex region, a medulla region. Done. Now, these are the medullary pyramids. On the medullary pyramids, your nephrons are there. On the medullary pyramids, what is there? Nephrons are there. Okay. And students, this medullary pyramids open into one region called as calyces. And this calyces open into a funnel shaped, broader structure called as the renal pelvis. This much you understood. Okay. Now, remember, students, this cortex is trying to come. Okay. It is trying to move into the medulla. And that portion is called as renal column or column of Bertini. Column of Bertini. Clear to all of you? Yes or no? Yes, done? Okay, perfect. Now, reddish brown color, students, reddish brown kidney is there. Bean shaped structure. Kidney is divided into cortex and medulla. Located between, yes, important. Where is the kidney located? Between the T12 and lumbar 3. Okay, 312 and lumbar 3. Yes? Don't get scared. Everything will be fine. Okay, T12 and lumbar 3. Projections called calyces, that is minor and major calyces are there. Students, that calyces is there, no, where the medullary pyramid is going to open. That will be major and minor. Major, minor. Major, minor. Okay. Major, minor calyces. Each kidney will have 1 million nephrons. Done, pakka. Did you all mark your attendance as well? Yes. Done. Yes. Clear? You can see my dark circles coming till my nose. Right? Mornings. Okay, morning there was dark circle, less dark circle. Now here this much will come, students. I think by end of next year NEET examination, I'll look like that panda only. Did Ratis really send panda symbol thinking something? Huh? Students, should we boycott Ratis? Or should we just let him go? Okay? Done. Okay? Sometimes we have to talk. Okay? Full time if you study, that's all. And gone. Brain will be like, what is this lady doing? For 1 year she didn't study and now she's eating my head in 1 day. Yes? Clear? Yeah. Since Pakka students, next year I'll look like one panda because of my dark circles, okay? It's coming till here now. So it will come till here. Yeah. Okay. Yeah, we are all having it, so it's fine. Okay, the struggle is real. The struggle is real. Okay, come on. Yo. Come on. Yes, he's not able to tell anything to me, so he's scolding you people, okay? Yes, come on. Look here. This is your entire nephron. Okay, nephron. So this is your entire nephron, students. Nephron has mainly two parts: glomerulus and renal tubules. Okay, glomerulus and renal tubules. So your glomerulus is one portion where this tuft of capillaries is called glomerulus inside a Bowman's capsule, and then there is renal tubule, students. That is PCT, loop of Henle and DCT. Okay, this PCT is proximal convoluted tubule, highly convoluted region which has your cuboidal epithelium, brush border cuboidal epithelium. What is there? Brush border cuboidal epithelium is there, and this will run down to loop of Henle to DCT, that is distal convoluted tubule, and it opens into the collecting duct. Now all of you tell me which is the region that is not present, not a part of the neuron, nephron. Students, when you teach so much human physiology, you forget. Tell me, students. Yes, tell me. Brush border, okay. Tell me which is the region that is not a part of the nephron. So if I tell neuron by mistake, just think that man's brain is glitching. Okay? Yes. Come on, quickly answer me. Quickly answer. Fast. We have to finish. Okay, we have to finish. And students, whoever stayed from the starting, I know almost all your names, whoever stayed, I want everyone to comment that, I stayed for this long and I am assured of this much marks, okay? Because that shows your real power. Okay, when other students open and see that, no, they'll be like, my God, yeah, this girl, I am competing with this girl and I am competing with this boy. My God, I really need to prepare. So I want every single person in this, in this life to comment and only go. Not for me. Comment for yourself. Comment for yourself that another person thinks that you are a competition. You are a tough competition because you are here. Okay? Will you do that? Will you do that? Okay, so come on. Yes. Now students, this is clear. So collecting duct is not a part of your nephron because collecting duct, remember, another nephrons also open into the collecting duct. Now students, glomerulus has afferent arterioles, efferent arterioles, all of you know. Efferent arterioles are going to again break down to peritubular capillaries. From peritubular capillary, this is going to further divide to form vasa recta, which is surrounded near the loop of Henle, which is going to help in countercurrent mechanism. Countercurrent mechanism. Correct? Clear? Thank you, Aarti. Okay. Clear? Yes or no? They won't put nazar. Okay, we will protect. Okay, no nazar. Okay, no nazar, only positivity. And students, even if they put nazar, it's not going to work in 3 days. Okay, and nazar will only work when you're traveling or something and you're posting that I'm going to Thailand, and then they put nazar and it gets cancelled. Okay, education. No one can take education, nothing out of you. Yes, look here. This is your this is your medullary pyramid and this is your nephron, and the loop of Henle is dipping into the medullary pyramid, students. And now understand there are two types of nephrons depending on the size of loop of Henle. One is called the cortical nephron, next one is called the juxtamedullary nephron. Cortical nephron, students, it works during the normal condition, that is the loop of Henle is short, vasa recta is not, it is negligible or it is highly reduced. Okay, highly reduced. Clear? Understood? Now, look here. Loop of Henle, students, one more is the juxtaglomerular nephron, where the loop of Henle is long, and when the loop of Henle is long, obviously vasa recta also will be long. And we also know one thing, that countercurrent mechanism works exactly during stress condition. That is, they are going to work when we are not drinking enough water, we have not had proper food. During that time only this will happen. Okay? Yes? Clear? Clear? Okay. Yes. So this is done for all of you. Now, very, very important students, JG apparatus, okay? Juxtaglomerular apparatus. Sorry, juxtaglomerular apparatus. Why did I write medullary apparatus? Now I'm seeing students, you people also didn't tell me. Juxtaglomerular apparatus. Okay. Now, juxtaglomerular apparatus, how does it work? Very simple thing, students. If the glomerular filtrate has some problem, then the filtrate is going to move right all the way through the tubules, and the moment they reach the DCT, distal convoluted tubule, if there is any problem with the filtrate, there are sensory cells which are called as macula densa, which are present inside the distal convoluted tubule. Just imagine there is a toll. Okay, there is a toll, and they notice that this car is very suspicious. Okay, because all of you are sitting and studying. So they'll be like, wait a minute, this car, people have some problem. They're very, they're very suspicious they are. So they will inform another person telling, you know what, there's a suspicious car coming, you please take care of the rest. Same way here also, when the glomerular filtrate is coming, the moment it reaches the DCT, the macula densa cells or the sensory cells will sense a problem with the filtrate. Immediately they will tell the JG cells, which are a modification of some of the smooth cells of afferent arterioles, that you know what, there is a problem. Immediately JG cells will be like, it's okay, let's release renin. Okay, let's release renin. Clear? Yes? Clear? Okay, they will release renin, and that renin will continue the RAS mechanism. Students, I understand if the brain is not braining, it's okay. Just let your brain just listen whatever is possible. Rest, it's okay. Okay, I do not expect your brain to work also for this many hours. Okay, that will be so unfair to make the brain work that many hours. Okay, yes. Now this is your urinary bladder. I will only tell you the main point here. Trigon of bladder. Okay, trigon of bladder is basically this one, where the two ureters are opening and one urethral opening is there. And students, the urethral spinsters, question have come about urethral spinsters. Please remember, urinary bladder has to contract, and the urethral spinsters have to relax. Okay, imagine you want to use the washroom, right? You want to go to washroom exactly in the maths period. So your urinary bladder was 500 fill. Okay. First you will go and ask ma'am. Ma'am will tell no, go sit there. Now only lunch break got over. Then you will start doing Bharatanatyam. Why do you start doing Bharatanatyam? Because the urinary bladder is getting full. It has got 600, 700, and after 700, Kathak, every kind of dance will come in your automatically in your body. Why? Because you're not able to control. Exactly what is happening, students. Urinary bladder is contracting and external spinster muscle, that is your urethral spinsters, are relaxing. Internal spinster has already relaxed. External spinster is in your Bharatanatyam control. Okay, your dance ka control, it is standing, but anytime it will open. Okay, because the pressure cannot be caught. Okay, clear to all of you? Okay, yes. So if the stretch receptors is not functional, students, then obviously there will be leak of urine, which we see usually in old people. Okay, old people. Clear to all of you? Yes. Urethral spinsters. Okay, urethra and ureter is different. This is ureter. Okay, ureter. This is urethra. Okay, tra and tra. Please my tongue is twisting by itself. I told only once, but my tongue only told 3 times. Okay, I want to finish this class before I don't tell some word that does not exist. Okay, yes. So compound epithelium, students, transitional epithelium is present here. I've told you this in structural organization also, because they are present in the urinary bladder, ureter. Okay, they are called as transitional or urothelium. Okay, urothelium. Understood? Clear? Come on. Now urine formation. Okay. Yes. Yeah. My tongue is doing by itself. Okay. The special dance. Okay. Wow. Entertainment has happened. Okay. Yazini is the entertainment for the day. Okay, come on, students. Okay, ignore all the dinosaurs like we ignored them 65 million years ago. Okay, yes, come on. Urine formation. Those who understood, put laughing emoji. Those who did not understand, you should try to decode it. Okay, Urine formation happens in ultrafiltration or glomerular filtration, reabsorption and secretion. Okay, ultrafiltration, reabsorption and secretion, students. Ultrafiltration means it happens in the glomerulus, so we can also call it glomerular filtration. So this is the first step. Okay, I know the photos might not be in maximum clarity, but you have the original notes with you. Okay, you can look into that. So this is your glomerular filtration, where the tuft of blood is coming and filtration is going to happen. Second step is going to be tubular reabsorption, and third step is going to be tubular secretion. Okay, tubular secretion. So students, remember 1000 to 1200 ml of blood will be taken. That is 1/5 of the blood. Okay, 1/5 of the actual flow of blood is there. No, 5 liters, right? That much blood we are going to take it for filtration. Okay, we are going to take it for filtration. 125 ml per minute and 180 liter per day. But do you actually give out 180 liters? No, you only give out 1 to 1.5 liters. Everything else will be reabsorbed. Okay, everything else will be reabsorbed. Clear to all of you? Yes or no? Now, here main thing, glomerular filtration. That is how we study respiratory membrane. Same way here also we have some layers to it. Okay, layers to it. Clear? Okay. Yes. Okay, now look here, students. Glomerular filtration, endothelium of glomerulus, basement membrane and epithelium of Bowman's capsule. There we studied epithelium of the alveoli, endothelium of the capillaries and basement membrane. Here also similar pattern, but they're just that side, this side. Okay, so here we are doing endothelium of the glomerulus. Again, remember this is also a capillary, so we talk about endothelium. Then we have a non-cellular layer, which is the basement membrane, and then we have an epithelium of Bowman's capsule, which is the cellular layer. So these three layers are going to help us in the movement or filtration, like a sieve. Like how we make tea and we sieve it, no? Like that only, the same thing is going to happen. Now understand, students, endothelium is there from the glomerulus, then there is basement membrane, and this epithelium, no? This epithelium of Bowman's capsule has this podocyte cells. Podocyte cells, which are arranged in an intricate manner, and in that intricate manner they are going to have the filtration slits. Okay, filtration slits. Can you see here? I have made the filtration slit or the slit pose. Clear? Yes. Layer same as blood alveoli. Yes, very good. Okay, very good. So these three are going to help in the glomerular filtration or ultrafiltration. Why is it called ultrafiltration? Because almost all the particles will go out of it except for the large proteins. Okay, large proteins. Now, look here, students, this arrow mark I have explained to you last time also. Reabsorption. Okay, reabsorption means what? Okay, students, let's take this. Okay, what is this? Okay, let's take this. Okay, this is the tubule. Okay, this is the tubule. And wait, I have tools. Okay, I have tools and weapons and everything. Okay, to teach you people. Okay, look here. Let's assume, okay, let's assume this is the tube. Okay, this is the tube in between this, and this is our body. Okay, this is the body, this is the tube. Okay, we can open it or close it, it's all make the sense. Now, look at the arrow mark. I told you this is the tube, this is the body. Now, reabsorption means what, students? This is also body, this is also body. That's why I opened and kept it. Okay, I opened and kept it. So, to show you like this. Okay, this is the tube. Okay, this is the body, this is the body. Reabsorption means what? Reabsorption means what? We are, whatever is not required for our body, we are giving it. Okay, we are giving it, that is the tubules are giving it to the body. Okay, now secretion means what? Whatever our body does not require. So here, first scenario, whatever the tubules feel is useful, they are giving it to the body. Okay, reabsorption, they are giving it to the body. Secretion means what? Whatever the body does not want, it is giving it to the tubules to excrete it out. Understood? Simple. Okay, simple. So, reabsorption means whatever the body requires, we are reabsorbing, giving it to the body, and whatever the body does not want, it is giving to the tubules as tubular secretion. Words might be confusing, but the arrows will indicate it properly. Okay, and students, remember secretion, NHK, you remember, like NH47, not NH47, no, what is that? AK47, not that. National Highway, NH7. Okay, NH7, like that only you remember NHK. Okay, NHK, that is national, not national highway, students. N for ammonia, H for hydrogen, K for potassium. My brain is going to give up any minute from today. No, sorry, from now. Okay, now secretion means what, students? Active secretion is going to be gang. Okay, gang. That is a new word. I think it's a gaming word or something. Gang. Okay. G for glucose, A for amino acid, N for sodium, K for potassium. These we will require energy to remove it out. Passive is nitrogenous base, water and HCO3. Okay. HCO3. Clear to all of you? Clear to all of you? RAS mechanism. Okay, RAS mechanism, very simple. Renin, angiotensin, aldosterone system. Now, this is what happens, students. I told you. If there is a problem with the toll, okay, if there is a problem with, let's say, the car, the toll people will inform there is a suspicious car coming. Please check the car. Okay, so same way here also. The JG cells release renin. What is renin going to do? It is going to convert angiotensinogen, which is in the plasma, which is inactive, to angiotensin 1. But we will ask angiotensin 1, can you actually do something? He'll be like, no, I am not the superior. You need to call angiotensin 2. So, but then we will realize that only renin can do this much. Then we will take the help of angiotensin converting enzyme from the lungs and we will convert it to angiotensin 2. We'll ask angiotensin 2, can you do anything about these criminals? He'll tell, yes, I can do, I can fix everything. Okay. So what is he going to fix? First, he is going to activate adrenal cortex or stimulate adrenal cortex to release aldosterone, which is going to do conditional reabsorption of water and sodium. Okay. Then angiotensin 2, again it will do vasoconstriction. Why? Because they are complaining that the GFR is less. Okay, GFR is less, blood volume is less, water is less, BP less. So to increase the GFR, that means to increase the blood flow, we will reduce the blood going out. Okay, so vasoconstriction of efferent arterioles. So RAS mechanism, students, renin, angiotensin, aldosterone system. Whenever there is a problem with the filtrate, the macula densa or the sensory cells in the DCT will send signal to JG cells. JG cells will release renin. Renin will go convert angiotensinogen, which is in the plasma, to angiotensin 1. Angiotensin 1 will be converted to angiotensin 2 and that angiotensin 2 will release aldosterone to do conditional reabsorption of sodium, which will increase the BP, conditional reabsorption of water, which will increase the water as the blood volume, and who will also do vasoconstriction of efferent arterioles, so there is more GFR. Okay, yes, clear, clear. Okay, good. Next is countercurrent mechanism, students. Here what is going to happen? So countercurrent mechanism happens during stress situation. Okay. Now this is your ascending vasa recta and this is your descending vasa recta and this is your loop of Henle. This is the blood vessel. Okay. Very simple way I'll tell you. Questions will not be complicated from here. Okay. Questions will not be complicated from here. 300 to 1200 ml, the osmolarity is going to increase. What we are going to do, students, this process is mainly done so we can increase the concentration of urine. Clear? Increase the concentration of urine. So here what is happening? Remember the water. Okay, students, remember this DWAE. Please remember these two letters, DWAD. Descending loop of Henle is permeable to water. Ascending loop of Henle is permeable to electrolyte. Okay. So, here you have your loop of Henle. Done. Correct. Here you have your blood vessel. Done. Okay. Now, the descending loop of Henle the water will come out. The water will go all the way. It will be secreted out. But how does this happen? This happens because the NaCl, okay, the NaCl which came out of this, okay, will be accumulated in this interstitial medium. And also the urea which is taken from the collecting duct also will be accumulated here, leading to a hypertonic solution. If an hypertonic solution is there, you all know the water will start coming out. If the water start coming out of the tubules, that means urine is automatically getting concentrated. Yes or no? Yes or no? Correct? So that is possible. Okay? Clear? Yes or no possible? Yes, very good. Descending loop. So students, here in the interstitial fluid, we are trying to make an increase in the concentration of salts. NaCl also will be there. Okay, because here NaCl will come out. Okay, this will be taken up. Please remember, this will be taken up and it will be given here. So this will automatically increase the interstitial fluid concentration. that interstitial fluid concentration will become hypertonic. Automatically water from the tubules will start coming out and making the urine concentrated. So the question will come in such a way, if loop of Henle is absent, then what will happen to the urine? Please tell the urine is going to be diluted. Urine is going to be diluted. They will not directly ask you what is the osmolarity. They will ask you like this kind of questions. Okay, like this kind of questions. Clear? ADH and ANF is very simple. ADH, you know, vasopressin, anti-diuretic hormone. So it is going to prevent diuresis. Okay, that is loss of water will be prevented by conditional reabsorption of water and also exogenous, that is thirst mechanism. Okay, which reminds me to drink water. Okay. Yes. Okay. ANF students, anti-natriuretic factor, which actually works opposite to RAS mechanism. That is, RAS will do GFR more, ANF will decrease the GFR rate. Understood? They work antagonistic in nature, otherwise they will not be balanced. They will not be balanced. Clear to all of you? Yes or no? Yes or no? Now, students here, understand, in disorders you have to study dialysis. Question will come from dialysis. Let's try to understand dialysis. This is a cellophane tape. Okay, this is a cellophane tape. Let's say you want to make dialysis machine. Okay, just like a practical, what is that called? Yeah, science exhibition types. Okay, you made a cellophane tape and there are pores on the cellophane tape. Now, what is high in your blood? All of you tell me what is high in your blood. Okay, that is not supposed to be there in this dialyzing fluid. Can anyone tell me what is high in your blood but is not supposed to be there in this dialyzing fluid? Ma'am, obviously, ma'am, nitrogenous base, urea is high. Okay, so students, when we make this entire setup, this dialyzing fluid will not have any urea. No urea. No urea. Why? So that urea will move from a region of higher concentration to a region of lower concentration and automatically the blood will be purified and given back to the person. So when we are taking the blood, we are going to add heparin, okay, anticoagulant, and when we are giving back the blood, we are going to add anti-heparin because the body has its own heparin. Released from where? Can anyone tell me heparin word? Where did we study today? Quickly tell me, where did we study today heparin word? Okay, come on. Students, renal failure, 2 kidney dysfunctions, you have to do dialysis, 1 kidney functions, no problem. Permanent solution is definitely going to be kidney transplantation and we have to take it from a close relative because graft rejection is going to happen and we have to give immunosuppressant. Excellent, 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 Rani. Basophils, we have studied it in basophils, okay. Basophils, we have studied about heparin. At the same time, yes, mast cells also we have studied this one. Okay, hemodialysis students, dialysis, that is many of them ask me a doubt, ma'am, if these 2 kidneys are not working, how will we attach the 3rd kidney? So, in 3rd kidney will be directly attached in such a way that it is connected to the urinary bladder. Got it? Clear to all of you? Yes? Okay, done. Okay, I am done with the chapter. Now, Basavaraj sir will come.
Well, ma'am?
Done.
Perfect timing.
23 minutes I completed a chapter. Students, more than 23 minutes, no? I think 30 minutes.
30 minutes.
Yeah, but 30 minutes, I thought it is impossible, but yeah.
Everything is possible, ma'am, now.
Exactly. Because they know the answers, no? It is faster.
It's exactly. Since they've been preparing with us for almost a year now, right? I'm getting little allergic reaction. I don't know why. Something. I started sneezing here a couple of minutes back continuously.
The filtrate is not good.
Yeah,
It's
I
not
think
clean.
the suspended particles would have come.
Yeah.
Wow. Student, ma'am, I'll finish it in 30 minutes. Right?
I changed your name. I changed everyone's name, gender. Okay?
Do you have any bandage? What is it?
Did you have bandage? I can't eat it, no?
Are you eating bandage now, ma'am?
If people make a wound in my heart, then yes, without writing it properly. You're very late. Go back to sleep.
I think 9 hours is done now, ma'am, I believe, right?
9.5. 9 hours, 24
So probably I'll take
Yeah,
30 minutes or
30
10 hours.
minutes.
So just say 10 hours, ma'am. Just say 10 hours.
And sir, it's amazing. 9 chapters in 10 hours.
Oh,
It's not
crazy.
like we're doing 3 chapters in 10 hours.
Oh, God.
Yes. And students, we are coming to Chennai. So when are we coming to Chennai?
6th.
Yes.
I
We're coming
remember.
to 6th. Astram, we are coming to 6th.
We are coming to Chennai on 6th.
Students, we are coming to Chennai on 6th. And which center?
Anna Nagar Center.
Do you know Anna Nagar? Wherever you are.
Do you know Anna Nagar?
I know. Students, they'll drop me there. They'll deliver me exactly in the
They'll pack ma'am. Yeah. They'll pack ma'am nicely. They'll send ma'am there.
Okay.
Oh God.
So, yes. So, they will drop me exactly in front of Anna Nagar. So, I will get inside. Okay. You people come to Anna Nagar. Okay. If any of you are going to miss after being in Chennai, I'm telling you, it's your loss.
Yeah.
Okay. Next year, we will not come to Chennai. Okay, we'll go to another place, sir. Vizag will go because many students have asked Vizag.
Why is
okay?
it?
Yeah.
We'll go to
Vizag.
Yeah.
Not Vizag, Vizag. Okay,
I'm
Vizag
like,
and Hyderabad. Hyderabad 5 centers are there, students. I think Shreyas sir will give you a clarity on that. Which center we are going. Again, they will deliver us there, so we will just walk in. So you people should know the location.
Students, you cannot see it from. I can see ma'am face has become this tiny now.
Yeah.
After all the teaching, ma'am face has become this much tiny.
Students, I took 6 chapters. Do you realize?
Ma'am.
Okay, 6 chapters. Sir took an extra crash course.
No, I did not take.
So that means sir is on the upper side. So I'm on the little. Wow, such a beautiful plan.
Nice, no? I'll show you one more. See, this is what I.
This is why I don't appreciate sir. If I tell one thing, he'll tell me 2 things.
I'll show you, ma'am. I'll show you. See this. Oh, sorry. First time. I'll show you more plants, ma'am. See this. This is more beautiful.
Actually, I'm going.
Wait, don't go. See this.
Hey, body fluids and mechanism. Yeah, your mechanism is upside down. Body fluids and circulation.
Look at this plant. It's like us right now.
Oh, that is me. You know, this students have put panda's face, okay? I thought seeing my dark circles.
Look at this.
Yeah, good.
Yeah. I'll show you one more now. Wait. This is where I'll be starting right now. Last chapter.
So do fast, okay?
Ha, ma'am. Don't worry. Anyway, I'm talking. My normal speed itself is 2x speed. Normal talking speed. So.
Yes. Why are you people so obsessed with Priyanka Chopra? Huh?
Yeah.
Yes.
No.
Okay. Quickly smash the like button, okay? And I want every one of you to comment that you sat for 10 hours. People who are coming in the last minute and commenting, 10 hours I sat. I know your names.
Yeah, we know who sat for 10 hours, who sat for 10 minutes.
Yes, who sat for 10 minutes, we know, and we know who came in the last 10 minutes also. Okay?
Yeah.
So
we remember.
Yes.
Now, ma'am, I'll start last chapter. Growth and growth.
Students, growth and reproduction, new chapter. If NTA is watching this video or NCRT is watching this video, new chapter.
This is what happens.
Growth and reproduction.
See,
I
was talking about unicellular organisms. You know, in unicellular organisms, every single time they grow, they reproduce, right? So, try to justify. Oh God. Okay. Now, students, let's just start. Let's just start with the chapter, students. Let's just start with the chapter. I don't think I can sugarcoat anything now, students. I'll teach you on point what you need to study, what you don't have to study. On point, we'll go right now, students. Okay, I'll give a quick example. Let's say, who should be next, ma'am? Tell one name, ma'am. We'll take one name right now.
who came very early, Priya Shakti.
Priya, okay. Students, let's say Priya, when she was in 4th standard, she was 4 feet tall. Now the Priya is 6 feet tall, students. Let's say Priya right now is 6 feet tall. Students, is that growth again? Can anyone tell me, students, is that growth? From 4 feet tall, she became 6 feet tall right now. Is that growth? Yes, it is growth. Because you cannot change back. You cannot hit on her head and make her go back to being 4 feet tall. No. It is permanent, irreversible change. That is called as a growth. Now take me for example. Last year, I was 60 kg. This year, I am 67 kgs now. Is that growth again, students? Is that growth again? That is not a growth. Why? Because let's say I start doing something called dieting, which is never possible. Okay. Let's say I start doing dieting and I don't eat any junk food or I don't eat any type of oily food. I can reduce my weight back to 60 kgs. So that is not growth. Me being reducing my weight is not growth because it is reversible in nature. Okay. So growth is supposed to be, students, growth is supposed to be completely, it's an irreversible permanent change. Growth is, students, what? Irreversible. Students, Imagine Priya is sitting on oxen or she has oxen on her head, then she will keep on growing. But right now, she doesn't have oxen right now. She doesn't have oxen. Okay. So remember students, growth is irreversible, permanent change right now. Growth can be intrinsic or extrinsic. Difference you understood. Difference. Intrinsic is from the inside, extrinsic from the outside right now. Extrinsic is from the outside right now, students. Okay. Now students, what about a guard cell? This is a guard cell right now. When the water is entering the guard cell and the guard cell is swelling up, students, is that growth again? Can anyone tell me, is that growth right now? It is not growth, because the guard cell can become flaccid again when the water is lost. So it is not growth. It is not growth. Now students, I am trying, I don't know why. I can imagine one human with an oxen on the head like a cap, and the person is growing now because of oxen. Okay. The apical dominance, no apical dominance. Now here is an example, students. Here is an example of your growth. That is expansion of leaf. Now let's say this leaf is expanding right now. It is growth now. Again, this leaf cannot go back to being a small bud. Not possible. It is the energy required process, and more anabolism is happening here. More of anabolism is happening right now, students. Okay. Now students, in the case of seed germination, it is also growth. Seed germination is a type of growth here. That is, seed is germinating. That is also called as growth here, students. Growth. Now students, question will come from here. Students. Okay. How will the question come? Understand. They'll say a line, growth is supposed to be indeterminate or unlimited. So plant growth is said to be indeterminate or unlimited. True or false? Friends, tell me in the chat, true or false? Plant growth is said to be unlimited or indeterminate. Very true, because plants can keep on growing throughout their life. Unlike humans, which will stop growing after some time, vertically, horizontally we can grow. Okay. So that is called as unlimited growth in the case of plants here. Plants can keep on growing because of meristem. Students. Now, plant growth is supposed said to be open. What is the meaning? Open means, students, new cells are always added. That is the meaning of open here. Plant growth is localized. It means that, students, growth happens wherever we find meristem. For example, we can find shoot apical meristem as well as root apical meristem here. Shoot and root apical meristem can be seen here. In the case of animals, it is uniform. That is, you will not find one human with long head human. Not find. You will never find a human with long arms, one small arm, one long arm. Not possible. Uniformity growth is seen in the case of animals. Except that one person in Bahubali film, you have seen Bahubali. One hand is like this, one hand is very big. But that is a disorder again. That is a disorder. Now, in the case of plant, students, number of organs keep on increasing. That is, leaf and flower will keep on increasing also. Leaf and this also will keep on increasing also. So plant growth is said to be open, localized, and it is said to be indeterminate or unlimited growth. Okay. Now students, what exactly is growth right now? Growth is nothing but, let's say this is one cell, and this cell is becoming bigger. It is eating all the food in the world, and this cell is becoming bigger. But what actually is the growth here? The growth here is the growth in cytoplasm or the protoplasm here. That is, increase in living part of the cell called as protoplasm. So protoplasm is actually increasing right now. But the catch here is, students, what is the catch here? This right now is very difficult to measure. Not possible. Very difficult to measure. So we have certain alternatives. See, for example, there are multiple ways to test your knowledge. Writing exam is not the only one. We have street smart, we have book smart. Students, tell me in the chat right now, are you book smart or street smart? Students. Okay. Students, quick tell in the chat right now. Are you street smart or book smart? In the chat. Both are different type of smartness. Right. So here we cannot really measure the protoplasm. So we find certain alternatives. What is the alternative? Here is the alternative. Increase in cell size, increase in cell number are the two examples here. For example, one watermelon cell can grow in 3.5 lakh times. One maize root apical meristem can grow in per hour around 70,500 times. Increase in fresh weight and dry weight can be seen. Increase in length, that is pollen tube growth. Increase in surface area, leaf can increase. Then increase in volume. So all of these, students, all of these are right unsmart, not smart at all. So that is a different category. That is a different category, unsmart. So all of these are called as different criteria to measure the growth apart from the protoplasm. Students. Now there is something called as phase of growth. Like how we have kindergarten, then we go to primary, then we go to high school, then we go to college, then we go for job. So you have in your life also, you go to different levels. So in the case of plant life also, it is plant cell also, there is 3 different phases. Meristematic phase, elongation phase, maturation phase. Okay, now 3 different phases here. The first one is called as meristematic phase, is the first phase where the plant is entering. The cells are entering here basically. Here cells are very small size, dense cytoplasm, vacuoles are absent here. Why vacuoles are absent? Because vacuoles are required to store something. If they imagine there is nothing to store, why do even they need vacuoles? So they don't need vacuoles here. Conspicuous nucleus means very prominent, well-known nucleus. Cell wall is primary here. Multiple plasmodesmal connection can be seen here. Multiple plasmodesmal connection. What is the meaning of that? Understand. Let's say I have one cell here and one cell here. And both of these are plant cells. Both of these are plant cells here. Plant cells. Now between the plant cells, students, here, there is a cytoplasm is connected here. The cytoplasm of this cell is connected to this cell, students. That is called as plasmodesmal connection. I am still in kindergarten. I am also in kindergarten then. This is called as plasmodesmal connection right now. That is, cytoplasm of this cell is connected to cytoplasm of this cell. This can be mainly seen in your young plants. Now, can anyone tell me, students, what do we have here, right in the middle? Here, what do we have, students? Here we have something called as middle lamellae. And what is middle lamellae made up of? Can anyone tell me in the chat? What is middle lamella made up of? Calcium pectate and magnesium pectate, students. Okay. Now, elongation phase. The cell length increases here. Vacuoles are present. Secondary cell wall formation happens here. Depositions. Maturation. Cell wall has lignin here. Cell is in protoplasm will grow. Vacuoles will retain maximum size. Will become maximum size. Okay. Let's see. You can see, students. This, if for example, root. The starting part is called as meristematic. Cells are very young, dividing, exploration. Elongation phase, increase in root length here. Then we have maturation phase here. Maturation phase. Okay. Students, can anyone tell me what is the small structure here? Anyone? This small beard-like structure here. Anyone, students? What is this beard-like structure here? This is called as root hair. So root hair formation always happens. Root hair formation always happens on the maturation, region of maturation, students. Root hairs. Okay. Now, students, now. Now, there are 2 types of growth. One is called as arithmetic growth, one is called as geometric growth. Understand, very simple concept here. In arithmetic growth, what happens? In arithmetic growth, one cell will divide in 2 cells. When it is dividing in 2 cells, one cell will remain meristematic. One will become permanent here. Again, if this cell divides, one is meristematic, permanent. This cell is dividing, meristematic, permanent. So every single time in the case of arithmetic growth, a single cell divides into 2 cells. One is going to remain meristematic, one is going to become permanent here. Okay. Now, what about geometric? In the case of geometric, students, in the case of geometric, what happens? A single cell, when it divides in 2 cells, both of them will be what? Both of them will be meristematic in nature. Both will be meristematic in nature. That is called as your geometric growth. That is called as, students, what? Geometric growth here. Now, this is the graph for arithmetic. This is the graph for geometric here, students. Geometric. And you can see, students, here we have a lag phase. Then we have a log or exponential phase. Then we have asymptote. Students, can anyone remind me, students, such graph, where else do you see? Do you see such graphs in the case of bacterial growth? Do you see such graph in the case of your logistic growth model? Logistic growth model, remember, in your ecology and ecology. Logistic growth model. Yes. Now, students, here sometimes they might ask you the entire, what is LT, what is the entire equation here. Very rarely they might ask. But here, students, what is more important here is that where do you observe arithmetic and where do you observe the geometric? Students, understand, all of you. Students, understand. You will notice arithmetic growth always in the root elongation. Root elongation. And what about geometric? Geometric is generally observed in all the parts. That is geometric growth. Happens in all the plants in general. It happens in the plants in general, students. Students, quickly smash your like button. All of you. Now, students, this is a curve. See the question, students. See the question. A typical growth curve is, typical means in majority of your plants. In majority of your plants, which type of growth curve do you see? In majority of your plants, which type of growth curve do you see? Okay, I know students, I just now told you, we see the sigmoid curve. We see the sigmoid curve. Okay, this is just sigmoid. This right here is sigmoid right here. Sigmoid. And this is called sigmoid curve or geometric growth. Okay, sigmoid curve or geometric growth, students. Okay, now there are ways to measure. Now, what is ways to measure a growth? For example, I told you, let's say last year I was 70 kgs. Last year I was 70 kgs. This year I am, let's say, 65 kgs. How many kgs did I lose, students? That is not growth, that is the reverse of growth. Okay, let's say last year I was 70, this year I am 80 kgs. Last year I was 70 kgs, this year I am 80 kgs. How much is the growth, students? 10 kg is the growth. 10 kg is the growth here. So you do here, that is called as your absolute growth. In absolute growth, what do? We do something called as final minus initial. We do something called as final minus initial, and we get the value. 80 minus 70, 10 kgs I increased. I increased by 10 kgs. Okay. Now students, now what is going to be your relative growth? Students, remember students, relative growth is increase per unit area. This one is much more better here. Understand why, students, understand why. Let's take Subra here. Her pocket money, let's say, was 5 rupees. We increased from the 5 rupees to 10 rupees this year. Let's take Chintu, nice name. Okay, Chintu's pocket money was 50 rupees. It increased to 55 rupees. Now, who is more happy, students? If you see the increase in pocket money, in both the cases, it is 5 rupees increase. Tell me yes or no. In both the cases, right? In both the cases, 5 rupees pocket money has increased. But what is the reality, students? Reality is different here. In reality, what is happening, students, here is that we do something called as final minus initial divided by initial into 100. And we notice that here, for Subra, it is around 100% increase. But for Chintu, it is only 10% increase. The pocket money may be now 55 rupees. But here, more happy students. So all of you understood? This is called as relative growth. Relative growth always, students. Relative growth always, right? Chintu ma'am's name. Oh, Chintu is ma'am's name. Okay, Chintu is more happy, student. Chintu is going to be less happy here. Only 10% increase. Okay. So here, more money here. Okay, 100% increase happening. Be happy in life. Okay, from what you have. Students, so always go for relative growth. You get a better idea. You get a perspective. Okay. Now, students. Now, all of you ready? This is one khichdi concept. This is one khichdi concept. All of you ready for khichdi right now? All of you learn this concept again one more time very well from me. Again, you never have to worry about it. Ready? All of you ready? Students, can I get some any emoji in the chat? Can I get any emoji in the chat, students? Even my brain is not braining to ask you any specific emoji. So add any emoji in the chat and I'll accept it. All of you. Ready? Students, ready all of you? Yes. Dedifferentiation, redifferentiation, differentiation. Ready all of you? Okay. Now, very simple concept. Just understand very well. Listen to me. First, what is differentiation? In differentiation, what happens? In differentiation, students, we have a meristematic cell here. All of you know, meristematic cells are capable of dividing. Yes or no? Yes. Meristematic cells are capable of dividing. Now, in differentiation, what happens, students? A meristematic cell. Let's say this one, she's one royal queen. You might have seen in Japan what happens. One royal queen, she gave her royalty and she became a normal person. Royal queen gave up her royalty and she became a normal person there. Same thing is happening here. A meristematic cell will lose the ability to divide. So, meristematic cell ability to divide. It will lose the ability to divide. It becomes a differentiated cell. Differentiated cell. This is called as differentiation. Differentiation means what? Losing the ability to divide. Differentiation. Example, students. Example is that primary xylem, primary phloem. Primary xylem, primary phloem. Permanent cell. Basically, a dividing cell became a permanent cell. Lost the ability to divide. That is called as differentiation. First is lose the ability to divide. Now what happens, students? What is dedifferentiation? In dedifferentiation, a primary permanent cell, this one, primary permanent cell will regain the ability to divide. It is now what? Regaining the ability to divide here. and again it becomes secondary meristematic cell. So here it was primary meristematic, secondary meristematic. So again it is regaining the ability to divide. That is called as dedifferentiation. In dedifferentiation, you gain the ability to divide, students. Gain the ability to divide here. Okay. So again, royalty is back. All of you agree with me? Royalty is back now. Royalty is back. She became a queen again. Now what happens, students? Now what happens? In redifferentiation, what happens here? In redifferentiation, what is happening here? We have dividing cell here. After regaining, we have a dividing cell here. It will now again lose the ability to divide. Again, mental students. Lose its ability to divide again. And it becomes secondary tissue at the end. So again, lose the ability to divide, students. So all of you in the chat, students, all of you in the chat, LGL. First, losing, gaining, losing. Ability to divide. Differentiation, dedifferentiation, redifferentiation. All of you, students in the chat, LGL. Here we have example is going to be medullary rays, which are your permanent cell, are becoming vascular cambium. They are becoming vascular cambium. Formation of vascular cambium is your dedifferentiation, because vascular cambium can divide again. It is a type of your secondary meristem. It is a type of secondary meristem, students. LGL. First, losing, gaining, losing. Losing, gaining, losing. Done. That's all, students. That's all. Students, how many of you understood? At this, after even 10 hours of marathon, your brain is working, means you should keep it in museum. We'll keep your brain in museum now, if it is still working. Okay, LGL. Okay, LGL. Now, students, see here, formation of interfascicular cambium from fully developed permanent cell. See, here we have a permanent cell. Permanent cell is again dividing to become meristematic. Here, permanent cell is dividing to become meristematic. What do you call that? We call that dedifferentiation. Students, we call that dedifferentiation. Simple as that. Permanent cell is regained and divided to become interfascicular. Dedifferentiation. In a tissue culture experiment, leaf mesophil. Mesophil is a permanent cell. It is a permanent cell, primary permanent cell. It is again becoming callus. Callus formation is happening. Dedifferentiation. It is regaining the ability to divide. Regaining the ability to divide. Okay. Students, any differentiated cell that lost ability to divide anymore, it is regaining the ability to divide again under certain condition. That is called as what? Dedifferentiation again. That is called as dedifferentiation again, students. Dedifferentiation. Regaining the ability to divide. Okay. Now, students, there is a small concept called as plasticity. Understand, students. Understand. What is plasticity? Plasticity is nothing but a plant is changing its life cycle. For, let's say, all of you throughout the 1 year you studied, but last 2 months you did not do anything. Will you be able to crack NEET? Not happen, because you have to be very consistent. You have to be very consistent. You have to keep on doing effort every single day. NEET journey is not a sprint, it is a marathon, and every single day counts, because if you are being consistent every single day, it will make a difference in the long run. So similarly, students, a plant life cycle is decided. A plant life cycle is decided now. Now, if it is deviating from the life cycle, if the plant life cycle is deviating, that is called as plasticity. Now, there are two main reasons for this deviation. Two main reasons. One is environmental, other one is phase of life. Understand, students? Understand one by one. See here. Okay. Here we have, first one is called as your phase of life. Phase of life, students. It is seen in the case of your cotton, coriander, larkspur. In the case of cotton, coriander, larkspur, students. Larkspur. Okay. Cotton, coriander, larkspur here. Now, students, in juvenile, it looks something like this. In adult, it looks something like this. Students, you might have observed. How was your hair when you were in your, when you were in second, third? I still remember. For me, it was mushroom cut. See, students, this one all was not allowed that time. Okay. If I do spikes, gone. Chop, 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 chop. Military cut I had. So when I was in second to 8th, I had a mushroom cut. Mushroom cut, all of you know. Here, the nice students, they keep all your hair down and they cut from here. They'll cut one here. That is called as mushroom cut. So in different phase of life, you look different here. Similarly, this plant also, in different phase of life, it looks different. It looks different here. Okay. Now, What is based on environmental? Based on environmental example is buttercup. Now students, you see, let's say I am both the plants, let's say in water. Both of these plants are in water right now. Tell me students, which will be more beneficial? Tell me students, which one will be more beneficial? This one. Why students? Because when the water is current is going, here very less hindrance, all the water will be able to pass through easily, but here this plant only will fly off. This plant will go off with the water. So this is going to be in terrestrial, this is the adaptation in aquatic. So based on the environment, change is there, adaptation is there. Okay, this is called as buttercup. Okay, this is called as buttercup, students. Students, I'll tell you, I had the worst time with my hair when I was in class 10th. See, all of you know how much of level we keep in class 10th. In class 10th, all of us think we are the best person in the world. In class 10th, all of us have that level to keep up. Class 10th, full ego is at the maximum in class 10th. After you go to class 12, everything becomes go down. I still remember, students, before my birthday, one day before my birthday, I went to a barber shop to get a nice haircut. That barber, okay, he had some 7 lives enmity with me. He cut my hair so short, you can see my brain. He cut my hair so short, he almost made me bald that day and he sent me back home. And as a matter of fact, when I was getting the haircut, it was looking nice. I'm like, oh, tomorrow I'll have birthday bash in the school. I will go enjoy. I come home, I take a bath and I see there is not even one strand of hair on my head. I can see my scalp there. He has made it that short. And I was fighting in my house that I will not even go to school. I will not even go to school for 10th standard. I was that mad at the barber. Okay. And I really wanted to go to that shop, throw some eggs and run away. But I was too scared again. Too scared to do that also. Okay. So, hair, students, hair on next level stories are there for hair. Now students, now. Coming back to the topic here. Students, tell me the answer. Students, tell me the answer. Which of the following plant does not show plasticity? See, cotton, coriander, buttercup shows. Maize does not show. Some odd one out here. Okay. The ability of plant to follow different pathway in response to environment leading to the formation of different kind of structure is called as what? Plasticity. It is called as what, students? Plasticity. Okay. Now students, development is called growth plus differentiation, right? Here we can see there are extrinsic factors, water, oxygen, temperature, nutrient, light. All of them play a very crucial role. But students, there is something called the intrinsic factor from the inside. Okay. Here we have intercellular and intracellular. Intercellular we have PGR, intracellular we have genetic factors. Genetic factors we cannot control. PGR also we cannot control, but we can stimulate externally also. We can give this externally also. External. So what is PGR, students? PGR is nothing but plant growth PGR, plant growth regulators, okay, and it is also called as phytohormones or PGR or plant hormones. Here we have 3 main categories here. Okay, students, similar to how hormones are produced in the body in very small quantity, which can control all physiological properties, these phytohormones can also control that. Okay, here we have something called as auxin, gibberellin, cytokinins for promoter, ABA, abscisic acid for growth inhibitor, dual nature for ethylene. Next level, this ethylene is ultimate boss here, students. Ethylene can do both the functions here. Okay. Now, students, all of you know entire thing. Let me just show you what to study here. Okay. Now, for auxin, what they can ask you, students? For auxin, they can ask you the chemical nature. They can ask you precursor by chance if they ask you. Then they can ask you the example of this one, NAA synthetic. They can ask you 2,4-D. Remember, students, 2,4-D is also used as what, students? It is also used by as a weedicide, to kill the weeds. Okay, to kill the weeds. Okay. Then, students, they can ask you discovery here. Francis Darwin and Charles Darwin, discovery. Then they can ask you, students, which topic did they do? They did on phototropism here, plant canary grass. They found that when a plant is growing, it will grow towards the sunlight. A plant will always grow towards the sunlight. Okay, and they found that, students, in the tip of this plant there is something present here which is able to guide it towards the sunlight here. Okay, now isolation by FW Wendt, plant Aviena sativa, bioassay Aviena curvature test. Okay, all of these are functions, students. All of these are functions here. Initiation of flowers here, root initiation, very important. Cell enlargement, cell division, feminizing effect, apical dominance means it will always grow tall here. Now, prevention of abscission, it will happen only in the case of students, young plants. Prevention of abscission will happen only in the case of young plants. But if the plant is old, it will say go die. It will say just go die, I don't care about you. Okay. So if the plant is old, it doesn't prevent. It will make sure the leaf falls itself. In the young plant, it will prevent. See, young people we need. Okay. Now students, see this question here. Phenomena in which influenced by auxin also play a major role in discovery. Phototropism. See this question, students. That's how the question will be asked in examination also. For auxin also, students. Same thing goes. Now, gibberellin, what happens, students? Gibberellin. Chemical nature, terpenes or terpenoids. It's a PYQ right here. Precursor acetyl-CoA, malonic acid, they might not ask you. Discovery, Kurosawa. How, where was it discovered? It was discovered in the Bakane disease or foolish disease. In this plant, there was a fungus growing. The fungus is called as Gibberella fujikori. Because of this fungus, gibberellin was released and this plant became very tall. Very tall plant with no brain. What do I mean by that, students? Very tall plant but no rice only in it. No rice only. So it's Bakane, foolish disease. Okay? Foolish disease. Students, this is very important again. We have 100 different types of gibberellic acid. GA3 is the first isolated one. GA3 is the first isolated type here. Okay? Now students, here this is very important here. Stimulation, stem growth here. Promote seed germination. Bolting in rosette plants here. Delays senescence. Bolting means what? It will make the plant grow very tall. What all here? Again, all of these are PYQs. Every single one, students, every single one is a PYQ, and I'm telling you, students, question will be asked from here itself. If they ask you from gibberellins, question will come from here itself. I'm telling you now only, students. Okay? Students, don't remind me of gingerly oil now. Don't remind me of gingerly oil. See, speed up the malting process. Spraying of juvenile conifers with GA3 hasten the maturity period. Increase in stem length, that is in sugarcane. Right? Increase in the grape stalk length. Okay. See this question. Spraying sugarcane crop with which of the following plant growth regulators increase the stem length? Gibberellins. Gibberellins. Spraying of the following phytohormones in juvenile conifers? Gibberellic acid. NEET PYQ, students. Plant hormone is 240. We decide. Auxin, that is. Then cytokinins. Then the cytokinins. Chemical nature, purine derivative, adenine. It is a PYQ. Adenine derivative, students. Discovery, Skoog and co-workers. Isolation, Miller et al. Okay. He is a Skoog, students. Now, what did they observe? They were trying to grow a plant in coconut milk with a normal entire broad this tissue culture media here. They noticed that whenever they added coconut milk or yeast extract or your vascular tissue extract, the plant was able to grow very well. Then they identified something is common in all three. The something which is common in all three was nothing but cytokinins. And cytokinins help in cell division. They help in cell division, students. Cell division. Okay. And students, they isolated from the herring sperm, that is kinetin. Kinetin was synthetic, students. Kinetin was synthetic in origin. Then students, then they observed something called as zeatin later on. Natural substance, zeatin later on, students. So main function is cell division. Main function is cell division here. So all of these are functions of cytokinins. Cell division, modifies apical dominance and promote lateral growths. So can I tell, students, can I tell cytokinins is actually against auxin here? See, auxin will do what? Apical dominance. Cytokinins, lateral growth. Yes or no? Yes. So it is actually against your auxin right now. Delays senescence, obviously. Promotes nutrition mobilization. Promote chloroplast development. Helps produce new leaves and adventitious shoots. Okay. So it is actually against the auxin right now. See here. Adenine derivative. See, question will come like this. 2022. Derivative. Cytokinin. Okay. Now students, we have ethylene right now. Okay. Okay. Students, I want all of you to revise this one more time today itself. I want all of you to sit and revise all these functions today itself, because you realize, right, time is, let's say I finish the class in next 15 minutes. Take a 2 hours break. Before you sleep today, before you go to bed today, right, just sit back and download this PDF and just read once. All of you promise me, students, promise me. So all you need to do is, students, just open this PDF today, go back to this part and just read once before going to bed today. This particular chapter as well as your chemical coordination at 10 o'clock, let's say 10 p.m. Today, 10 p.m., just open this PDF and just read through once before you go to bed at 10 p.m. or 11 p.m. whenever you go to bed. Okay, do not go after 11 because I know all of you are very exhausted today. Okay, so at 10 p.m. just go through this once, just read this once, just glance it. It'll stay better. See, all of you might be aware, whatever you read before the bed, going to bed, right, that stays much stronger in your head. So try to revise this and chemical coordination before you go to sleep today. Okay, all of you promise? Yes. So here we have ethylene, students, gaseous hormone. Precursor is methionine here. Discover H's cousins. Okay, cousin people are discovering what to do. Okay, breaking seed dormancy, initiate germination on seedlings, swelling of emineral axis, apical hook formation. Shoot, break seed dormancy, sprouting in polar tubers. Very important, students. Internode and pedal elongation in deep rice water plants. If this is not given, then gibberellins. But if it is deep water rice plants, then ethylene. So when you say internodal length increase, please read the question carefully. If they have given you deep water rice plants, then ethylene. Then we have to go for ethylene, students. Okay. Now again, students, see this. Root hair formation. If it says root initiation, then auxin. Root initiation, auxin. But if it says root hair formation, then ethylene. So please read very carefully. You might go wrong here. Students. Okay. Induces flowering similar to your auxin, pineapple and mango. Promotes female flower development. Okay. Ethophone for ripening. Okay. Senescence and abscission, again, killing. Both effect is present. See here. Internode of PTO elongation, deep water rice plants. You might go for GA3, but GA3 is not the answer. What is the answer? Ethylene is the answer. See this question. Used in plants, gaseous hormone, killing of dicot, that is auxin, speeding of the malting process, gibberellins, promote root growth and root hair formation, ethylene. Overcome apical dominance. Students, can anyone tell me? This can be cytokinins as well as ABA. Okay. Cytokinins is better actually then. Students. See here. Production of cucumber has increased manifold in recent years. Application of which of the following phytohormone has resulted in increase in yield as hormone is known to produce female flowers. That is going to be your ethylene here. Feminizing effect is going to be ethylene. So students, they deliberately did not give auxin here. See, auxin is not given here. See this. Okay. Last is called as ABA, called as abscisic acid. ABA, students, discovery is very funny. Here we have three scientists who were independently working. One scientist called it abscissin 2, one scientist called it dormin, one scientist called it as inhibin B. Now later on, students, these three scientists got to know all three of them are working on the same chemical itself. Okay, and they together call it as abscisic acid here, students. Okay, chemical nature is carotenoids here. Now what is the function here? Increasing tolerance to stress, general plant growth inhibitor, inhibitor of plant metabolism, yes, seed development and maturation, promote seed dormancy, like you stay quiet, it will promote dormancy. So it is against gibberellic acid, students. Stimulation of closure of stomata during stressful condition. If there is a stress, it will change it. Okay, close it. Students, here is the entire list of summary of all the phytohormones. Auxin functions, gibberellins function, cytokinins functions, ethylene, all the important functions is given. Students, it is given in one slide right here. All the main functions, students. Everything is given in one slide. Okay, students. So with that, chapter is also over actually. Students. Now again, I would all of you want to open your NCERT before this because there is one small 2-3 lines are missing here, especially the walnut one. Remember walnut and everything. So that point is missing here. So just check once that also. Okay. Students, solve this question. Use the remaining brain cells, students. Use the remaining brain cells and answer this question. I'll upload it today itself. Use the brain cells and solve this question quickly. Ethylene, senescence, cytokinins, ABA, gibberellic acid. Students, use your brain cells and solve this question quickly. Use all the brain cells and solve this question, students. Done? Done, students? Done and dusted, out of this world. Done and dusted. Quick answer. Abscisic acid. And this is the end of the class right now, students. Again, I'm telling you, students, we are coming to Chennai on when, students? When are they coming to Chennai? Tell me in the chat, all of you. We are coming to Chennai on 6th of May. After your NEET examination, when the exam is over, you can relax and meet us. So we didn't want to keep it before. We want to keep it after NEET examination. So all of us, you can meet us very well. Chennai, Anna Nagar. Timing will tell you, students. Timing will tell you. Now, students, if you are anywhere in Hyderabad, if you're anywhere in Bangalore, again, we're reminding you, students, all of you, we have, if you are a 2026 aspirant, next year aspirant, or if you have any juniors, also, I want all of you to tell your juniors, right, that Vedantu offline center is available there. Please visit it once. You will see a real change in the offline also, students. Okay. So, friends, how many of you attended the entire 10 hours? Tell me honestly in the chat, students. Honestly in the chat. All of you. Students, Bangalore, we will see. We are in Bangalore all the time. We are in Bangalore all the time. No, friends. I'm in Bangalore right now also. Okay. I will tell Gopika ma'am to upload the notes. Don't worry, students. So I want every single student to comment in today's video. How was the 10 hours marathon of human and plant physiology, students? I want everyone to tell me in the chat. Okay. I want everyone to tell me in the chat right now. Not right now. In the comment section. How was the 10-hour marathon from our side? We did not want to do this actually in the starting. Then we decided, students, all of us, we were discussing. Some of the students told us, please do it. So 10 hours, we'll sit 10 hours. We'll finish the entire human and plant physiology in one go. And here we are done, all right, students. Okay. So bye-bye, students. Take care, all of you. And I will scoot right now, students. Again, I'm reminding you, if you have any juniors who are going to write 2026 aspirant, ask them to check out right now the entire Vedantu offline center. 40 to 50% off is going on there. So ask them to visit it once. They'll see a game changer. We have in Hyderabad, we have in Chennai, we have in Bangalore, JP Nagar, right? So students, thank you so much. Bye bye. Until then, take care. Next class, I will see all of you tomorrow again for one mega question practice. See, tomorrow we will have question practice. I want all of you there. Okay. So bye bye, students. Bye bye. Take care, all of you, and sayonara. Boy, all of you. Boy.